Right.
Yes, you can start, Kshitija.
A very good evening to everyone connected to us in India, and a very good morning to all our attendees from abroad. I welcome you all to a two-day virtual centenary conference organized to celebrate the establishment and success of the Department of Biochemistry ISC. On this day, 100 years ago, our department was established in ISC, and as we step into the 101st year, we bring together all the past and present members of the department, distinguished speakers and alumnus to cherish the moment. So a very happy 100 years to us all. I welcome you all once again. Stay connected with us for the 2 days of science and history, achievement and efforts. With this, I request our department chair, Professor Rangarajan, to inaugurate the conference with a welcome address.
Thank you, Kshitija. So good evening and good morning to the overseas participants. Good evening to the Indian participants. I welcome you all for this two-day symposium. Today actually marks exactly 100 years since the Department of Biochemistry was established. So we are at the last day of the 100th year. Tomorrow we're going to step into the 101st year. So to celebrate these two occasions, we have organized this two-day meeting. To facilitate participation of our overseas alumni, we have scheduled it for the late evening. I hope our alumni will be able to join. Just to be 100 years, out of established. So we are please unmute. 100 years. No,
some audio problem. Yeah.
Echo.
Some audio related issues.
Okay. Now it's okay. Just to recapitulate that the centenary celebrations of the department actually began exactly 1 year ago on February 20th. We had an inaugural celebrations, which went on rather smoothly because 1 week after the inauguration, the COVID embargo started and then lockdown and all those things started. So we are very, very lucky that the inauguration happened right at the nick of the time. And we had an excellent inauguration for the 1 day. We had an inauguration symposium. We had an exhibition, and then we had an evening gala dinner. Many people, alumni both from India and abroad participated. It was really a memorable occasion, and I thank all of them who participated in this inaugural meeting. The following inaugural session, we are supposed to have a international conference and an alumni reunion in December. Unfortunately, because of the COVID, we had to postpone it. Today and tomorrow, actually, we're supposed to have a concluding session of the centenary celebrations, but instead, we are going to prolong this. We hope that the COVID outbreak will disappear, and by December, things will be okay. There will be people traveling, and people will be able to move around, and COVID will probably go away. So we will take a call sometime in May or June to see whether we can actually have a international conference where many of you can actually come over and cherish some of your memorable moments that you spent in the department as a graduate student. I would like to mention here that a lot of effort has gone in the last year to make sure that this centenary year is a memorable one. Faculty, students, alumni, and many well-wishers have made excellent contributions and excellent cooperation to make this a memorable event. I especially would like to thank G.N. Rao and Professor Deepankar Nandi, who painstakingly gathered all the email IDs and built an email directory of the alumni, without which you would not have been able to contact many of the alumni. The list now stands around 650. Still, many of the emails are bouncing back. So I appeal to the alumni that if you don't hear from us or if you're not receiving our emails, please contact us so that we can actually
Without speaker, people are saying that. Yeah, no.
Okay, can you all hear me now?
Yeah, it's better.
Okay, I removed my headphones. So I appeal to the alumni that please update your email IDs, share with us so that we can update this email directory. And communication between the department alumni will not be just a one-time event during the centenary. We would like to continue over the years so that we'll have a active dialogue, and would like to reach out to all the alumni. Would not like to miss out anyone. So I sincerely appeal to all the alumni, please do not mistake if we are not reached out to you. It is purely because we don't have your email ID or many of the emails are bouncing back. Please send us about the correct email ID. We certainly would like to share many of the events that's going to happen, not only this centenary but also in the years to come. And I also would like to thank IIS Jana for their unstinting support. Many of the donations were actually channeled through IIS Jana. We have received all the donations. Just to update you that so far, from the alumni, sponsors, government agencies, institute, all the donations put together, we have received about 73 lakhs. We have spent about 20 lakhs for the inauguration ceremony, and we still have with us 53 lakhs, of which one alumnus contributed 20 lakhs on his own. And I'm also happy to share with you that we have in the audience another alumnus who has now come forward to establish an endowment in the honor of his PhD mentor. I would not like to reveal the name now. We are in the final stages of finalizing all these details. And there will be an award instituted to recognize and reward PhD students who have spent about 5 years. As you know, PhD students are our backbone. They contribute to most of our research activities, and this endowment interest that is accruing
हाँ भाई इसीलिए सर
will be
देख तो मैं
used to
रूम पे
reward
कौन
PhD students who have completed at least 5 years. So I thank the alumnus. I will reveal more details once everything materializes. So over the last 1 year, we also had 2 centenary lectures by Nobel laureate Jim Allison and Dr. Doug Bushland from UC Berkeley. For about 500 people participated, both through YouTube and Zoom in these 2 meetings. I also would like to take this occasion to thank all my faculty colleagues, students, alumni, well-wishers, and the IIS administration, the past and present director, and the past divisional chairman, Professor Umesh, who is right here, and the current dean, Professor Usha Vijayaraghavan, for the outstanding support they have extended to celebrate our centenary celebrations. I'd like to mention here that all the alumni who are here at this meeting very fondly remember the Biochemical Society. It is an integral part of our PhD journey in the department. Now the Biochemical Society, or PICS as it used to be called, has now become a full-fledged cooperative society, answerable to the Register of Cooperative Society. It has permanent office bearers. It has a Twitter account, BICS_IISC. So if you're not receiving update from us, just go to the Twitter and join the Twitter account. You will get updated on what is all going on in the department. So I thank the big students who have actually set up all these accounts and are playing a very active role now. I think with this, I would like to conclude the one important part of this inauguration of this symposium is that Professor G.N. Rao and Professor Utpal Tatu have now brought out a coffee table book. And I would like to welcome Professor Utpal Tatu to inaugurate this coffee table book and say a few things about it. Unfortunately, it has to be done on the electronic manner, so he will probably share give you a glimpses of some of the key aspects of this coffee table book. And before I welcome Utpal Tatu, I would like to thank all the speakers, all the chairs of the sessions, and all the participants for accepting our invitation and participating and agreeing to give talks as well as listen to the talks. Thank you very much, and I hope you will enjoy this good. Professor Utpal, please.
Okay, thank you. Thank you, Professor Rangarajan. Thank you for giving this opportunity to talk about the coffee table book. I'm going to take 5 minutes to describe what we have done to capture the magnificent 100 years. Am I audible to?
Yeah.
I'm okay. All right.
Yeah.
Okay. Right. So I'm going to share my screen first, and so just give me a minute because yeah, and right, there you go. Okay, so yeah, such a wonderful pleasure to be connected to the community of Indian Institute of Science, Department of Biochemistry all over the world. I'm sure large number of people have tuned in and are looking forward to hearing about the progress and all the developments that have happened in the department. And Professor Rangarajan nicely summarized some of the activities that we have done earlier this year to commemorate 100 years, and the ongoing activities that are in progress and will continue later during the year to celebrate the centenary. We apologize that the audio wasn't particularly good in the first part when Professor Rangarajan was talking, but I'm sure he will email and send you a gist of what he speech subsequently. It's a pleasure for me, and thank you, Professor Rangarajan, for giving us this chance to talk about the coffee table book. While I'm going to present some of the glimpses of the coffee table book to celebrate the 100 years of the Department of Biochemistry. It's really the team who has made this effort. So before I start, let me just let me show you a little bit about the book and about the contributors. I hope my audio is okay. Once in a while, just confirm if everything is, people are.
I can hear well.
It's
Okay,
very
great.
good.
So, as you can well imagine, it's an impossible task. Really, it's an impossible task to capture what's gone on in 100 years in this great scientific institution, in this great department, in a few pages. You can imagine the challenges that one would face in capturing the excitements, the scientific advances, the history, individuals, students, scientists, support staff, administration, and all of and whatnot, and the visitors and the connection. So it's a humongous task, I think. But what I think what we have tried to do is to capture glimpses, some of the most important or some of the more important moments in the history of this great department in the last 100 years. What my personal thoughts on this topic are that such an honor, really, such an honor for all of us to belong to a department which has this huge, this vast history of 100 years. How many departments in this country can claim to have a history of 100 years? Yes, and that's such a big privilege for us to belong to a department which we can say that in 100 years ago we existed, and this is what was going on in the department. And this is really the passion that we have tried to capture in the book that we have put together for you. And what you are seeing on the screen is really the most precious look of the department that we all identify with. That is the front end, the facade of Department of Biochemistry as it existed from its inception in 1921 to about 10 years ago. You can see the picture. That's really the first page of the book, the front cover of the book, the picture of Department of Biochemistry and capturing the greenery around it. This particular department and the building still exists, except many of you know that we have now moved to integrated biology building, and many of our labs are now situated there. But once in a while, out of nostalgia, all of us pay a visit to this department, to this building, and travel along the memory path to think about the old moments. So this is really the cover page of the book, Department of Biochemistry, Celebrating a Century of Excellence is the title that we have given. There were many suggestions that were given for the title, but we thought Celebrating Century of Excellence would be something that would capture all the moments of excitement in a concise manner. Next slide, I'm showing you the last page, really. I'm showing you the back cover of this book, which actually shows you the grand central building of Indian Institute of Science. Now, while this building is used on many occasions as to show the ambience of Indian Institute of Science, and it's there all over the world. In Google, you type IIC, and right away, hundreds of different versions of this book come together. But for us, there was a specific reason why we chose to put this as the back cover of the centenary celebrating book. Many of you may know actually that even before the Department of Biochemistry started as an independent building, even before that, the original Department of Biochemistry actually began in one of the.
We are still on only the first page. Is somebody moving the slides?
Sorry, we can't hear you properly, Rangarajan.
No, we are still in the first slide.
Okay, I still can't get you.
Or you are doing it yourself?
The first page. You are still in the first page. Yeah.
Okay, I moved to the last page, but
no,
let me.
Not moving.
Utpal,
Yes.
also can you disable annotations? There are some marks coming up.
Oh, okay. All right. Screen, black screen, white screen. Well, let me just show this in this manner. Okay, annotations. Now are you able to see is the last page?
No.
No, I'm going to stop sharing and share again.
Yes, maybe that
I think you shared a PDF file, Utpal.
All right,
Probably
that
that
might
would-
be
Yeah,
even
that
better.
explains it.
That was going to happen, but I wanted to actually show some things. So maybe let me go back to this. And sorry about this. We are, yeah. Are you able to see the last page now or not yet?
Yes.
Yeah. So now this is the central building, the photograph, which I think I don't need to really show. Everybody knows about it. But the reason we chose this as the last cover is because Department of Biochemistry actually started. The genesis happened early on in one of the buildings, one of the rooms in this central building. That's the reason we sort of tried to bring this as the back cover of the book. Moving on, I just want to get back to the book. As Professor Suma suggested, I should actually show you the PDF. But before that, I want to just share with you the people who have done all the work while I'm talking about the book. But of course, it was a team. Needless to say, I mean, it's obvious that this kind of a humongous task can be done only by a team of scientists and people who help. So Professor Rangarajan was the driving force. He had the idea, along with Professor D.N. Rao, that we must do something of this nature. And he really gave us a free hand. He said, do implement your ideas as you like, but his support was so immensely important because there are so many aspects of it, the finances, permissions, and nowadays even government permissions to actually have a coffee table book. But he was fearless and said, no matter what, I will support you. Go ahead and make the best book that you can and leave the rest to me. And that's how we have this wonderful book with us. And I think some of the hard copies are there where Professor Rangarajan and Professor D.N. Rao are sitting. So they will show you in a moment. But before that, I just want to say that Professor Rangarajan gave us the freedom to do what we liked. But most important was Dr. Narmada Khare. Dr. Narmada was recommended by the Office of Communication at Indian Institute of Science as the senior editor. So she was given as a help to the Department of Biochemistry. And what a wonderful pleasure it was for us to have help from Dr. Narmada. Not only was she an excellent writer and editor, but now I can doubtlessly say that if there is anybody in the whole world who knows enough about Department of Biochemistry, it's not the members, former or past or current, but it is Dr. Narmada, because she has done so much reading about the department. And for the last more than 6 months, we have been interacting, all of us as a team, so closely, and we could really see the excitement that she carried about what has gone on in this wonderful institution and in this department for the last 100 years. And she has tried to put her little personal touch in. Not only, I would say her job was not just as an editor, but at a personal level, she was emotionally involved in development of the book. Thank you so very much, Dr. Narmada, for all the help that you have rendered. And I think she's joined us online as we speak and on the Zoom link. I will not spend too much time, but of course, Professor D.N. Rao, I think, not just the coffee table book, but the whole centenary celebrations. And I would say that almost anything that happens in the department, the first name that has to be Professor D.N. Rao. He's always spearheading the efforts. He's always ahead. He's always the most excited. He always has more ideas than anybody can handle. And that's really was the case also with coffee table book. He was really the driving force in terms of how to get people together, how to get data together, and most importantly, how do you get data of people who have been in the past, 1921, 1925, 1930, 1940? How do you get? And immense network that Professor D.N. Rao has. He has connections with almost everybody who was associated with the Department of Biochemistry in the past. And that was really the reason why we could actually access people as well as photographs. And I saw the kind of collection he has of photographs of the department and people.
We're running out of time.
Yes. I'm really coming to an end, but I think that was really a driving force for us. And then myself, together with the new faculty, now no longer new because they're all, now they are newer faculty, but Professor Prushat Rajaguru and Professor Sandeep, who actually did all the hard work already at the time of putting together the exhibition. So all the information that they gathered by several visits to the Office of Communication, to the library and on the internet, amazing research that both have done, which is what served as the raw material, the core of the book. And that's really how we have in front of us a wonderful book that's there. And needless to say, any activity that happens in the department cannot happen without involvement of students, and bunch of students. In fact, a large number of them, we are just putting few names here, but some of them are students of the department. And I think Neetu, Ranjini is of course from Office of Communication, but Sangeeta, Vinitra, and then of course Gayatri, they played very important role, but many others. Now the writers list includes people who actually wrote small articles. Neetu actually made sketches that are there in the book, but also most others, Sangeeta, Vinitra, and Gayatri did a lot of writing, which makes important parts of the book that I'm going to very quickly show you before we wrap up, because the scientific sessions are going to start. So here's the book. Can you all see the book, that PDF?
No.
No?
No. We only see the PPT.
Okay. I'll have to do that 1 second. And all right, there you go. This should work. Can you see the book now?
Yes, sir.
Of course. So this is the sort of the PDF of the book. You can see on the left side the cover page, and then I'll just browse through some of the pages. Second and third page, Department of Biochemistry, celebrating a century of excellence, and then the foreword, beautifully written by none other than Padma Bhushan Professor Padmanabhan, who gave his personal touch because he's been associated with the department for such a long time. So on the right side, you see the foreword that's almost 2 pages. And again, what a wonderful, how nicely has condensed the history of the department in brief manner, and personally signed by him, the foreword, which really condenses excitements of 100 years. Thank you, Professor Padmanabhan, for taking the trouble and putting your thoughts into this wonderful foreword. As the time is short, I'll just very quickly show you that the important things that the people will want to see in this book is what is not visible today, and that is the early years, 20s, 30s, and wonderful people early on. Gilbert Fowler, the first chairman of the department in the British Times of India. He was the first chairman and pioneered today, again, it has become such a hot topic, water sanitation. He was a sanitation biologist, and in 20s and 30s, he brought that concept, and he sort of carried out this kind of research at the Department of Biochemistry, followed by Professor V. Subramania. In 30s, already in 20s and 30s, nutritional research. He was a nutritionist who did some outstanding, wonderful scientific work at this department, and some excellent scientific papers on this topic. I'm sorry I'm moving a bit fast because Professor Rangarajan has already indicated we are running out of time. So just taking you through decades, 20s and 30s, then in 40s, M. Vishweswaraya, wonderful scientist who did some phenomenal work in the area of enzymology and fermentation biology. What this particular page, which is on page 10 and 11 and 12, shows you is the old building with some of the old members and the old labs, where you can see one of the first sequencing gels that Professor Padmanabhan is pouring, and this level at which we were at that time. Of course, as a professor, he was still doing experiments. We all know his passion for science. And on the left, you see Professor D.N. Rao pouring a polyacrylamide gel, or loading a polyacrylamide gel, again, capturing the protein and molecular biology history of this department. On the right, you see now the modern laboratories in the new biology building, how they look for those of you who are joining us from abroad, and the current members of the Department of Biochemistry, which includes, of course, honorary professors, as well as the ongoing faculty. Professor Muniappa, of course, been the chairman for so many years. Professor Jay Bhaskaran, before Professor Rangarajan, he was the chairman. Professor D.N. Rao, Professor Savitri, and then, of course, all of us here. And then the 4 new faculty. The most important focus will have to be on this. They've recently joined. Just about a few months back, 4 of them have joined. Dr. Devbrata Raha, Dr. Mahipal Ganji, Dr. Sarvanan Palani, and Dr. Kesavardhana. They've been now the future of the department. And then, as you can see, some nice sort of artwork that captures some of the research that had gone on in the department. I'll not spend too much time, but in 40s, then Professor M. Vishweswaraya, so many nature papers. Altogether, the Department of Biochemistry had more than 30 nature papers. in the early part of the history of the department. But Professor Srinivasaya, of course, has huge legacy that our department follows. But as I said, fermentation biochemistry and enzymology were one of the most important things. He shared editorial board of BBA with likes of the Nobel laureates and some people whose textbooks we read today. Professor Srinivasaya was counted one among them in 30s of the department. Professor K.V. Giri, of course, came up with this concept of paper chromatography, circular paper chromatography, that is nicely captured with some original images in the book, which I think is an inspiration to all the modern day chemists and scientists, that some original thinking has gone on in this department. And I apologize for going a bit fast, but in one of the most important other things that happened in 50s was, of course, Professor M. Subramania actually is credited with identification of chromosomes in yeast, Saccharomyces cerevisiae. It appears that he was some of the first observations on chromosomes in yeast took place in this grand department, and some of his original drawings are part of this book. Yes. And this, of course, the microscopic images of the nuclei showing, with Fulgen stain of Saccharomyces cerevisiae way back in 40s and 50s. I'm going to jump and say that in 70s and 90s, then, of course, that's the era when wonderful research on protein biochemistry, enzymology, molecular biology, all of this really took place. And in late 90s, then, of course, now we have seen DNA repair is really the fashion. That's where Professor Muniappa, Professor D.N. Rao established fantastic foundation, and that some of the new faculty have taken it forward. All of that is captured. I'm just flipping through some of the pages. Here, again, Professor Subramania with his child, the Padma Shri that was given to Professor Subramania back in 50s. Professor Sharma's work on neuroladrism, which was one of the sort of nutritional problems that I think in Kesari dal, there was a contaminant that he identified. And then many new institutions such as CFTRI, which were founded. Then, of course, the startups that came out of Indian Institute of Science in the last 10 or 20 years, those are being captured in various pages of this department. Professor Suma's startup and her work on computational biology, which is a strong force in Indian Institute of Science now and in the country, that's highlighted. And how can you miss the most important contribution of development of vaccines by Professor Rangarajan, the current chairman. He developed for the first time out of academic institution a product which is out there in the market. Thousands of doses have been sold, and what a great contribution in contemporary times, which is all narrated in this book. Finally, Professor Padmanabhan's book of contributions start right from the beginning, as I said, as a foreword, but also his science on malaria research, his anti-malarial drug discovery, role of curcumin, and his experiences with making curcumin a potential drug for malaria are all narrated beautifully in this book. I'm just here to highlight that, of course, the department has huge history. Professor Ram Sharma, Professor Mahadevan, Professor Kumar, and then wonderful collection of pictures of students and staff. With that, I just want to sort of come to the last page. There's so much, but the inauguration already happened for this for the centenary, and those pictures from last year are there in the book, which I think will also serve as very important historical perspective, and then the timeline from the time that it was founded to currently important events in the historical backdrop of what happened in the country during those years. Ending with the pictures of all the scientists, 59 odd scientists that we've had, 59 faculty whom we've had, including the 4 recent ones. We were able to get most of them, but a few we were not able to get. And with that, I want to conclude, but by saying that it's a department's work. Everybody contributed immensely. And I think the book is now available. The hard copies are there with Professor Rangarajan. He'll show you on the video, but shortly we'll be able to send you some books across. And with that, I want to thank again, all of you for your kind patience. I've tried to do some small justice to this book, and thank you again to everybody who made this task possible of creating a coffee table book to celebrate the centenary of Department of Biochemistry. Thank you very much. Back to you, Professor Rangarajan.
Kshitija, over to you.
Yeah. Thank you, Professor Rangarajan and Professor Tatu. I would like to request my colleague Sheetal to coordinate the first session.
Okay. Thank you, Kshitija. Once again, I'd like to welcome you all to the virtual centenary conference of Biochemistry Department. Before starting the session 1, I'd like to request our speakers and session chairs to adhere to the timings. Apart from our centenary and inaugural lectures, every talk will be of 25 minutes, followed by 5 minutes of question and answer session. Attendees can verbally ask the questions on Zoom or post the question in Zoom YouTube chat box. Session chair will coordinate the question and answers either by allowing the attendee to ask the question or read out the question from the chat box. We also request our speakers to disable the annotations from View Options tab after sharing their screen. So starting with the session 1, I'd like to welcome Professor M.R.S. Rao, our first chair for this session. He is a distinguished alumnus of the Department of Biochemistry IISc and well known for his work on chromatin biology. He was a faculty in our department and also served as chair for several years. Currently, he is an honorary professor in JNCSR Bangalore. Sir, you are most welcome to start the session 1.
Professor Rao, you're muted.
Okay, it's okay?
Yes, yes.
Okay, thank you. Thank you, Professor Rangarajan and Ian Rao for asking me to chair this first session. The inaugural lecture will be given by Professor G. Padmanabhan. I don't think he needs too much of an introduction. He's one of the most distinguished alumnus of the department. And also he started as a student in the department and grew up to the status of the director of the institute. And also he has contributed very significantly to the growth of biotechnology in the country. I think he's one of the few people who has devoted all his lifetime to nurture and promote the biotechnology industry in the country. He also received the Padma Shri and Padma Bhushan from the Government of India. I'm happy to share that I'm one of his mentee. I'm very happy to request Professor GP to give his lecture on the heme story. I spent entire life on the heme metabolism, both in the animal systems, but also the parasite system. I think he's going to share with us the journey of his almost 50 years on the heme story. Professor Padmanabhan.
Thank you, Professor Rao. Sandeep.
Yes, sir. I am sharing the screen.
Thank you. It's great pleasure and honor for me to have been invited to give this inaugural talk. It's even a greater privilege to give the lecture chaired by your own first student, Professor M.R.S. Rao. Thank you, Professor Rao. Also want to thank Professor Rangarajan and Professor D.N. Rao and the conference committee to have given me this opportunity. I joined this department in January 1961. That would make it 60 years and maybe 1 month or so. So, such a long period, you tend to do so many different things, small projects, short projects, long projects, many things I have done. Of course, Professor Rao mentioned about my involvement with industry, biotechnology. So I was looking for tell you briefly about the science I have done. And among the many projects, as Professor Rao rightly said, Heme is one connection which has a theme, which has a continuous theme award. So I thought I will share this. Maybe I should make up for the last time, 45 minutes. I will try to wind up my talk. My guru was Professor P.S. Sarma, Padmadri Subbaraya Sarma, and he gave me enormous freedom to do my research. Next slide. And we started with many projects. The one of consequence is the heavy metal toxicity in Neurospora. Next slide, please. In particular, I found if you have cobalt toxic Neurospora, allow 50% growth of the organism, it will precipitate typical iron deficiency. And among that, the characteristic feature was the secretion of a peptide. These are all siderochromes, desferrioxamine. All that you need to do if you grow in cobalt toxic condition, add a drop of ferric chloride, the whole media will turn red because it will form the iron complex of this very coprogen will form coprogen. And in fact, I wanted to do biosynthesis of this molecule. It has 3 hydroxamate bonds, N-hydroxyarnithine, and I needed labeled ornithine. And no question of getting radioactive ornithine because my grant for the whole year was 5,000 rupees. Next slide, please. So I thought I will skip all the applications which I have done. Next one, please. I'll stick to the basic science part. So what does coprogen do? That is the iron complex. It has an enormous binding affinity for iron, 10 to the 30. EDTA is 10 to the 11 or 10 to the 10. But interestingly, the fungus can use the iron because it reduces the ferric to the ferrous, and the iron is incorporated into protoporphyrin and forms heme. And that is why I thought I will rather start with the heme. Next one, please. Next slide. We had the first JBC paper with Muthukrishnan. All Sarma had a very untimely death. Which let me have his lab, acquire his lab, and all his students and colleagues. We had the first paper in Journal of Biological Chemistry. This is the entire pathway of heme, succinyl-CoA plus glycine giving ALA, and ALA dehydrogenase giving porphobilinogen. Through a series of porphyrins, you have protoporphyrin and iron is incorporated. And interestingly, we found those days, the concept was the first enzyme is regulated by the end product, heme. But here we found the second enzyme, ALA dehydrogenase, was regulated by the penultimate product, protoporphyrin. So if you grow in iron deficiency or cobalt toxicity, you will not see this enzyme. But if you add iron, there is a huge induction of ALA dehydrogenase. And we had this model proposed that the iron will be donated to protoporphyrin and form heme. Protoporphyrin is the co-repressor, apo-repressor forms the repressor and represses the dehydrogenase. And if you add iron, protoporphyrin is converted to heme and the repression is released. And this was our first paper in JBC. Next slide, please. Then I really wanted to get into the multicellular function of heme and also move away from the fungus to higher organism, that is the rat. Next one, please. And that was the time, 1973. I went to Murray Rabinowitz laboratory in Chicago, where he was working on cytochromes. But he asked me to work on tRNA genes in yeast, but I didn't mind any project because I wanted to learn techniques basically, but I didn't take any of my project. But I learned all the molecular biology techniques from Murray Rabinowitz's lab. He had muscular dystrophy, you can see him, but still he would come to the lab and he was a great inspiration for me. With all his defects, physical defects, he was still attending to science. Next one, please. That was his wife. And, at the end of the year, Murray said, why are you going back to India? You stay here, we'll give you the same job you're holding in Bangalore. Then I said, sir, I have a lab in Bangalore and I want to build it up. Please help me. And he was so generous and said, I appreciate your sentiments. You can come to my lab anytime. And I literally made use of that offer and I went to his lab 10 times. Between 73 and 86, picked up all techniques of cloning, sequencing, hybridization protocols, etc. Next one, please. And I looked at the mechanism of cytochrome P450 induction in rat liver. And this was started actually by Professor M.R. Rao, including the assay for cytochrome P450. That was the concept at that time was, eukaryotic systems are a black box because the responses are very slow. But I chose this system because the cytochrome P450 is induced very fast. For example, if you inject phenobarbital to the rat, in 6 hours, you will see the protein. Within 12 hours, you can see substantial increase in cytochrome P450. Therefore, we thought the messenger RNA should be induced much before. Next one, please. And therefore, we chose the translation system at that time for quantifying messenger RNA. All that you do is take the RNA from the liver with phenobarbital treatment in vivo, translated in a wheat germ cell-free system we made We used to get wheat from Yeshwanthpur market, and Rama, the attendant, would make the germ, and we made the germ extract, cell-free extract. And here is uninduced, and here is the induced, and you have radioactive labeling, and then run a gel and quantify the mRNA product. That was how messenger RNA was quantified this by translation. Next one, please. And then, we cloned the cytochrome P450, quantified mRNA by cell-free translation, as I said. We could do run-on transcription with nuclei, and we used G-free cassette, which is a standard technique those days. If you want to identify a promoter elements, you link it to G-free cassette and transcribe it in a nuclear extract system. The other one is gel shift assays. I don't know whether, I hope people still do these things. You have those suspected upstream elements, bind it with the nuclear extract and run a gel, look for gel shift. Receptor media, I will come to this in a minute. Next one, please. And this is just a cloning part. Next one, we cloned the cytochrome P450 gene partial clone. And the bottom line was, if you have to induce cytochrome P450 with phenobarbital, we found you need heme. The messenger RNA was induced. If you, for example, inhibit heme synthesis with cobalt chloride, the messenger RNA was not formed. That was the concept by which we proposed that heme is required for transcription of cytochrome P450 gene. Next one, please. And this is the digression I took. I felt, if we have a promoter region which is only minus 173 base pair upstream as a promoter, instead of putting cytochrome P450, can I put a growth hormone gene and then inject it into the rat as a polylysine sialoglycoprotein complex, because this is known to go into the liver. And then we thought, if you inject with phenobarbital, would growth hormone be produced? That was the idea. The experiment was done long back, published in 2000. Next one, please. And as a control, we had a metallothionein gene, a clone obtained from elsewhere. And if you inject with zinc, you can see the messenger RNA. Similarly, when I injected with phenobarbital, we could see the growth hormone mRNA. What we thought is growth hormone, but we realized subsequently that the mRNA was not defective. So it was not forming growth hormone, and we were waiting for months for the animals to grow bigger, because that was the time that the growth hormone was a great recombinant product. Next one, please. At the same time, I didn't want to get into this gene therapy area, because that was the time with Gelsinger, I don't know, some of you would know, died because of gene therapy. The gene was introduced along with adenoviral particles, and he died of adenoviral toxicity, and there was a whole furor over the globe, and we were all asked to re-examine all gene therapy techniques. So I said, from India, it's very difficult to get into gene therapy and abandon. And this is the final model which we proposed for regulation of cytochrome P450. We had identified a negative element, we have identified a positive element, all by gel shift assays and run-on transcription. And this we could publish in PNAS, saying the uninduced state, we had the protein binding to the negative element and preventing the initiation of transcription. And if you inject with phenobarbital, the protein got phosphorylated and probably involves heme. Now it shifts from negative element to the positive element, and transcription ensues. And this paper we could publish in PNAS. Next one, please. Now, at the end of it, we had several data. Heme regulates translation, very well known, EIF to alpha phosphorylation. Heme regulates translocation of proteins, especially ALA synthase. I told you, first enzyme in heme pathway is nuclear coded and has to be translocated across the mitochondrial membrane, and found heme will block it. Therefore, it was the end product regulation by preventing translocation. Heme regulates transcription of cytochrome P450. Heme regulates differentiation of hematopoietic cells. We didn't do this experiment. This was done by other groups. Ultimately, we could publish a small review in tips saying we wanted to say heme is a second messenger, but the editor did not accept that word. but he said you can use this word multifunctional cellular regulator. We used this and published it. Next one, please. And then, that was the time. Next one. While I was doing the cytochrome P450 research, nuclear extract and so on, chromatin, Professor Rao would know, entry has been made and chip assays. And I was wondering whether we should get into chromatin-based transcription studies. That was the time, early 1980s, Astra came to my lab. Thanks to Dr. S.A. Kumar, AstraZeneca, which became Astra Research, wanted to start a foundation working on infectious diseases, and they came to me for help. And they actually, Astra worked in my lab for 2 years, and I started 4 projects for them: malaria, tuberculosis, cysticercosis, and diarrheal disease. We also recruited the first employee of AstraZeneca, that was Dr. Raman Roy, and he purified certain restriction enzymes, half a dozen restriction enzymes in the laboratory. Astra made a very substantial funding for almost a decade. I never went to anybody else because Astra funded me. And many of you may not know, the entire north wing of the old biochemistry department was built out of Astra money. And I moved upstairs. Next one, please. And that was the time I thought, what am I doing? Should I not get into something which is closer to society? And I decided to start on malaria. And and also there was a heme connection. That was another reason why I went into malaria. I'll come to it in a minute. Malaria has come down globally now, last decade, 500 million to 250 million. And the mortality has come down from 1.5 million to 400,000 plus. It's still a major risk in the globe. In fact, WHO says in the malaria elimination program, 11 countries are suspect. 10 are African countries are a problem. Now, outside Africa, the only concern is India. But although we think we are eliminating malaria, India is still on the radar. And the decrease is essentially possible because of bed nets, pyrethroid coated bed nets and so on, and this wonder drug artemisinin. We can clear the parasite in no time, but artemisinin resistance is spreading. There is no vaccine for malaria. And one of the issues which I was interested was cerebral malaria. The severe cases of malaria, I'll come to get to it in a minute. The mortality is very high. Next one, please. And for those who are not in the field, the parasite cycle is simply this. The mosquito injects sporozoites, gets into the liver, multiplies in several hours. And you get merozoites, which infect the red blood cells, and you get the ring form, trophocytes, and the cycle continues. The cells will lyse, the parasite is released, and infect fresh cells. And that is where you get the chill and the fever. But in the process, it also generates sexual forms, gametocytes, which get back into the mosquito, and macrogametes, microgametes, they form oocyanides, and the oocyanides become oocysts and sporozoites. So the sexual cycle goes in the mosquito, the asexual cycle goes in the red cell. And simple malaria, yes, it's treatable, but we all know chloroquine has become resistant, artemisinin resistance is spreading. But I was more interested in this cerebral malaria. You can have infected RBCs lining the endothelium, preventing blood supply, and you have multi-organ failure. And multi-organ failure, and then you have happening in that in the brain, blood supply is blocked, and then you have cerebral malaria. This can happen to placenta as well, and the growth of the parasite is inhibited. And the growth of the fetus is inhibited. Next slide, please. And this is the pathway for heme biosynthesis. As you all know, in the mammalian system, for example, it is partitioned between the cytoplasm and the mitochondria. The first step is in mitochondria, the last step is in mitochondria, is of consequence to us, ALS synthase and ferrochelatase. This is in the mammalian system, whereas in the plant system, ALA is formed by a weird 3-step reaction of glutamyl tRNA. Starting from glutamyl tRNA, but subsequently it forms the same pathway. But at the end of it, it has two options. Either it can form chlorophyll or it can form heme. Now, this is the mammalian system, the plant system. And next one, please. As I said, the parasite is actually swimming in a pool of hemoglobin. As I showed you in this figure, it's swimming in a pool of hemoglobin and gets tons of heme from the host. But I was asking this funny question, would the parasite make heme? That seemed to be rather a stupid question, but I nevertheless went ahead and simply used, glycine plus succinyl-CoA gives ALH. Glycine 2C14 is a very specific precursor for heme, and therefore we used glycine 2C14 and then used a falciparum culture. And in falciparum culture, we found the parasite, isolate the parasite and show glycine incorporation into heme, because it can only be parasite heme because RBC cannot make heme. So that was the first paper with Namita Cerulia showing that the parasite can make heme and it is a drug target. I'll come back to it. Why did we say it's a drug target? Next slide, please. And at that time we didn't know how does the parasite make heme. So we thought the host doesn't have mitochondria. RBC doesn't have mitochondria, so it doesn't have the first enzyme or the last enzyme, but it has the intermediate enzymes, ALA dehydratase, and intermediates can be imported. And we showed that the second enzyme, dehydratase, can be imported into the parasite, and it's a drug target. And we published it in Nature Medicine, actually. And it has a meaning. Now we can understand what it means. Next one, please. And interestingly, that was the time, I think 2003 or something, the entire falciparum genome was published. Amma, the parasite pathway was published. Parasite genome was sequenced, and then all the heme pathway genes, putative genes were present. Therefore, once and for all, the skepticism was laid at rest. Yes, parasite does make heme. Although it gets tons of heme from host hemoglobin, it can still make heme. So then we worked out every step in this pathway. And there are very interesting features which I will not get into it. Just to mention, I told you the human pathway or the mammalian pathway is split between the mitochondria and cytoplasm, whereas in the parasite, it has a three-way pathway. That means mitochondria, first and last enzyme. It also has an apicoplast, vestigial chloroplast. Some steps are there in this apicoplast, and there is a step in the cytoplasm. So it's a very interesting pathway in the malaria parasite, which we worked out, complete detail, every step, every enzyme, every gene was cloned. And interesting features, many of them are targets actually. Next one, please. And many other groups started following this. Heme pathway has become, I should say, a lot of interest around the globe, Australia, US, London, and so on. Next one, please. And we have several publications. Next one, please. Now, we said it's a drug target using an inhibitor for ALA dehydratase. We use succinylacetone, 1 to 2 millimolar, and we found the heme pathway, heme is inhibited. Glycine incorporation into heme is inhibited. The growth of the parasite is also inhibited. So we said it's a drug target, but soon realized 50 micromolar is good enough to inhibit heme synthesis, but nothing happens to the growth. Therefore, the use of succinylacetone inhibition of growth was actually an off-target effect. Therefore, we've said the pathway is there, but it is not essential for the growth of the parasite in the blood stage. Next one, please. So we wanted to answer this question. Why does the parasite make heme? If it is not essential for growth, then we went into the blood stage, we went into the animal model, and we thought the pathway may be essential for growth of the parasite in the mosquito, for which we need a insectary, we didn't have an insectary at I say there was one at ICMR lab you would have seen closer to the airport. And I remember Arun, going up and down with the cages with mosquitoes. It was a very difficult exercise because it is very finicky, to grow the, I'm a patient, to grow the parasite in the mosquito. It was a hell of a problem because you need 19 degrees centigrade temperature, you need 70% humidity. Finally, we managed to build a small insectary thanks to Dipankar in the CIDR, and then looked at the Berghii. The question, we were trying to answer the question whether the parasite growth in mosquito requires heme. Next one, please. So we made knockouts rather than using inhibitors. We made knockout of the first enzyme, of the first gene, ALA synthase knockout. We made a knockout of the last ferrochelatase, that is the dihydrofolate reductase replacement. These are the proof, and here simply you can clearly see the Western. The knockout parasite doesn't form ferrochelatase, for example. Wild type it is there, but the wild type and the knockouts grew merrily in the animal. The blood stage, you can't find much of a difference. Next, there is some lag which I will come to. But then we went into seeing whether eukaryotes are formed. Yes, eukaryotes are formed. The knockouts form the eukaryotes, whether it is ALAS knockout or FC knockout. Next one, please. But when we came to oocysts, yes, oocysts were affected, severely affected. Next one, please. And the final stage is the sporozoites. You can see the sporozoites here, wild type. And the ALAS knockout, you don't see the sporozoites. In the FC knockout, you don't see the sporozoites. Sporozoites are in the salivary gland, you remember, and they are injected into the animal, and they go into the liver actually. And this experiment was also very exciting. If ALAS knockout, you don't have the parasite. If you feed ALA to the mosquito, what will happen? We fed ALA to the mosquito and showed that oocysts are now formed, clearly showing that heme is absolutely required, not only for the mosquito, it also required for the liver stage, because the parasite first goes to the liver and then comes out and then it is infecting the blood. Now, this was a very exciting observation. Next slide, please. Next one. And the next one. Yeah, this is neat. Yeah, this is fine. This is fine. Now, this also clearly showed, we could show the, by gel analysis and so on, using again radio-labeled succinyl acetone to inhibit the parasite heme synthesis. Clearly, you can see what we do is to get the Plasmodium berghei to grow in the animal, isolate them and put it in culture. Therefore, you can do short-term experiments. We added succinyl acetone and looked at ALA labeling. Yes, heme synthesis is wrong. But we could clearly identify by Maldi that these two bands are cytochrome oxidase. Whether it is inhibitor or no inhibitor, you see the cytochrome oxidase complex, but heme is not. Heme is inhibited. So obviously, the parasite is getting heme from hemoglobin. So basically, it means heme is not required for the parasite growth in the blood stage, but it is required for it to grow in the mosquito. Next one, please. And this is the experiment which actually supplementation with ALA clearly showing the parasite will inhibit the mosquito. If you allowed to bite the animal, you will see the parasite in the blood. That is, if you have supplemented the mosquito with ALA, clearly showing the liver stage also requires heme. Therefore, we know that heme is required, biosynthetic heme is required for the mosquito stage, it requires for the liver stage. But still it was bothering me. Why does it make it in the blood stage? Next one, please. Now, this is a very interesting, next one. Next one. This paper, when we published that it is required in the mosquito stage, was published in PLAS Pathogens, and it had a wide citation because it was the first time we were showing not only parasite makes heme, it's essential for the parasite growth in the mosquito, and many other groups followed it actually. Next one, please. But everyone, next one, this was confirmed in falciparum by other groups. But I was asking this question, why does the parasite have different routes to make heme, in addition to acquisition from host hemoglobin? Next one. This pathway is very, very interesting. You have the RBC. Other groups showed, although it doesn't have mitochondria, it doesn't have the first and the last enzyme, the intermediate enzymes are present in RBC. And that is how we could show in the Nature Medicine paper that ALA dehydrogenase is imported from the host. Whereas Plasmodium berghei or Plasmodium vivax, as you know, there are two plasmodia which are very important for a human parasite. That is, one is falciparum, another is vivax. In India, it is almost 50-50. And vivax prefers reticulocyte. And in reticulocytes, you will find a mitochondria is present. That means first and last enzymes are present. It means complete heme, and heme is important. Therefore, we were asking, I was wondering, why does parasite has so many routes? It makes heme by the biosynthesis. It imports host enzymes, host intermediates, host heme, and huge amount of hemoglobin from RBC. Now, why does it have? And this heme is very toxic, and that is why heme is converted into this pigment called hemozoin. This happens in the food vacuum. And that is where drugs act. In fact, that is how I got into this area. Chloroquine, for example, inhibits, binds heme and prevents conversion of heme to hemozoin. One of the important steps of artemisinin in action is this. Therefore, the heme is detoxified, hemoglobin heme is detoxified because it doesn't have the biliverdin, bilirubin roots actually. But at the same time, there are so many other mechanisms for acquisition of heme. I was asking this question, why does it make heme? For 20 years, everybody was quoting, yes, parasite heme is required only for growth in the mosquito. It is not required for the blood stage. I was not satisfied. I was finally thought, it took 20 years, it may have a role in virulence. Next one, please. This is the last part. It is not published. I will very quickly try to summarize in 10 minutes. I'll try to see. So I thought the heme, probably parasite, makes heme to sustain virulence. Next one, please. This is the model which we use. There is a C57 black cerebral malaria model. If you infect it with Anka strain of Plasmodium berghei, it will show typical cerebral malaria systems, and the animals will die between 8 to 12 days. You can clearly show blood-brain barrier is broken. If you inject Evans blue in the C57 black, you will see the brain is stained blue. This will not happen with bulb C mice. Bulb C mice will sustain the parasite, grow in the blood. They will all die later due to anemia. Next one, please. And this is what the previous slide. And yes, this is where, again, we had some preliminary results. We were very excited. So far as virulence is concerned, this is the blood-brain barrier study with Evans blue. This is the wild type. This is the ALS knockout. This is the FC knockout. And the parasite did not die of cerebral malaria. They all prolonged, and the animals died later due to anemia. That means heme is required to cause cerebral malaria. That was the initial idea. Next one. And this is what we have. A lot of data, which is, as I said, unpublished. We have brain capillary occlusion with several cells in the brain, whereas the knockouts do not have. Similarly, the hemorrhagic foci you see in the wild type, you don't see it in the knockouts. Next one, please. And this was bothering. There is a lag when we did with large number of animals. The knockout parasites grow a little slower in the beginning. Of course, they continue to live. As I said, parasites grow and they die much later. But they do not cause cerebral malaria because there is a lag. So here we have injected 10 to the 5 RBCs, infected RBCs. So to compensate for that, we injected 10 to the power of 7. So therefore, the lag is compensated. But in spite of that, they didn't cause cerebral malaria. And the animals all died much later. Next one, please. I'm rushing because of I don't want to make up time. And there is a score called a rapid murine coma and behavioral scale, which is very high for the knockouts. It's about 15, 16, whereas it is only about less than 5 for the wild type, indicating that the knockouts do not cause cerebral malaria. Next one, please. And we have a huge amount of data, but this is important to show that the infected RBC goes to the brain. And this was a matter of debate between human cerebral malaria and the mouse malaria till this technique was available where you can label the parasite with luciferase. You have the M-Cherry GFP clone where you can incorporate the gene of your choice. We made the ALS knockout, we made the FC knockout, expressing luciferase. And wherever the RBC containing parasite is there, if you inject luciferin, you will have bioluminescent product, which ranges from light blue to red when the parasite is loaded. Just this slide alone will tell you, you can clearly see the parasite in the blood, in the brain, but you do not see, especially in the FC knockout. It's not there. Infected RBC, you do not see in the brain. And you can do ex vivo imaging. You can also clearly see there is no parasite in the brain. You can clearly see it in the wild type. Next one, please. And of course, we have generated all the data. Again, Evans blue, blood-brain barrier. You can clearly see the blood-brain barrier is not there in the wild type, but again, it is counteracted in the case of ALS knockout and FC knockout. And we have several capillary in the brain. We have extravasation, luminal and abluminal leukocyte presence, IgG extravasation. For example, in the wild type, you can see the IgG extravasation in the brain. You don't see them in the knockout. Like that, we have, for example, you can clearly hear this amyloid precursor protein in the brain, these are all classical symptoms of cerebral malaria, which you do not see in the knockouts actually. And you can see I hope you can see the spots in the parasite in the brain. You do not see them again in the knockouts actually. Therefore, we have a huge amount of data collected, imaging data to show that the heme is required to cause cerebral malaria. Next one, please. And we have said for cytokines, suffice to say, all the inflammatory cytokines, whether it is IL-4, TNF-alpha, interferon gamma, they're all inhibited. And we also have RT-PCR data from the brain mRNA comparing ALS knockout and FC knockout. FC knockout gives much more significant results than ALS knockout, maybe because of the import of the host intermediates, compensation is happening. And this is actually the facts data, which is summarized here. Next slide actually gives you the raw data. Can I go back to this slide? I just want to explain that many of the leukocytes in the brain, CD3, CD4, CD3, CD8, plus granzyme, plus perforin, plus interferon gamma, all of them are inhibited in the knockouts. And here you also have the Western. For some of these, you can, for example, clearly see IL-1 beta being completely inhibited in the knockouts. Similarly, phospho-NF-kappa being inhibited. That means all the inflammatory processes are not happening in the knockouts. When you knock out the first enzyme and the last enzyme of the heme pathway, you do not cause cerebral malaria, and inflammatory responses are not happening. Next one, please. And this is the raw data for the facts analysis. Next one. I will not try to explain. And again, the most important data we have is the inhibition of the hemozyme formation. In the knockout, what I'm trying to tell you, if you block the biosynthetic heme, the conversion of hemoglobin heme into hemozyme is inhibited. So you have, in this knockout, whether the ALS knockout or FC knockout, essentially that is the important summary of all these data which is given here, that the hemozoin formation is inhibited. And hemozoin is a pathogen associated or a danger associated molecule. You know, LPS is a danger associated molecule. Teichoic acids are a dangerous. These are all classified as damp and so on. Next one, please. Hemozoin is actually a inert molecule so far as the parasite is concerned. When the parasite is lysed and hemozoin is released, it is very toxic to the host. And hemozoin you can see in the spleen, you can see in the liver, but in the knockout, they are all inhibited. This is hemopexin, I will not get into that. Clearly showing hemozoin formation is inhibited. Next one. And again, you know, how does it happen? How does biosynthetic heme inhibit conversion of hemoglobin heme into hemozoin? 3 parameters we have identified. One is you need an acidic pH for hemozoin formation, which we can clearly show by lysotracker. You know, if you lysotracker staining, I hope you can see in the wild type. You don't see it in the FC knockout, for example. That means the pH of the foot vacuole where this is happening is compromised. But at the same time, the neutral lipids labeling, you know, with the P32 or body P staining or Nile red staining, they are not affected. So what else is affected? One is pH is compromised. Next one, please. We found unsaturated fatty acid labeling is significantly inhibited. Oleic acid formation is inhibited. I hope you can clearly see in the knockout. Therefore, the unsaturated, the membrane associated unsaturation is very essential for the pH maintenance. That is the second one. The third one came out from the proteomics data. You know, we did a lot of proteomics of the food vacuole. All the study results I'm presenting are in relation to food vacuole. We had the, you know, trypsin digestion, and then LCMS-MS was done. And the next one gives you the summary, I think, of the proteomics data. Next one. Before that, the most important protein that was missing in the knockout was the type 5 proton ATPase. This is essentially maintaining the pH of the food vacuole. There are 2 experiments I have shown here clearly showing the proton ATPase is more or less absent or considerably decreased. Next one, please. And this is a summary of the proteomics data. We could see 250 to 201 proteins in the food vacuole, wild type and the FCK, the ferrochelatase knockout in particular. The food vacuole proteomics, very hardly, you see, I think there's just a single publication of food vacuole proteomics. As I said, we have yet to publish this part of the data. And 175 proteins are common, saying our preparation of food vacuole is reasonably okay. And you know, we have shown in the proteomics analysis, the proton ATPase is absent. And there's another enzyme called bergipine, which is a cysteine protease that is required for hemoglobin degradation. Next one, please. Therefore, there are 3 things which are happening when your biosynthetic heme is not made. The food vacuole pH is compromised, and this is because the membrane unsaturation is compromised and the proton ATPase is absent. Bergipine is absent, therefore heme itself is not formed from hemoglobin or poorly formed. Next one, please. And this is the last but one slide, I think. We thought, you know, with this kind of data, when we can prevent cerebral malaria in the knockout, that means heme is required to cause cerebral malaria. Biosynthetic heme is required for causing cerebral malaria. Can you inhibit ferrochelatase? And do you have an inhibitor for ferrochelatase? And we were very fortunate in finding griseofulvin, which is available in the market. It is the FDA-approved drug. People take it for weeks to treat ringworm, you know, fungal infection of the skin. But it has a secondary effect. It is reported to inhibit ferrochelatase. So we tried it in the animal. Next one. Interestingly, we found we can reproduce all the effects of the knockout. I will not try to go through. These are the data. including blood-brain barrier breakage and delay in the death of the animal. All data, hemozyme in particular, inhibition of hemozyme, all are happening with griseofulvin. Depending on the dose, here what you see is, depending on the dose of griseofulvin, you can see the cerebral malaria is not there, but the animals will die later on. Therefore, we are now proposing, next one, that we can use a combination of artemisinin and griseofulvin to prevent. Artemisinin will take care of the parasite and griseofulvin will take care of the cerebral malaria. And this may be a new therapy that is possible. And this is the summary of this part of the thing. The parasite makes heme, and what really happens is this heme is required for the unsaturation of the brain, of the membrane of the food vacuole. Proton ATP is absent, the food vacuole pH is compromised, and that is where you see the hemoglobin heme. Berkepine is absent, therefore heme itself is not formed, and therefore hemozyme formation is inhibited. And that is next one. And this is something very, very interesting. A huge amount of heme pool, which is derived from hemoglobin heme, is regulated by a small pool of this, even much smaller pool of heme, which is coming from biosynthesis. This is something very exciting to us. It is still unpublished data, and we think this is the one that is regulating. So in all, we think we have a new molecule, griseofulvin, which can be used to prevent cerebral malaria. Next one. I'm just summarizing. The epicomplexon, Plasmodium is an epicomplexon. There are thousands of epicomplexons known, but there are 7 of them which are of consequence to the human. Plasmodium, Babesia, and they vary. Cryptosporidium, for example, cannot make heme at all. Entirely dependent on host hemoglobin. Whereas Plasmodium has the entire pathway for making heme, it also gets heme from the host. Therefore, these are all, toxoplasma is another which is of great interest to scientists. Next one, please. So I have taken a lifetime to identify 2 molecules. Curcumin, Professor Rao mentioned. I didn't go into that because I wanted to talk about the heme story right from the beginning, from Neurospora to cytochrome P450 to the malarial parasite. And we have at the end of it, I'm 83. So coming up with griseofulvin as one possible combination with artemisinin. At least 35 of the 50-odd PhD students and postdocs have worked on this heme story one way or the other. I have many students coming from other labs. Those who had problems with other labs used to migrate to my lab with the permission of the guide. The guide himself would come to me and said, please take my student. He is a problem to me. So the 50 includes all those, and many of them worked on the heme story. And this has been my life, one of my main themes. And this is now being continued by Dr. Arun Nagaraj at the Institute of Life Sciences, Bhubaneswar. And I am only a sounding board at this point of time. Next one. I think I'm done. I want to thank the BC, Professor Rangarajan, and all the chairman and IISc for providing me shelter for 60 years. As I said, I joined in January 1961. I am still around. Thank you so much. Thank you, Professor Rao. Thanks, everyone.
Thank you, Professor GP, for sharing your long journey, which is still continuing, ongoing. And then also it is really fine example of what you can do with a single-minded devotion to solve a problem. I think it's a great example to many of us and the future generation. Thank you, Professor GP.
Thank you. Thank you, Rao. Thank you.
So we can have a couple of questions. Is there any question? I think nobody has raised a hand. I think everybody enjoyed your talk. Thank you, Professor GP.
Thank you. Thank you. Yeah.
So we can move to the next talk to be given by Professor K.S. Ullas. Ullas, you are there?
Yes, sir.
Okay. See, the previous speaker was my mentor. The next speaker is my mentee. So it so happens that I'm sharing the dais of the 3 generations in between myself. Okay. So Professor Ullas did his PhD in 1997 to 2003 at the Department of Biochemistry. And then he spent some time as a postdoc in the IGBMS Strasbourg. And he returned back to take up the faculty position at the TFR Mumbai. He is now with Professor of Biological Sciences at TFR. I think his main interest is to make a connection between the cellular metabolism to epigenetics phenomenon. He's one of the very rare contribution person in the entire world to take this forward, and he has made very significant contributions in this area. More particularly, he works on the proteins called serotonin, linking metabolism to cellular processes. I think we are eagerly waiting for Professor Ullas to give his talk. Professor Ullas.
Yeah, thanks a lot, sir. Indeed, it's an honor and pleasure to be part of this. And thanks to Professor Rangarajan, DN, and all the entire organizing committee for giving me this opportunity. Yeah, it's like really an overwhelming feeling. I'll start with my presentation. I'll share the screen and I'll do what's been asked, which is patient screen. Can you see my screen?
Yeah.
Okay.
We can see. You can make it full screen.
Yeah.
Make it full screen.
Yes.
Yeah. Go ahead.
Okay. Good. Thanks. Okay. So, of course, like Professor Rao mentioned, I joined the department in 1997 in the old iconic structure where most of the history of course with. Then I moved to Professor M.R.S. Rao's lab in 1998, 1999. And really thanks to him, probably without him, I would not be in science today. So all credit goes to him. Of course, I would like to take this opportunity to thank all my contemporaries, students, all the entire faculty, not just within the biochemistry department, but everywhere else, who have been having, who had a huge formative influence on me. And most of what we do, what I do here in the lab, is all thanks to the foundations laid by them and all the peers then. I'd like to also take this opportunity to thank my other mentors in the field, both Paulo Sazanakarsi, who unfortunately passed away recently, and Irvin Davidson from IGBMC. I would like to mention that neither Paulo nor Irvin, at least Paulo when I joined the lab, was not working on CERT-WINs. This is something that I started on my own, and he gave all the freedom for me to work on CERT-WINs. Irvin never worked on CERT-WINs before and after, so thanks to them for giving this freedom, and that's really helped me to shape the career and the questions that we're asking. I've been at TFR since 2008, where essentially we look at nuclear mitochondrial crosstalk, looking at how both nuclear transactions and mitochondrial functions, both in the context of metabolic homeostasis and energy homeostasis, together actually maintain cellular organismal physiology. And one of the things that we are hotly pursuing is whether causes of disease is because of conflict between evolutionary history and life history. Probably that's a separate talk that I can give at another time. Over the years, we have done several things, especially looking at organ system hierarchy, intertissue communications, in the context of aging and evolutionary conservation. Recently, I've started another lab at TFR Hyderabad. So I have 2 labs, one in Mumbai and Hyderabad, pursuing the research questions that we have. I'd also like to thank animal facilities at Actric and Isoponia, because we never had an animal facility in Mumbai for my work, and it's been really a huge help for us to travel. Although it's been a nightmare, but we have been greatly benefited by the gesture provided by those institutions. Now let's talk about the science itself. So why first about dietary inputs or cellular metabolism? Probably, for a biochemistry audience, I don't think I need to give this introduction, but nonetheless, what's turned out is that all the boring biochemical reactions that we think about are actually important in encoding and decoding information. Now, there's a huge hype about how phenotypes are influenced both by an interaction between the genotype and the environmental inputs. And over the decades, what has emerged is that most of these interactions are largely due to the post-translational modifications that are happening both in the epigenome and also all the cellular proteins in the way of all the modifications that you think about. So the classical view of biology, or a molecular centric view of biology, where everybody thinks that genes contribute to phenotypes, and in fact the gene products are the one that dictate metabolites, has seemed to have been turned upside down, where we now know that in fact it's the metabolites that control genes, gene products, and molecular components. What we still lack in the field is, at least from a systems perspective, is how energetics and thermodynamics couple all of these. Something that we've started to look at. Of course, we've taken some baby steps in this context. So the point is that identifying genetic and molecular architecture is insufficient. What you need to actually do is understand how components interact with each other to give the phenotypes. For students who are not convinced about why metabolism is important, and in the context of gene expression, now we know that pretty much everything that the epigenome does in the context of gene expression regulation has one way or the other link with metabolism, because most of the modifications that you think about that's happening on the histones or chromatin are all derived from metabolic inputs. So therefore, metabolism as a whole has an overriding effect on genome organization and functions. This is something that's been driving our interest in this field. Right now, we do many things in the lab. For, of course, obviously today's talk, given the time limitation, I'm going to only talk about some of the recent work that we have done on the epigenome organization and function. A key aspect that we follow in the lab is to ask whether daily perturbations in our feed-fast cycles actually contributes to long-term consequences on development and aging. This is interesting because often what people in the field have done is like really do high-fat diet or long-term perturbation, which rarely we as humans go through. I mean, it's very rare to find somebody eating 35% fat or 40% fat every day in and day out all through the life to get some diseases. Essentially, what's happening is that something about daily fat cycles is going wrong, and that's possibly what is contributing to long-term phenotypes that we still don't understand how this is being brought about. So in this context, we do many things. Like I said, I won't be able to talk to you about mitochondrial functions and the whole lot of work that we've done in metabolism and energetic flexibilities. I will talk to you about largely the nuclear functions, although I don't particularly like only the nuclear-centric view of biology, but I think it's easier for me to convey that today. When you talk about gene expression, I'm sure all of us will agree that there is a concept of 3D chromatin architecture. The epigenetic landscapes can change both in a short-term scales and long-term scales. There are many open questions in terms of coactivator and transcription binding specificities, splicing fidelities and efficiencies, and post-transcriptional gene regulation. All of this put together can give you fitness, and here is what I'm going to talk to you about. And for this, again, we take a liver-centric view because although we all acknowledge that liver is one of the most important organ, and as Professor GP mentioned, even in his talk, the hepatic view of biology came out very well. We believe that liver, despite having so much buffer capacity, is one of the organs that is involved in the onset of the diseases. And we would like to go back and ask, what is it that actually causes some of the diseases that the humankind is plagued with. Now, therefore, liver becomes interesting. One of the questions that we set out to ask is a simple-minded question: is that all of us know that we go through fat-fast cycles. During starvation, for example, you have transcription of a whole lot of genes involved in metabolic homeostasis and energetic homeostasis. Now, refeeding typically happens within tens of minutes, and the system doesn't have so much time to rewire the gene expression profile, and you need to immediately inhibit the translation of all the transcripts of the starvation genes when you refeed. Now, if you don't do this, one can already imagine that this will go into a metabolic overdrive, and that could cause diseases. Now, in the field, although there was some amount of work that was done on inhibition of transcription, there was no idea about what mechanism takes care of these transcripts to give you this immediate transition from a fasted to refed state. So in this context, we surmise that possibly there is something called a hepatic mirinome, which specifically happens in a fed state. Of course, mirs can be expressed throughout both fed and fast cycles. We specifically asked whether microRNAs that are expressed more in a fed state, could they inhibit the fasting responsive genes? turned out to be the case. We identified a bunch of microRNAs, all of which could regulate beta oxidation, mitochondrial biogenesis, and oxidative phosphorylation, and gluconeogenesis, which is interesting because this showed us that there is something called convergent action and additive action of MIRs, where the same bunch of MIRs could act both on the upstream transcription regulators and the downstream genes to bring about a concerted inhibition of the entire pathway of catabolic genes, so that the anabolic response could be triggered when the organism went from a fasted to a refed state. What's interesting is that we found that these bunch of microRNAs also oscillated in the liver, not just their levels, but also the risk loading of the mirrors, the microRNAs also oscillated, which means we found that between fed, fast, and refed states, the microRNA levels of the fed microRNAs actually oscillated, which is really fascinating. Now, does it really matter? So one of the questions that one could ask is, fantastic, we have identified a bunch of molecules that could be potentially regulating physiology, but do they really regulate physiology? So what we did is we went back and sponged off these MIRs by simply providing complementary strand, which is done routinely in the field. And once we sponge these microRNAs only in the liver, of course, we found large-scale changes in metabolism or metabolic flexibility and physiology of the organism. But this was best captured by what is called as respiratory quotient, which is like a gold standard of asking what is the fuel that the organism chooses to burn, whether it is in a fed or a fasted state. Typically, a respiratory quotient of tending to something to 0.7, which happens when you're fasting, is because of the organism burning lipids or fats, and the organism chooses to burn glucose, then your RQ ratio becomes 1. Now, when we sponged off these MIRs only in the liver, interesting, all of the molecular perturbations are happening only in the liver, and yet we are seeing a whole body phenotype for the energetic levels. This was very reminiscent of what happens in diabetic or pre-diabetic individuals, which is really interesting. So then we asked, why is this happening? Indeed, we found that there was a large-scale perturbation of both fat metabolism genes, mitochondrial functions, OXPHOS, and gluconeogenic potentials. So this was interesting. Now, this was recently published, where we showed that actually oscillating microRNAs are probably important for maintaining fitness during every feed-fast cycles. And what we have seen is that some of these microRNA oscillations are actually perturbed in old animals or in high-fat diet-induced obese animals. We don't know whether this is a cause or a consequence, something that we are pursuing, whether changes in oscillation frequencies or phase shift in some of these small molecular components, which are probably very difficult to otherwise score in a normal fed-fast cycles, are they actually contributing to long-term consequences? This is largely led by a graduate student who is now a postdoc in Denmark, also assisted by a postdoc called Chandrika. An amazing thing that we found serendipitously is that there is something called molecular anticipation, where these microRNAs are actually formed and kept as precursors in a fasted state, and their processing seems to be the one that is regulated. And this is interesting because you want to have a rapid response and a rapid changeover from a fasted to refed state. We don't know the mechanisms behind this, so we are working towards this to ask what is the molecular anticipation. And what's interesting is that the fasted state seems to prime the fed state in the liver. Now, this is all about post-transcriptional control, but nonetheless, the genes that are involved in whether you talk about fed state or a fasted state, if you look at the promoters, you have a bunch of transcription factor binding sites. In fact, this is true for most of the genes. Now, one could ask a very generic question: why would you need a whole lot of these transcription factors to simply upregulate a gene under a fasted state? The reason is because no state in biology is a end state, and you have a dynamic oscillation. And the extent to which you want to mount gene expression is dependent on the extent to which the stimulus is actually received. Now, here again, while we have most of biology or molecular biology has invested in understanding cis-trans interactions of transcription binding specificities, we don't know how co-activator and co-repressor specificities are brought about. That means when does a P300 or a CBP is able to recognize which transcription factor to bind and activate a gene. So that's really largely unknown in the field. So we wanted to ask this because earlier, some time ago, again, using Certmin as a candidate gene, we are not married to Certmin, but we just use this as candidate genes. we actually found that SERT1 also has an intrinsically disordered region. Many people in the audience might appreciate that intrinsically disordered region, which were again thought to be useless parts of the protein a couple of decades ago, have come back with a vengeance. And it's almost like these ideas are the one that are ruling a lot of protein interactions that we still don't understand very well. So we found that we and others actually in the field found that SERT1 also has an intrinsically disordered region. An intrinsically disordered region are usually overrepresented in transcription regulatory mechanisms. And so we asked whether IDRs are the one that could be involved in regulating this. But more fundamentally, we wanted to ask, how can SERT1, which is upregulated in transcription during fasting, get downregulated in a fed state? So we wanted to ask whether glucose inputs or nutrients can directly switch transcription. Now this is interesting because while there are a lot of signaling cascades, including glucagon, insulin signaling, a lot of things that are happening, we want to ask if nutrient input can directly control transcription. This is important because SERT1 associates with transcription factors of both fed and fasted transcription factors. It inhibits fed transcription factors while it activates starvation transcription factors. When you go from a fasted to a refed state, SERT1 has to get disengaged, and so that you relieve the repression on fed genes, but bring the repression on the fasted genes. This has been largely unknown. How can you now regulate binding and unbinding events of this co-activator or a co-repressor complex? So we asked whether glucose itself can do this. Indeed, we found that SERT1 gets glycosylated, and that SERT1 glycosylation is brought about both in an insulin and a glucose-dependent manner, which is really fascinating. What we found is that glycosylation is important to regulate transcription of downstream genes, both of the anabolic genes and the catabolic genes, where the repressed genes get deep repressed and activated genes actually get inhibited or otherwise when you have a glycosylation being perturbed. Now, we wanted to test this as a proof of principle. We identified the site of glycosylation in SERT1, mutated the glycosylation site, and indeed we found that there was a whole, the entire reprogramming of gene expression from starvation to a refed state is perturbed. All of these molecular mechanisms is fine. Is it relevant for the mouse? Is it really happening in a normal context? For that, we either took we either used an overexpression model where we overexpress the mutant SERT1, which can't get glycosylated, or we knocked out SERT1 only in the liver and rescued it with the mutant SERT1 that again can't get glycosylated. In both of these cases, we found really wide-ranging, severe metabolic and physiological problems where glucose homeostasis and fat homeostasis was completely perturbed, which is really interesting. Again, mind you, all of our perturbations are liver-centric, and yet we find whole body phenotypes, which means that this glucose that binds to this protein as a glycosylation moiety is able to switch the transcription between fasting genes and fed genes, which is really amazing. We also show that this in fact contributed to hepatic inflammation, and we are now trying to ask whether this lack of switching over of transcription between fasted and fed genes by the SERT1 is actually what is also could contribute to metabolic perturbations in the long term. Now, this is consistent with what has been happening in the field as well. We were partially scooped by a molecular cell paper, which just came about around the time that we were writing our manuscript, where also it showed that SERT1 IDR is important for switching over transcription from a fasted to fed state, but the molecular mechanisms were not very well understood, and now we propose this. What's interesting is that we show that glucose takes away SERT1 functions. In fact, after 10 minutes, this glycosylation has some impact. After 20 minutes, it has some other impact, and after about half an hour, the entire SERT1 gets degraded because of glycosylation-dependent ubiquitination, which is really fascinating. And we find that this glycosylation is indeed contributing to probably downregulation of SERT1 in a different context, whether it's an obese model or an aged model systems. And in all of these contexts, we see SERT1 glycosylation to be high. Now, this is really interesting because IDRs have come up in a big manner. We are also asking whether how do these co-activators or master regulators of genome functions choose between transcription versus repair or replication, because ultimately you need to parcellate your functions. And how do you now distribute your activities is an interesting question. We are going there. Now, essentially, what we are doing in the lab is asking what are the mechanisms that govern fitness or fidelity of physiological phase transitions if you perturb these physiological phase transitions because of molecular defects, which essentially could be contributed by metabolic oscillations or circadian rhythm or a perturbation of both, which could lead to altered physiological states, which actually compound the problem. And by the time you actually see the phenotype, it's too late, and probably you're not able to go back and ask what the causal mechanisms were. We are also asking several high-level questions now to ask whether plasticity and memory of molecular and physiological scales. One of the things that we want to bring in, and we've already started doing work on, is about thermodynamics and energetics, and also kinetics, something that we have done extensively, how molecular scale problems actually now are manifested at macro level scales, which is the organismal physiology. Now, because of all of this work, we have also started contributing to some of the national missions, where our lab is privileged to be associated with ISRO, where we are now asking metabolic fitness in space travel, and whether aging itself and all the dietary inputs, how does it manifest in a microgravity context, either in the space itself or upon return to Earth. So we are collaborating with ISRO on this, and we are part of Gaganyaan 1. Something that we have started also is to understand double burden of malnutrition. Double burden of malnutrition is really important because India seems to be much worse affected than almost all of the world, including sub-Saharan Africa, where we have really bad, high prevalence of stunting in children and undernourishment in children. Most of these children are known to actually develop cardiovascular diseases, cancer, and obesity later in life. This is called double burden of malnutrition. WHO has flagged it, and all nutrition missions have shown that, in fact, while you can probably rescue stunting, which is really the height and the weight proportions, it's not sufficient to simply look at gain in size or weight. What you need to ask is whether physiologically they are fit. This is something that a lot of studies have not been able to look at. And there is therefore an urgent need to get into basic science of looking at what are the mechanisms that predispose undernourished children to develop age-related problems later in life. We think it's also got to do with an interplay between macro and micronutrients and the ability of your calories to be partitioned between fat and lean mass versus energy production, all of which again goes back to nuclear mitochondrial crosstalk, the core theme of the lab. With that, I'll end my talk. None of this would have been possible without really a bunch of motivated, hardworking students, and a lot of funding from many different organizations, and all the people who work in the background in the form of facility managers and supporting staff. Thank you, and I'll be happy to take any questions.
Thank you, Professor Ullas. Very lucid presentation of the very exciting work, not only with respect to the normal physiology, but also with respect to the microgravity and space program. I think your future program in the stunting will be very promising. Wish you all the best in that.
Thank you, sir.
Okay. Any questions to ask to Professor Ullas?
I have a question. Sorry, I don't see a hand to raise. I think it's disappeared from the screen. Yeah.
Go ahead, Suma.
So yeah, great talk, Ullas. Really enjoyed it.
Thank you.
So I was wondering how much is known about glycosylation of sirtuin and diabetes, and is it similar in any way to aging? Would you put them both on the same spectrum at different places or the same place, or what are your views?
Yeah, so it's interesting because this brings to the fore about, there's this whole mad rush to overactivate SIRT1. I mean, there's this billion-dollar industry that is wants to find magic drugs for everything, right? What we show is that actually overactivating SIRT1 is as bad as underactivating SIRT1. We do find that SIRT1 is hyperglycosylated in a diabetic state, but then a chronic diabetic also there is actually energy deficiency. So I think it's not exactly the same. Aging and diabetes, I wouldn't want to compare the two because your metabolic state is not necessarily the same. But we do find that both in obese, diabetic, and in age context, SIRT1 is hyperglycosylated. Absolutely, yes.
Thank you.
Okay. Patrick? Patrick, you can ask question. You can unmute yourself.
Yeah, so Ullas, nice talk. This is regarding the hyperglycosylation IDR region. Does it really promote to form sort of like a stress granules, or does it really work other way around?
Very good question. We don't know. We are in fact trying to ask whether IDR. So we found that the glycosylation site also can get. I mean, we think that it also can phosphorylate it because it's the same serine and threonine residues. We are trying to ask whether unmodified or modified SIRT1 can be present in either in condensates because IDRs can form condensates, and whether they can be associated with certain transcription zones. Or with other regions in the nucleus. We don't know. These are open questions. We are pursuing this, Patrick. Very nice.
Okay, thanks.
We can have the final question, Ritvik.
Hi, Ullas. Hello from Cambridge.
Yeah, hi.
Great talk. I was actually wondering, when you call fed state and fasting state, are these bistable states at a molecular level, or is it a continuum of how much of food you eat and how much you starve yourself? So is it at a molecular level in terms of glycosylation or what have you? Is this a bistability or is this a continuum?
So it's both. It's both bistable and continuum. It depends on the duration that you're looking at. So in fact, what I didn't get time to tell you is that we look at this in a normal in a normal ad libitum fed fast paradigm where the animal has unlimited access to food. And then we also do time restricted feeding and 16 to 24 hours of starvation. The molecular patterns are very different, which is where I think the field has to be extremely careful in asking whether a 6-hour fasted will be exactly the same as a 24-hour or 16-hour fasted. But nonetheless, the mechanisms that are involved in putting back to a fed state or a fasted state would nevertheless be the same. But the robustness and the engagement of downstream components might be slightly different. So the master regulators would remain the same, but the engagement would be different. And sometimes the changes would be so small that you would probably ignore, because they would come in the noise range, which is something that we are trying to put together. How much of this is really noise versus signal? Because that's also becoming important.
Okay. Thank you, Professor Ullas. I think we are a little behind time. We will move to the next speaker, Professor Vijaya. Vijaya, you are there?
Yes, very much.
Professor Vijaya is also an alumnus of the department. After completing her PhD in the department, she moved to USA for a couple of years of postdoc, returned back to institute to join as a faculty member at the Center for Genetic Engineering, which was later merged with the microbiology and cell biology department. She continued there and then grew up to a professor. And then just last year, she superannuated. Her research area is mainly to understand the host-pathogen interactions and the molecular basis for pathogenesis of 2 human infectious agents, Mycobacterium tuberculosis and Japanese encephalitis virus. I think, Vijaya, the podium is yours.
Yes. Thank you, Professor Rao. And I'm also honored to be speaking at this memorable occasion of centenary of the department where I graduated from. Take this opportunity to remember my thesis supervisor, Professor Ramshak, with us exactly a year ago when we began this year, the centenary year, and left us after he witnessed all the gala function and dinner. So I learned a lot under him, and I fondly remember the building photographs in being shown, where I actually graduated from. And at the expense of losing a minute or two, I want to recall an instance. GP, as you all know, and as was pointed out, would work at the bench. And I remember one day I had completed my dinner and come back, and I was in the common instrument room across from the office to weigh something in the fine metal balance. And there was GP ahead of me trying to weigh something. And I learned something a technique from him that I use to this day in the lab when I work. He was weighing a very small amount of something on a fine spatula. So he had his right hand holding the spatula with this fine powder on it, and he placed it above the weighing paper on the balance pan, and then with his left hand, he tapped his right elbow ever so gently so that really tiny quantities would fall at each tap. And I learned from him that day how to weigh very fine, small quantities. So this is something I can never forget. So I just thought I would share that one instance before I start my talk. I am going to share my screen. Not able to share. Yeah, can you all see my screen?
Yes, ma'am, we can see your screen.
You can see the screen. Okay. And so I will start with my talk. The title is Bangalore to Bellary, and it tells you about an outstanding student, Preeti Kumar, I had, who started this work of human cellular immune responses to Japanese encephalitis virus in my lab. And Bangalore to Bellary refers to the hospital in Bellary. It's a state government hospital where we went to, because Bellary lies at the heart of the Japanese encephalitis endemic districts of both Karnataka and Andhra Pradesh. And we would go all the way from Bangalore to Bellary by train to get these samples, because we didn't have any luck with NIMHANS, where we all had a collaboration, but it was highly unproductive. And also in this slide, I have shown you the 3 independent introduction of JV into the country. And much of South India, you have the Japanese origin strains of JV. So we would take the train, Preeti would take the train and go to Bellary overnight. She would have called all the patients by letter, and they would all have shown up. She would bleed them, set up fax stimulations, and board the train that night and come back the next day. And she would work non-stop for about 2 days with all the blood samples. And she was truly outstanding. And I would also like to take today's opportunity to acknowledge what a phenomenal amount of work she did, how original it was. She went to Bellary, she handled every aspect of the project, and she started this new area in my lab, which continued and really took shape in multiple ways. And I will go on to tell you how her work flourished in my lab long after she left. She is now at Yale as an associate professor. Goes to show how outstanding she was and continues to be. And you can see that in flaviviral immunology, which is one aspect of flaviviruses that we pursued in my lab at the MCV department. 5 full publications all came out of her PhD thesis, and a lot of other work was initiated by her and continued by other people. So this was a hospital where in JE endemic regions, because children are immunologically naive to the virus, you would only see encephalitis cases in pediatric patients. The adults, due to constant exposure, would have mostly become immune due to protective immune responses from natural infections. And to quickly show you the structure of the viral genome, it's a positive strand RNA virus, about 11 kilobases long. At the 5' end, it codes for 3 structural proteins: capsid, membrane, and the envelope. And then it has 7 non-structural proteins: 1, 2A, 2B, 3, 4A, 4B, and 5. And the virus particle is shown here. the same color coding that you see here. All these proteins are synthesized as a single polyprotein and then processed by host as well as viral encoded proteases. And the NS3 protein is a protease at the N-terminus and a helicase on the C-terminal half, the NS5. The largest protein is the polymerase. So, Preeti first put many of these genes in baculovirus expression systems, and using these proteins, she took a bunch of patients from Bellary, proved that they were JV exposed as opposed to the controls that she recruited, because their serum would immunoprecipitate metabolically labeled viral proteins from viral infected cell lysates. And then using these antigens, she set up T cell proliferation assays, thymidine incorporation. She's shown that the responses are very specific to the proteins of JV, and she incorporated various controls like BCG, phytohemagglutinin, which are either specific or non-specific T cell stimulatory agents. And she showed for the first time that you cannot distinguish based on antibodies a healthy control who's infected with JV but naturally protected from a patient, whether it is neutralizing titers or total IgG to the envelope protein. However, she showed that T cell responses clearly distinguish and interferon gamma production both from CD8 and CD4s are very robust in those who control JE virus infection and don't come down with encephalitis, as opposed to patients who come down with encephalitis. They have a serious shortfall in T cell stimulation and interferon gamma production. Not only did she show that interferon gamma producing T cells help to prevent infection progressing to disease, she showed that in those who recover from encephalitis, the amount of damage to the brain can be minimized if only the T cells of these people produced enough interferon gamma. And in this slide, you can see the beautiful Spearman's correlation coefficient, very, very significant, that those who have very high clinical scores and a lot of damage to the CNS following recovery from encephalitis hardly produced any interferon gamma from their T cells, whereas those who recovered with very little neurological sequelae, that means no permanent damage to the central nervous system, produced a lot of interferon gamma. So interferon gamma, Preeti showed, was important both to protect from becoming sick and in preventing sequelae and long-term neurological damage in those who suffered encephalitis. She then expressed multiple proteins of the JV virus, and using these recombinant proteins, she showed that NS3 was the primary target for T cells. She didn't just stop there. NS3 being a protease as well as a helicase, she made multiple C-terminal and N-terminal truncated constructs. Using them, she mapped the CD8 and the CD4 epitopes to a very narrow region which was housing the helicase domain, the active site. And she showed that multiple HLA alleles, both HLA class 1 and class 2 alleles, mapped to this region, and it was highly invariant because it was the active site of the enzyme, and the virus did not have the liberty or the luxury of mutating these epitopes. This is very important because subsequently when Lance Sturdy came from the University of Liverpool as a welcome postdoctoral fellow to pursue this study, and he used a series of peptides to cover the entire proteome of the virus, he basically proved that Preeti's data was very correct. These blue bars, which represent CD8 epitopes, are all very dominant in healthy individuals from the same Bellary region, whereas those who come down with JV, the JV patients, predominantly had CD4 T cells and to the viral structural proteins. These are the green bars. Each bar is an epitope. And this is the same region that Preeti mapped, the NS3 helicase domain. Lance confirmed that. And you can see in this little inset, in the healthy people who control the virus, the H index here stands for the extent of variability among thousands of JV isolates that have been sequenced. And you can see that these epitopes rarely show any variation. Whereas in JE patients, all these epitopes show a lot of variation. That means being an RNA genome, of course, there's a lot of variation in the sequence of these peptides. However, those who are healthy and control the virus have the ability to target by their T cells epitopes which are highly invariant. And that seems to be part of the trick for successful immune control of JE infection and preventing it from progressing to encephalitis. So Lance went on to show that these epitopes, which are highly invariant, are cross-reactive, that they are conserved among multiple flaviviruses, whether they are dengue, yellow fever, West Nile virus, and using peptides of those dengue and yellow fever and West Nile virus, he showed that there is complete 100% cross-reactivity. And using tetramers, Lance showed that it is the very same T cell which sees the JEV peptide as well as the dengue or the West Nile peptide. He also showed that the CD8 positive T cells, which are stimulated by MHC class 1 bound peptides of primarily the NS3 molecule of the virus, are highly cytotoxic. They produce perforin, granzyme, they degranulate, and they are cytolytic in nature. So primarily, in this first part of my talk, what Preeti and then Lance confirmed was to show that virus-specific CD8 plus cytotoxic T cells were the strongest correlates of protective immunity in naturally exposed individuals. And Preeti also showed that it is the NS3 protein of JV, which is the dominant target of human cytotoxic CD8 plus T cells. At this time, Lance got from the Chengdu Institute of Biological Sciences in China multiple vials of the SE14-14-2, which is the live attenuated Japanese encephalitis vaccine that was developed by the Chinese in the 1980s, and they were using it on their population with great effect, and it turned out it was extremely safe and very efficacious. And finally, in 2003 to 2005, the WHO reviewed a lot of the data and said that it was safe for human use. So Lance vaccinated a small number of people with this vaccine, and this vaccine, relative to its parent, the wild type SE14, has suffered 24 amino acid changes, 8 of which are in the envelope glycoprotein. And this is very significant because the envelope glycoprotein, this is just a structure of the viral particle, it's on the surface of the viral particle. There are 90 dimers of the envelope glycoprotein arranged in this manner in the virus particle, and all these changes, that 8 amino acids that the viral vaccine has mutations in, are all on the surface of this particle. So when we looked at these vaccinated individuals, we found that only CD8 T cell responses were boosted in those who were vaccinated with this live attenuated JE vaccine SE14-14-2. We did not see any such significant difference between natural infection, whether they are healthy controls or JE patients, in the CD4 subset. We only saw it in the CD8 T cell subset. So the vaccine was able to elicit a very strong CD8 T cell response in the vaccinated individual. So when we looked at the cell biology of this virus, we compared the wild type virus, the P20778 velour isolate, with the vaccine strain in infected mouse neuronal cells. And we looked at the ER stress sensors, we looked at basically ER stress pathways, and we showed that the PERK, which is the protein kinase, are like ER resident kinase, which is similar to the PKR of the cytoplasmic localized double-stranded RNA stimulated kinase, which phosphorylates CIF2 alpha and shuts down translation. The wild-type virus we found was able to degrade the PERK molecule, as a result of which we saw total dephosphorylation of EIF2 alpha, which allowed the virus, the wild-type virus, to have viral proteins translated without any block by the host cells. Then we looked at the CHOP molecule, which mediates the feedback dephosphorylation of phosphorylated EIF2 alpha. We found that the wild-type virus had also overexpressed the CHOP. It had induced CHOP, and the vaccine strain was very weak and unable to do that with the same ability. So this is the pathway that wild-type JV very efficiently knocks off both the upstream phosphorylation of EIF2 alpha. It also dephosphorylates phosphorylated EIF2 alpha, which is why you saw complete dephosphorylation of EIF2 alpha in wild-type JV infected cells. And this was accompanied by a huge boosted expression of all the chaperones in the vaccine virus infected cells, which told us that there was enormous protein folding stress. So we related this to the enveloped glycoprotein, which we knew from the sequencing of the vaccine strain was highly altered and had 8 amino acid changes. We therefore focused on the folding kinetics of the enveloped glycoprotein. So on the upper panel, you are seeing a metabolically labeled wild-type virus infected cell lysate, and the bottom panel is the vaccine strain infected cell lysates. So what we did was to flood the monolayer after we had labeled metabolically for 5 minutes with DTT, so that we reduced all the proteins. And here what we've done is to also treat the monolayer with actinomycin D to prevent the metabolic labeling incorporating any of the methionine and cysteine into the host proteins. So you can see the NS3 protein, the NS5 protein, and these are the envelope proteins. The envelope protein, soon after it is synthesized at 0 minutes, you can see in the wild type, it's already sufficiently oxidized. So this is the fully reduced form, and we freeze all these forms using alkylation with N-ethyl-melamine. And when we washed off the DTT after we reduced everything at 0 time point, that is at the end of 5 minutes of metabolic labeling, flood the monolayer with DTT for 5 minutes, 100 millimolar, wash it off, and then at 1 minute, 2 minutes, 5 minutes, etc., up to 5 hours, we follow it. We flood this monolayer with NEM just before harvesting so that we trap all the various forms of the sulfhydryl groups still present in the envelope. And it has 12 cysteine residues which form 6 disulfide groups. And if those are not yet formed, the disulfides are not yet formed, then the sulfhydryl groups will be alkylated. And you will be able to see the reduced mobility of the reduced form. It R1 as we call it, it goes through an R2 form and forms the oxidized. You see a perfect precursor to product relationship between the reduced and the oxidized form through this partially reduced R2 form. And there are a couple of intermediates which are much more visible in the vaccine strain. And what you will see in the vaccine strain, as I pointed out here, that there is selective degradation of this reduced form of the vaccine virus envelope glycoprotein, simply because it's unable to oxidize itself and move to the oxidized form. So after about 30 minutes, 15 to 30 minutes, the endoplasmic reticulum, which would have given it about half a dozen attempts at glycosylation and deglycosylation, passing between talnexin and talreticulin, to attempt folding, finally decides to send it out to the proteasome for degradation. And you will see, while NS5 and NS3 are very stable, only the envelope is highly degraded. And sorry, I went wrong place. So here I have only highlighted the envelope glycoprotein part of that gel, and we quantitated. You can see the rapid degradation of the envelope glycoprotein or the mutant vaccine protein, whereas the wild type protein is very stable, becomes oxidized, and this is also reflected if you look at already oxidized protein and ask, is it vulnerable to reduction by DDT? And for how long does it remain vulnerable? The wild type protein, after about 1 minute post labeling, becomes completely resistant to total reduction to the R1 fully reduced form, whereas the vaccine envelope remains vulnerable to reduction to this fully reduced form for at least an hour, beyond which it gets degraded, so you don't see it anymore. And what is able to fold to a more resistant, DTT resistant form, escapes this degradation in the ER. So when we looked at a whole panel of monoclonal antibodies that we raised in the lab, we found that the viral particles which are extruded into the culture medium are also not being recognized by monoclonal antibodies that were raised to the wild type protein. And the monoclonal antibodies in red were raised to the vaccine strain. And those, while they can beautifully recognize the protein, intracellular protein, one of them fails to recognize the virus particle. Whereas D10A is the only monoclonal antibody that recognizes both the wild type P20778 and the vaccine strain equally well. So we then looked at the processing of the pre-membrane protein to PR and membrane. So in the host cell, it is made as a pre-membrane, and while it is being extruded from the cell, while it exits the Golgi particle, Golgi stacks and gets to the surface of the cell, the furin enzyme cleaves the PRM. Now in all flaviviruses, this cleavage is very efficient, and viral particles will only have part of the PRM cleaved. On the other hand, we saw in the SA14 vaccine strain, the PRM was totally cleaved. We believe that this is perhaps because the envelope is not fully folded and furin has much better access to this furin cleavage site. Whereas in the cell lysate, you will see only the PRM, whether it's wild type or vaccine strain. So we think this is the take-home message that when you have a misfolded envelope glycoprotein and consequent unresolved ER stress, the entire cell's machinery, the autophagy that it induces, I did show you that data, causes a very efficient cross-priming of CD8 positive T cells by the dendritic cells cross-presenting. And we think this is why the vaccine is efficient. And we believe that this could be a universal approach for attenuating viruses, live attenuating vaccines. So with that take home, I will stop my talk. I want to thank the organizers for inviting me and all the people who did the work. Preeti, Ramarao, who was really the push to go to Bellary when we never got any samples Bangalore, all the people who helped. Bellary later on, Nancy came and did all the subsequent work with the cross-reactivity. Saurabh did the cell biology work, and Pallavi did. Thank you all. Are there any questions?
Thank you, Professor Vijaya. We can have just one question if there is any. There was somebody raising the hand some time back. I don't see. Okay. Thank you, Professor Vijaya.
Thank you.
Okay. Thank you for all the speakers. I think we just made in the right time finally. So let me hand over to the organizer to continue for the next session. Who is taking over?
So Sheetal will take over. Please give us a minute.
Okay.
Yeah.
Professor M.R.S. Rao for chairing this session. Now I'd like to welcome Professor D.N. Rao for chairing the Centenary Lecture. He is an honorary professor in our department and advisor at Challakere campus IISc. Professor Rao is a distinguished alumnus of our department and well known for his studies on restriction enzymes. He served for several years as chair, Division of Biological Sciences IISc. Sir, you are most welcome to start the Centenary Lecture.
Venki, online?
Yes, I'm here.
All right.
I don't know if you can see.
Hi, Venki. Yeah. It's indeed a great pleasure to chair this session. And I hate to say introduce Venki Ramakrishnan because I think everybody knows about him. But just for the students who have just joined us, recently, this is a small introduction, a very brief one. Venki Ramakrishnan is a structural biologist who shared the 2009 Nobel Prize in Chemistry with Tom Stites and Eda Yonan for studies on the structure and function of the ribosome. He is a group leader at the Medical Research Council, the MRC's most famous lab, the Laboratory of Molecular Biology, the LMB, on the Cambridge Biomedical Campus in UK. He is a fellow of the Trinity College in Cambridge and served as the president of the Royal Society from 2015 to 2020. But for many of us, especially students, I think I'm going to give you a little more introduction about Venki. Venki did his undergraduate studies at the Maharaja Sayaji Rao University in Baroda on a National Science Talent Scholarship, graduating with a Bachelor of Science in Physics in 1971. Immediately after graduation, he moved to the United States where he obtained his PhD, the doctorate degree in physics from Ohio University in 1976 for research into the ferroelectric phase transition of potassium dihydrogen phosphate. And then he spent 2 years studying biology, again as a graduate student at the University of California in San Diego, while making a transition from theoretical physics to biology. Professor Ramakrishnan began work on ribosomes as a postdoctoral fellow in Peter Moore's lab at Yale University, and he continued working on ribosomes as a staff scientist at the Brookhaven National Laboratory. In 1995, he moved to the University of Utah as a professor of biochemistry, and in 1999, he moved across the Atlantic to his current position at the Medical Research Council Laboratory of the Molecular Biology in Cambridge. Now, I'm not going to tell too much about his work because I'm sure he's going to give us a good introduction to the ribosome and the work. Ramakrishnan has obviously a number of distinguished recognitions. He was elected as a member of the European Molecular Biology Organization in 2002. He is a fellow of the Royal Society, the FRS, in 2003, and a member of the US National Academy of Sciences in 2004. In 2007, Ramakrishnan was awarded the Louis Jean Thétard Prize for Medicine. And in 2009, as I mentioned, he was awarded the Nobel Prize in Chemistry. And 2010, he received India's second highest civilian award, the Padma Bhushan, Vibhushan. And in 2012, he was knighted in the New Year's honors for the services to molecular biology. Now, he left India when he was 19 years, and only in 2002 he came back. He visited India to deliver the G.N. Ramachandran Memorial Lecture in Chennai. And at that time, Professor Balram, then director of the institute, invited him to come to IISc. And then he spent a number of years in IISc campus as a visiting Ramachandran professorship, and also the IISc DSc Centenary Chair, where he spent a long number of days on a campus giving lectures, talks, interacting with students and faculty. It's indeed a pleasure to have Venki, and we're very happy that he agreed to give this lecture. So Venki, it's all yours.
Oh, thank you very much, Deanne. First of all, can people, I'm going to start sharing my screen, and I just want to make sure that people can see it. So can people see the screen share?
Yes.
Okay, good. So first of all, it's a real pleasure to give this talk. I only wish I could have done it in person because, you know, visiting Bangalore is over the last almost a decade and a half now, has been, you know, one of the pleasures of my life. Just coming there, reading, writing, thinking, and just talking to people. It's been a really enjoyable part of my scientific life over the last 15 years or so. And so hopefully I can make it sometime in the future when things are back to normal. So I'm going to talk today about translational initiation, but I want to tell you a bit about how we got there and tell you a bit about the ribosome problem. So of course, everybody knows that the main problem of the ribosome is translation of genetic information in DNA, which goes via mRNA into protein. And the key player in this is really tRNA, which was first predicted by Francis Crick and then discovered by Zemechnik and others experimentally. And so what people didn't realize at the time was that this doesn't act on a naked template. Cell biologists found out that newly synthesized proteins were found in these particles on the endoplasmic reticulum. And when they were isolated, they were all about the same size, but they all seemed to consist of two subunits called a large and a small subunit. And eventually, in the late 50s, these were given the name ribosomes because they were ribonucleoprotein particles. And subsequently, a number of biochemists worked with ribosomes from both bacteria and humans, and they found that in both species, the ribosome is about 2/3 RNA and one-third protein. But the 1/3 part that's protein is actually split up among 50 different polypeptide chains. So there are about 50 proteins in the E. coli ribosome. And the human ribosomes are substantially larger. They're almost about 80% larger than the bacterial ribosome. Now, I won't go into all the gory details, but our big breakthrough in this field came when we solved the structure of the 30S subunit in 2000. And then a few years later, we're able to solve the complete atomic structure of the entire ribosome with mRNA and tRNAs. And this seemed like quite a difficult task at the time. But actually, if you wanted to understand how ribosomes worked, you had to, I don't know how this slide got in here. You had to go through this entire cycle, which is to ask, how does the ribosome begin translation? How does it add amino acids? How does it recognize the right tRNA and add the right amino acid? And how does it move along mRNA? And what happens when it comes to the end of the message? And so each of these states had to be painstakingly trapped using some biochemical tricks, and then tried to crystallize. And we were fortunate that we were able to get a number of these states, but you can see a key translocation state. This is the intermediate step in translocation. Is still elusive. And despite the fact that we were able to find one of the factors that allows the ribosome to initiate correctly very early on, we weren't able to make any progress, even in bacterial initiation. And so things didn't improve until a new technique was invented. And actually, I wonder if I can actually go back to that. This is the guy who invented electron microscopy, Ernst Ruska. He invented the electron microscope in 1933. He was in Berlin, I believe, at the time. And but soon after that, World War II broke out. And after the war, electron microscopy became very widely used, and people simply forgot that there was somebody who had actually invented it. And suddenly, almost 53 years later, the Nobel Committee woke up and gave him the Nobel Prize, and then he died 2 years later. So if he had, if they had waited another 2 years, he would have missed out on a really fundamental contribution to lots of areas. So how does electron microscopy work for biological systems? Well, the fundamental principle goes back to how you can reconstruct something in 3D from what are essentially 2D projection images. So when you have an electron microscope and you shine an electron beam through it, what you get as an image is a two-dimensional projection of the object. And if you look at it from a different angle, you would see a different projection. As if you were to look at your hand, and those of you who can see me can see, I'm looking at my hand this way, and now if I look at it this way, you'll see a completely different projection of my hand. Now, what Aaron Klug and his colleagues realized is that if you took these two-dimensional projections and looked at all possible projections, there was a way using Fourier transformation methods to reconstruct the original 3D object by combining all the 2D projections. And this is actually the same principle as when you get a CAT scan. So if you have a fracture or a cancer or something, and you get a CAT scan, you're getting a series of two-dimensional projections that the computer reconstructs into a three-dimensional image. So how does this work? Well, what you do is you take a solution of your particles and you put it on a grid. Now, a grid looks something like this, and this 1 millimeter scale shows you it's about 2 or 3 millimeters in diameter, and it's usually copper, or nowadays it's made of gold. And you have this mesh, and on the mesh is a carbon substrate or some polymeric substrate which has holes in it. And essentially what you do then is you take this grid, put a small amount of liquid on it, and then blot it so that the layer of solution is very, very thin. And then you simply plunge it into liquid ethane so it cools so rapidly that there's no ice formation. So it's essentially a glass, or what's called vitreous ice. Vitreous, of course, means glassy. So if you were to take a look at one of these squares, what you would see is this kind of substrate with these holes, and it's these holes in which the particles are visible, okay? So if you blow it up, then what you see this is an image of mitochondrial ribosomes, and you can see that there are lots of mitochondrial ribosomes, and they're in all different orientations. And if you blow up one of the particles, you see this extremely noisy image of these particles. And somehow you're supposed to be able to combine these different projections into a three-dimensional object. And I'm just going to show you a little movie made by Wolfgang Baumeister about how this all works. So you'll see that particles are sort of wiggling around while they're in solution, but when they're flash cooled, they suddenly stop moving. And that's the idea behind it. So here they are. So you can see the particles are sort of thermally moving around. And then when this solution is frozen, you can see effectively they're now at liquid nitrogen temperature and they're flash cooled. And they're essentially a thin layer. So the particles are in a thin layer. You have an electron beam. And what the beam does is it collects images. And that's what the two-dimensional images look like of the particle. And you get different views. And there are very sophisticated algorithms which will tell you which view of a particle you're looking at from the two-dimensional projection. So if you're then able to figure out what the view is and then collect all of the different views that you can get from your two-dimensional data, then you can eventually end up with a three-dimensional image of the object. Okay, and using this, basically, you can see how they reconstructed, this sort of helical filament in their reconstruction. Now, using this technique, Joachim Frank and his colleagues obtained the three-dimensional images of the ribosome. It was the first time that we could actually see mRNAs and tRNAs inside the ribosome. But of course, these structures were at very low resolution. They're what we call about 25 or 30 angstroms resolution. Those of us who were doing crystallography dismissed all this as blobology. What you saw were a bunch of blobs, and it wasn't going to tell you a lot about how the ribosome worked or about the chemistry involved in the various processes. But around the same time, Richard Henderson had published a theoretical paper showing that actually you could, if the conditions were right, obtain high-resolution structures by electron microscopy. And what really spurred a step change in the field was the advent of much better detectors, which are fast direct electron detectors, and better image processing to take advantage of the power of these detectors. For example, doing motion correction and classification of different conformational states or biochemically heterogeneous groups within the same sample. And so as a result, the EM field has taken off. And as you know, in 2018, Richard Henderson and Joachim Frank and Jacques Dubochet, who invented that freezing technique, or rather cooling technique, shared the Nobel Prize for cryo-EM. So we, of course, as soon as the technique became available, applied it to the mitochondrial ribosome. It turns out mitochondria have their own ribosome to translate the 13 genes encoded by the mitochondrial genome. And this is the work of two postdocs and then a graduate student, Nirupa Desai. What they did was to solve the mitochondrial ribosome from both yeast and humans. And you can see they're both very different from each other, as well as from the bacterial ribosome, even though mitochondria actually descended from bacteria about 2 billion years ago. So with this technique working, we thought it would be good to apply it to translational initiation, which had eluded us using crystallography. We had tried for almost 15 years to get crystals of various states and were never actually successful. And so it was a time to revisit the problem. Now I'm going to tell you the problem not in historical chronology, but rather talk about the latest work. And then I'll allude to some earlier work we did on initiation. So what is the problem with translating mRNA? So of course, when the genetic code was discovered, they didn't even know about RNA, about mRNA. But once they knew about mRNA, they had thought that, well, mRNA would simply have a coding region, and something would start at the beginning and simply go on until it ran out to the end of the mRNA. Well, very soon, people discovered a set of codons which were originally called nonsense codons, but which today we know are stop codons. And these are codons involved in telling the system that this is the end of the reading frame, of the open reading frame, and you have to stop making the protein at this point. Okay, and we've done quite a lot of work in the last 20 years on how stop codons are recognized in both bacteria and in eukaryotes. But then it was also realized that ribosomes don't just start at the beginning. There are a few mRNAs where, especially in bacteria, which are so-called leaderless mRNAs, where the first codon is really at the 5' end. But those are unusual. Most of the time, the initiator codon, which is typically an AUG codon, which codes for methionine, is at some distance from the 5' end. So the ribosome somehow has to find the correct AUG. There'll be other AUGs throughout the mRNA, and it has to make sure that it doesn't start on one of those. And of course, there'll be some AUGs which are not in the correct reading frame. So, it's very important to find the right AUG, because that also defines the reading frame for the particular message. So how does this work? Well, in bacteria, there is a special sequence called a Shine-Dalgarno sequence just upstream of the AUG, which is complementary to the 3' end of 16S ribosomal RNA. So that complementarity means that the ribosome is roughly positioned so that the AUG can enter the so-called P site of the ribosome, which is the middle tRNA site. And the precise reading frame is defined when the initiator tRNA actually binds to the 30S subunit and interacts with that AUG. Without that, the AUG doesn't actually have to be in the P site. It has some flexibility. But when the initiator tRNA binds, the base pairing between the initiator tRNA and AUG in the P site effectively set the frame for the beginning of translation. Now, this doesn't happen by itself. It actually happens with the help of 3 initiation factors, IF1, IF2, and IF3. And I'll be just telling you a bit briefly at the end. Actually, I'll just show you a movie about it at the very end. I'm going to focus mostly on translation in eukaryotes, where the problem is very different. In eukaryotes, there is no Shine-Dalgarno sequence, but the mRNA has a very special structure. It has a 5' cap. It's a modified guanine at the 5' end. Most eukaryotic mRNAs also have a long poly-A tail, and it has an AUG and a stop codon. And typically, what happens in eukaryotes is that the ribosome begins by binding near the 5' end, and then it has to scan along the mRNA until it encounters the AUG, and then that's AUG, when it reaches the P site, is somehow recognized, and a number of things happen. A bunch of factors fall off, and the large subunit joins, and then translation begins. So it's an enormously complicated process that requires a large number of factors. So I'm going to show you roughly how this works. So here you have the small subunit, which in eukaryotes is called the 40S subunit. You have the E, P, and A site. This is the aminoacyl site that brings in new tRNAs. The peptidyl site, which normally only in initiation brings in the new tRNA, which is the initiator tRNA. And this is the E site, the so-called exit site. So at first, what happens is a number of factors bind. So EIF1 and 1A probably bind first, although it's not clear that there's a specified chronology. And then you have EIF3, which is a large multi-subunit protein. It has 5 subunits in yeast, but it has about 13 subunits in humans. And in fact, in humans, the EIF3 alone is about the size of a bacterial 30S subunit. It's, you know, about 500,000 Daltons. And then what happens is subsequently you have a ternary complex of EIF2, which is also itself a heterotrimer, which binds initiator tRNA, and then also another factor, EIF5. So these form what is called the 43S complex, which is the complex of the 40S with these several initiation factors. Now, while this is happening, you have the mRNA, which, as I said, has a 5' cap, a start codon, and a poly-A tail. What you have is a poly-A binding protein, which binds to the poly-A tail. And you then have what is called the cap binding complex, which is also known as EIF4F. And this consists of EIF4G, EIF4A, and EIF4E. And this cap binding complex binds to the 5' end. And in particular, it's EIF4E that recognizes the cap. And there's also an interaction between EIF4G and the poly-A binding protein, so that in effect, in eukaryotes, the mRNA is often topologically circularized as a result of this interaction between poly-A binding protein and EIF4F. And then what this does is it recruits the 43S to the mRNA. So you then get the 43S binding to make what's called a 48S complex. And then this whole assembly basically scans along the mRNA until the AUG is reached. So now the AUG is reached and it can be recognized by the initiator tRNA. And at this point, a number of these factors fall off, and another factor called EIF5B binds, which facilitates the recruitment of the large subunit. And then you get protein synthesis followed by termination and recycling, and then the whole process starts over again. So we had unsuccessfully tried to crystallize a number of these components. And for a long time, certainly 2 postdocs, Tanwir Hussain and Jose Yasser, tried to crystallize this 48S complex. They had a lot of success with the 43S complex, but it was very difficult to get to the 48S complex using the yeast system, and we still haven't been able to do that. Before I go on, I just forgot to tell you that although there are about 12 factors in eukaryotes and only 3 factors in bacteria, the 3 bacterial factors do have counterparts in the eukaryotic system, so that EIF1 is like IF3, EIF1A is like IF1, and EIF5B is like IF2. So there is some common ancient core that has been conserved in initiation between bacteria and eukaryotes. So there are a number of crystal structures available. For example, there's a structure of EIF4E, there's a structure of EIF4A that is bound to mRNA. But what we don't know is how these actually work as a complex, and especially in connection with the ribosome. There is also another problem, and that is there are a couple of models for how translation can work. One model would be that you have the EIF4F binding at the 5' end, and that somehow recruits the 43S complex just downstream of it to bind to the mRNA. And so this would be sort of a downstream binding. And because the end is not accessible, the mRNA would somehow have to slot into the 40S or 43S complex. Another model which was proposed by Pestova because to explain mRNAs which had a very, very short 5' UTR was that the 4F somehow binds downstream at the entry site, and then this cap binding protein somehow dissociates and the mRNAs is somehow threaded into the ribosome, into the small subunit. So we thought a structure could shed some light on what is happening. So this is all basically a collaboration with Chris Fraser's lab at UC Davis, and where the biochemistry was done by one of his postdocs, Masaki Sokabe, also known as MASA. And so what we did was to look at a reconstituted system. One of the things the Fraser lab has done is actually purified all these factors from culture-grown human cells. So we have biochemically pure human factors, which is one of the big stumbling blocks. And they also had a long message, and we had to ask, is this system dependent on 4F? And what you can see is that if you look at whether it's reaching the AUG, so there are two AUGs here, this would be the first AUG it would encounter after it scans. And so you can see that when you add ATP, there is an enhancement in the cleavage at the AUG. So what is done is that you have an enzyme called RALE, which will cleave the mRNA in the A site where the ribosome has stalled, okay? And the reason that it can't proceed any further is we're not giving it the large subunit or anything else. So it will scan, reach the AUG, and then it'll stall. And so the question is, do you see the stall? And you can see that it's reached the AUG in all of the conditions, whether you have an ATP or a slowly hydrolyzable analog, ATP gamma S, or you have a relatively non-hydrolyzable analog. But what you can see is that it's significantly enhanced as a result of ATP or ATP gamma S compared to if you don't have, if you have AMP PNP. And of course, if you leave out EI4F, then you get very little or very slow cleavage. And so what this is telling us is that the system we are looking at is actually 4F catalyzed, it's 4F dependent. And so it's a physiologically relevant system. So this is the structure of the human 48S complex. And it is to date the highest resolution structure even of a 43S complex, because the data for the majority of the structure were extremely good. We were able to see 4A, 4G, and we could also see a blob, which we think corresponds to 4E, which is sort of over here. And the problem, though, is that 4A and 4G are at relatively low resolution compared to the rest of the structure. And this is perhaps because they're not bound as tightly to the rest of the complex as the other factors are. Now, one of the surprises from this structure is that 4A and 4G actually bound to subunits of EIF3 that don't exist in yeast. Okay, so this came as a bit of a surprise to us because you have 4F in both yeast and in humans, and so you would expect that it would have some common mode of binding in both humans and yeast, but actually it turns out that that it is quite different. So what can I say? So here is the 4F binding, and you can see that you've got 4F here and 4G here, and it's binding to these components, 3L and 3E, which are, as I said, not present in species. So what we think is that EIF4F in yeast will probably bind in a somewhat different way to the 40S subunit. But during evolution, with the evolution of EIF3, the sort of binding mode has also evolved to be different. Now, this is what I said about EIF4E, that we could see a density that corresponds to EIF4E. We can't be sure because at this resolution, it fits reasonably well and we think it's probably correct. And our mass spec data suggests, we did cross-linking mass spec, which suggests that 4E is likely to be where we're seeing it. So it does agree with biochemistry and it suggests that this is where the 5' end of the mRNA is. So the mRNA probably goes through 4A and into 4E where it stops. Now, one important thing we can deduce from the structure is because we know roughly where 4A and 4E are, we can calculate what is the approximate distance between that 5' end and the P site of the mRNA. And that distance turns out to be about 30 to 40 nucleotides, okay? What this means is that if you have an AUG that is substantially closer to the 5' end than 30 to 40 nucleotides, you will not be able to place it in the P site, so you will not be able to initiate on it, okay? So our prediction is that if the mRNA works by slotting, as our structure is suggesting, because our structure suggests that 4F binds on the 5' end, so the mRNA has to somehow slot into the groove in the 40S subunit, not be threaded in, as you would see from the alternative model. And if it's slotted in, then there's no way that a closer AUG could get to the P site because it would be, it would be sort of constrained because the 5' end is over here. So Massa and others decided to, and Chris Fraser's lab, decided to test this data. So what Massa did was a series of experiments. So he took mRNAs where he put AUGs, he always had an AUG at position 50, okay? But he then put a second AUG at position 10, 19, 30, 40, 50, and 60. And the prediction would be that you don't get initiation on these 10 and 19. You might get weak initiation on 30, but then 40 should be okay. And 60 should be weak because it should initiate mostly on the 51. So that's exactly what we see. So if you have it at 50 and 60, it goes to the first AUG that it sees, which is position 50, and then there's some read through, so you get a start at position 60. But if you get to 19 or 10, you see there's almost no initiation at 10 or 19. It goes straight to the 51. Same for 30. And with 40, you see some weak initiation at 40, but really still mostly the 51. And what this suggests is there's a so-called blind region. That is, there's a region of about 30 to 40 nucleotides from the 5' end where you cannot initiate, okay, using this mechanism. Now, what does this mean? Well, how's this relevant for human translation or translation in eukaryotes? If you look at where the AUG occurs, what is the length of the 5' UTR in humans? It's typically about 200. The median is about 200 nucleotides. So well above the minimum required of about 30 or 40. So there are very, very few mRNAs that have a very short 5' UTR where the AUG is so close to the 5' end that this mechanism would not be possible. And we think that for those particular mRNAs, they must have a different initiation mechanism. And this is the mechanism that occurs in really most messages that are cap dependent and 4F dependent. Now, the other thing is that 4A is a helicase. It hydrolyzes ATP. And the idea is that it somehow marches along the mRNA and melts the secondary structure. But in our structure, the 4A is behind the small subunit. So how could it march along ahead? Well, what we proposed as a result of this paper is that actually it's pulling the ribosome through, pulling the mRNA through the 40S subunit rather than unwinding mRNA ahead of the 40S subunit. And that's what our model for recruitment and for the blind region, all of that would be completely in agreement with that. However, what we've done recently is, but there are a number of other factors, I should say, that bind to the entry site, which are known to be involved in translation of mRNAs, which have very large amounts of secondary structure in the 5' UTR, et cetera. So there are special mRNAs which require additional factors in order to be translated. So what Jay Querido, I forgot to mention one of the most important people in this work, and that is Jay Querido, my postdoc, has been really spearheading all this work in my lab. And what he did was now look at a 48S complex with a much larger piece of mRNA, but which also has considerable secondary structure in the 5' UTR. And when he does this second complex, and here he has stalled the 4A using a particular sequence as well as a compound called ROCK-A. When he does that, what he finds is a 4F exactly where we saw it before, but now he sees a second complex of 4A near the entry site, which is to say on the other side of the 40S subunit. And he also sees an additional bit of density, which could be EF4B, which is thought to assist 4A in translation. So what is happening? Now we're saying that we might have more than one 4A. Well, I think it is possible. If you have a very easily translatable mRNA, you might not need the other 4A. On the other hand, for many mRNAs, you might require a 4A to melt downstream mRNAs as the scanning process proceeds. I think of it as, normally you would only one engine to pull a train. But if you're going from Mumbai to Pune, then you'll notice that it'll stop at Lonavala, where a second locomotive is attached, because you have a rather energetically difficult climb to make in order to climb the Western Ghats. So you can think of it a bit like that. If the energetic barrier is very high, you might need more than one 4A complex to bind. And I should say this second 4A is very consistent with work from John Lorsch's lab, not only in its location, but the fact that that binding is 4G independent. And that's exactly what we see. We don't see any connection between this 4A and the 4G. So you can have 4A that's part of the cap binding complex and possibly a 4A that is independent of the cap binding complex. And we're planning to test these ideas biochemically. So this is a sort of model for highly structured mRNAs. You still have 4F at the 5' end at the exit site of the 40S, but then now you have other factors, which could be 4A or other factors like DHX29, which help to melt the mRNAs as you go into the scanning process. So what happens when you reach the AUG? So this is really the work over probably 5 years or more of two postdocs, Tanveer Hussain and Jose Yasser. And I'm just going to condense, many years of work of theirs into 2 movies, okay? So this is to show you what bacterial translation looks like. And you can see that here's the 30th subunit now in bacteria. And you have the 3 initiation factors that bind, and they were able to see it by cryo-EM. And then what happens is that you get a series of conformational changes. So you have a conformationally variable head in the small subunit, and it's a result, and you can see that this is the neck that connects the body with the head, and you can see a compression of that neck helix, okay? So now you have recognition of the start codon by initiator tRNA, and what they will show you is that this recognition really involves a series of conformational changes which positions the tRNA properly into the P site, so more deeply into the P site from the initial state, okay? So you can see that you now have a sort of somewhat open state, and then you can see how it moves as the initiation process goes on. So effectively, you've got a more open state that transforms into what is called a more closed state, or sometimes referred to as a kind of P-in state, where the tRNA is inserted so that you get complete recognition, and then that signals the end of initiation. And this involves, as you've seen, very, very large conformational changes in IF3 during initiation. Now, what happens in eukaryotes? Okay, sorry. Okay, so eventually you get to a state where the IF3 promotes 50S subunit joining. Now, what happens in eukaryotes is that you get a very similar closure on start codon recognition. So even though the system is much, much more complex, and you have these huge array of factors around the subunit, you can see that recognition of the initiated tRNA involves a similar movement, a movement of the head and a movement of the tRNA accompanied by factors upon start codon recognition. And that start codon recognition then triggers a series of events, including first substitution of one factor by another, and then eventually a release of a number of initiation factors and binding of EI5B, and then followed by large subunit joining. So we're starting to make inroads into this very, very complicated process. I mean, we started this eukaryotic work in 2002. In fact, the first bacterial complex was published in 2001. So it's been almost 20 years of work after the first atomic structures of subunits came out, even to get to this point. But there's still a number of mechanistic questions required to really understand, how does it scan? How does it work? We still don't know how poly-A binding protein interacts with what the circularization is doing, and what is the conformation of the mRNA as the 40S subunit moves along during the scanning process. So lots of questions still to be answered. And I think I will close there, but I'd like to thank the people who did my work. So the eukaryotic work on the 4F complex was really driven by Jay Querido, and who was collaborated with Sebastian Kratz, another very talented postdoc in the lab, and Yulia Gordienko, who's a senior research associate in the lab. And the mass spec cross-linking work was done with a collaboration with Mark Scale, who runs our mass spec facility. And of course, this entire human complex work was a collaboration between Chris Fraser and Masa Sokabe. And this is a very fruitful and enjoyable ongoing collaboration. And I should say the earlier work that was just condensed into those two movies that I showed you was really many years of work by Tanvir Hussain and Jose Yasser, often with the help of Yulia Gordienko. They were joined in the bacterial initiation work by Brian Wimberly, who was spending a sabbatical year here. And all of it was a collaboration with John Lorsch and Alan Hinnebusch, who are two very distinguished ribosome initiation people who work on the yeast system at NIH. So thank you for your attention, and I will stop there now. Thank you. I don't know how to, I'm trying to stop my share, but, there it is. Okay, there we are. So I'm done. I see Umesh there. Hello.
Hi, Venki. A few questions. I don't know whether you want to answer them.
Yeah, I always dread questions from Umesh because he asked me a number of questions and I never have an answer to any of them. So so you can go ahead and ask the questions, and as usual, I'll say I don't know.
No. So 4F, you are saying it remains stuck. Up to what point does it remain stuck?
We don't know, because we don't know actually where we are on the mRNA, okay? Because the way the experiment is done, the different 40Ss could have stopped at different points along the mRNA, so that what we see for the bases in the P site is a mixture of bases, right? So we can't tell where it is along the mRNA. So we don't, and also we can't see the mRNA near the 4E because the resolution is so low that we don't see the mRNA there. So we don't know whether 4E is still bound to the mRNA or whether it has let go of the mRNA. There are two conflicting bits of data. One bit of data says it's bound throughout the process until the start codon is reached. Another says that it actually is released. And then, possibly you can have more initiation going on.
It would make more sense if it is released for reinitiation.
Exactly. So if you want to have a sort of, a queue of ribosomes all initiating one behind the other, then you wouldn't want the one ribosome to hang on to it until you get to the AUG. But I think that's not really clearly established.
One more point. I have few actually, but any clarity on how Kozak sequence works?
Not completely at the moment. I think Tanvir and Jose did have some ideas from their structure, but so maybe, I don't know if Tanvir is here, but maybe he will have a clearer idea. But
yeah, okay, yeah.
Umesh, can we ask somebody else to ask a question?
Yes, please.
All right. Muthukrishnan?
Hi. Occasionally, the GUG codon is used for initiation, at least in prokaryotes. Is that a case of a wobble?
it is, you can think of it as a kind of 5' wobble. But the main thing is that the reason that you can't tolerate it in a sense, during elongation, is because there the decoding is done in the A site, and there's very, very stringent requirements for Watson-Crick base pairing at the first position. That doesn't happen. That's not a requirement at the P site, and so that's why you can tolerate other bases at that first position. Oddly enough, it's more fussy about the third position during initiation, so which is almost the opposite of normal, decoding during elongation.
Yeah, that makes sense. Thank you.
Prasad?
Yeah, thank you so much. Dr. Ramakrishnan, that was an amazing journey to hear from you. I was looking forward to an answer to the following question. So we don't know much about how the AUG, the actual AUG gets selected at the end of the scanning journey, right? All we know is about the Kozak consensus sequence. I was hoping you might have some structural insights into how the ribosome,
No, actually, no, I thought I brushed over it by just showing you a movie, but actually there is quite a good idea. And the number of papers and, I don't know if you're on the ISC campus, but, Tanveer Hussain, who did a lot of that work, is actually a faculty member there. And in their papers, they've very clearly shown how base pairing is recognized when it reaches the AUG, and how the various factors like EIF1A actually go and monitor whether the start codon is recognized, and how that could lead to conformational changes that result in release of these initiation factors. So I think, although we don't understand it in complete mechanistic detail, we have a much better idea of how AUG triggers at least a series of changes, which then results in a transition to elongation. So I would say what you're saying was probably true 10 years ago, but it's not true today. But I simply alluded to it in a very kind of global view of it by just showing you the movie, it's sort of in broad context.
Nagaraj?
Great talk, Venki. And the question is how the internal entry higher side, the ribosomes.
Sorry?
Is there any insight how the translation?
No, I couldn't hear you, Nagaraj.
Can't hear you clearly.
Now I can't hear you at all.
We can't hear you, Nagaraj. No.
You're muted now. Yes, now you're not muted, but we're not able to hear you. At least I'm not able to hear you.
The question can be put in the chat box.
Yeah.
Patrick?
Yeah, so my question is what about the conformational flexibility of the head region in eukaryotic 40 complex? Is it really seriously constrained due to having multiple initiation factor compared to bacterial system?
I haven't compared the level of flexibility, but certainly the movement of the head is very similar in bacteria and in eukaryotes. In both cases, you have this swiveling movement of the head that allows an insertion of the tRNA deeper into the P site. So the direction of the movement and the sort of nature of the movement are similar, suggesting that perhaps initiation evolved in a common way and then really diverged between bacteria and humans, or eukaryotes. But I haven't compared quantitatively whether one is more flexible than the other.
Okay, thank you.
Pushot?
Yeah, hi. I am just curious the role of 4A in pulling the mRNA that you're proposing. Is this an extension of the role proposed in recruitment earlier by...
Sorry, could you repeat that, please?
Yeah, I'm curious the role of EIF4A in pulling that is being proposed here. Is this an extension of the earlier proposed role of EIF4A in recruitment by John Lorsch and Hinnebusch? And if it is so, then why would you need a helicase, a dead box RNA helicase to do the the recruitment role?
You would need a helicase because you need, so the ribosome itself, the small subunit itself is a helicase. That's known. For example, during translocation, it has been shown that the 40S, the small subunit in bacteria can act as a helicase. That's the work of Harry Noller. So we know intrinsically that entry channel can act as a helicase in order to melt secondary structures.
Too many things. Can I ask? Can I?
Somebody is interrupting.
Yeah, I wonder if people could mute their.
Yes.
Anyway, so, but the point is that if you have a helicase, you do require energy. In translocation, that energy can come from a couple of sources, from peptide bond formation itself, as well as from GTP hydrolysis by EFG during translocation. So where would the energy come from in order to have a unidirectional movement of the thing? So the idea is that you would have ATP hydrolysis by 4A, that you could have some sort of a Brownian ratchet. So for example, the 40S may stochastically move down the mRNA, but then 4A would hydrolyze ATP and move ahead, and that would prevent the 40S from going back. So effectively, you're using energy to drive it unidirectionally along the 3' end. So towards the 3' end or towards the AUG.
Thank you. There are two questions. One is, could the blind region be present in any random stretch of nucleotides after the start codon, or is there a sequence that predicts the presence of these blind regions?
No, the blind region is simply what I thought, maybe I didn't make myself clear. The blind region is simply the minimum mRNA 5' UTR that you need to be able to translate an AUG. That if you have an AUG that is closer to the 5' end than this 30 or 40 nucleotides, it cannot be translated by this normal mechanism. It might require some other mechanism.
And Nagaraj's question was, any insight into the IRES mechanism?
Yeah, there's a lot of work done, especially on cricket paralysis iris vectors, So we had a structure of cricket paralysis iris, which requires no initiation factors. And there, what is done is that the secondary structures of the mRNA mimic tRNAs in the E and P sites and in the A site, and then require a translocation by EFG in order to start translation going. Now, there are other irises which require varying subsets of factors. So some of them only require EF3, some of them require, some of them actually even require EF4F. But the common theme is that the mRNAs somehow mimic the tRNA AUG start site. And so you don't need to go to scan along until you find the AUG.
Right. One last question, which has come on the chat box, Venki. Do pulling actions support that presence of monosomes always and all the time for particular RNAs?
You mean support the thing of monosomes?
Monosomes, always in all time.
No, I don't think it matters. I think, this is related to what Umesh asked, whether the 4E releases the 5' end or not. In other words, does the 4F complex stay bound to the 5' end, or does it let go? If it lets go, then as it scans, especially if you have a median length of 200 nucleotides for the 5' UTR, then of course other initiation complexes could reinitiate on the same mRNA before the first one has even finished scanning. So I don't see this is not a pulling versus not question. That's what I'm trying to say.
One more question from one of our alumnus from the US. Could you speculate on why there appears to be differences between mitochondrial ribosomes in humans versus yeast versus bacteria? When would when one would have predicted more similarity?
I think the real reason is that mitochondria are evolving extremely rapidly. So if you look at the number of proteins that are synthesized by the mitochondrial genome, it's 13 in humans, but it's only, I think, 8 or 9 in yeast. But it can be as much as 40 in Tetrahymena. And then there are others like this malarial parasite where there are only 3 protein genes, okay? So the rate of evolution of mitochondria is extremely rapid. Moreover, there's no crosstalk between mitochondria of different species once they're embedded in their host species, right? So they're evolving independently of each other. So that's why, yeast mitochondria are not meeting human mitochondria in any recombinant sense. And so they're evolving completely independently for 2 billion years or so. And so I think that's the reason why they're so different.
All right, one last question. I think we'll stop here. Is the circularization a strategy to check if messenger RNA is intact and complete?
It could be a strategy to stabilize mRNAs, because as you know, mRNAs are often degraded from the ends. And so, by circularization, you're stabilizing the mRNA. The other advantage is that you're placing the termination close to the 5' end. So when the ribosome comes off and gets terminated and then recycled, the free 40S subunits will be close to the 5' end. So it might speed up reinitiation. So it might be a way to make translation more efficient. So that's the idea. But there is debate about how much circularization there is and how ubiquitous it is. And I have to say that the field is a bit confusing.
All right. Thank you very much, Venki, for this wonderful talk. And thank you for accepting our invitation to speak. Hopefully,
Thank you.
We'll see
it's
you.
a real pleasure. And I hope to see you all in person.
Sure. Thank you very much again.
Thank you.
All right. I'll hand over the thing to the organizers.
Yeah. Thank you, Professor D.N. Rao, for chairing the Centenary Lecture. Now I'd like to welcome Mani to
Thank you so much, Sheetal. Before starting and inviting the chair of this session, we would like to quickly re-announce some of the key points for our speakers and attendees. For speakers, each talk will be of 25 minutes, followed by a 5-minute question and answer session. We sincerely request all the speakers for this session to adhere to the time. We also request all the speakers to disable the annotation option from the View Option tab after sharing their screen. For attendees, questions are invited at the end of a talk by raising your hands or by also writing in the chat box. For our YouTube attendees, we request all of you to post your question in the YouTube chat box. Session chair will coordinate question either by allowing the attendee to ask verbally or read out the question from the chat window. With these instructions, I would now like to introduce chair of this session, Professor K. Muniappa, who is a distinguished alumnus of our department. He is well known for his studies on genetic recombination. He was a faculty and also served as a chair of a department for several years. Currently, he is an honorary professor and a CSIR Bhatnagar fellow. Over to you, Professor Muniappa.
Well, thank you, Mani, for that introduction. I would like to extend very warm welcome to all the speakers in session 2. Particularly, this session is a session which is going to be talks by our outstanding alumni. And as our brand ambassadors, they have made the department very proud. Can you hear me?
Yes, sir. We can hear you. Yeah.
Wonderful. We do really appreciate you all for being the flag bearers of this glorious department. Before we start this session, I would like to ensure that all the speakers joined us Subramanian is joined us.
Yes, I'm there.
Oh, wonderful. Mani?
Yes, I'm here. Thank you.
Danny?
Yes, yeah.
Wonderful. Finally, Dr. Bhatt? Maybe you will join us as we go along. Without much further ado, I would like to start this session by inviting our first speaker, which is Professor K. Subramaniam. Subramaniam obtained his PhD from the department in 1994, working with Professor S. Mahadevan. Subsequently, he moved to US to do a postdoctoral at Johns Hopkins in the worm model, which is C. elegans model. And there he did quite a nice piece of work. And he returned probably, I think, early 2000 to India and started his work first at IIT Kanpur, and he rose through the academic ranks very rapidly and became a full professor. Few years ago, he relocated to IIT Chennai, where he is a professor in the Department of Biotechnology. During these years, while in India, he has done pretty interesting work focused on understanding the roles of a class of RNA binding proteins in germ cell differentiation in the nematode model C. elegans. I guess today he's going to be talking to us about his work on a component of RNA biology in worm, which is the PLP1, which I believe encodes a component of which is a paralog of human single-stranded DNA binding protein, and it tends to suppress or in a way a number of endogenous genes all along the genome. It's my pleasure now to invite Subramanian to tell us about his more recent work. Subbu?
Yep.
It's all yours.
Thank you, Professor Muniyappa, for a nice introduction. It was quite generous introduction, I should say. And before I begin, I want to thank the organizers, particularly DN, for giving me an opportunity to talk at this venue. It is an honor indeed. And a little bit about my experience at the biochemistry department. I'm trying to figure out how to share. So is my slide visible?
Yes, we can see that.
Okay, that's great. So when I came to the biochemistry department in 1988, till then I have learned science only in textbooks without paying much attention to how the concepts and the details, like what Venki just explained, came into the textbook. Only at the biochemistry department, I became aware that a lot of research goes behind in finding out all those details. And that is where, I became aware of the process of research and scientific inquiry. So now I'll go ahead and share our recent piece of work, which actually starts with the PUF family of RNA binding proteins. So the PUF family of RNA binding proteins is an obsession in our lab. So probably the rest of my career also will be on that. This finding about PLP1 came while we were pursuing the RNA binding protein PUF8. This is a highly conserved protein present in all eukaryotes, in both animals and plants. So this cartoon highlights the importance of germ cells. Germ cells are the ones that are capable of undergoing meiosis and producing gametes, and therefore the biology of germ cells is really important to understand how sexual reproduction happens. And second, at the end of a generation, all these somatic cells shown in these blue circles, they will all die, so they end. And only the germ cells in the form of gametes continue their lineage to the next generation. So thus germ cells provide continuity between generations. So these aspects fascinated biologists for a long time, for more than a century, starting from the soma germ cell distinction recognized by August Wiesmann. And it continues to fascinate us as well. And we study them in the model organism C. elegans. So that is shown here. So it is head to tail about 1 millimeter long, microscopic nematode. I won't get into the details, which is, details are really very well-known textbook information. I'll only focus on two important points that make this organism an excellent model to study germ cell development. So as you can see here, highlighted by this dotted red line, are the two arms of its gonad. Okay, so these are mirror image like structures. So I'll just focus on one side. So this is a tube-like structure closed at one end as shown here. And then it comes and loops and then enters into a uterus. And this uterus is common to both these tubes. And these oval-shaped structures you see here are the embryos. So as you can see, the organ of interest, the gonad, is really large compared to the body size. And it's very easy to take this out. All you need is just make a dissection here with a needle and the gonad will come out like this. This is the closed end of the tube and this is what empties into the uterus. And in this gonad, the germ cells at different time points in development, the temporal stages, are arranged spatially in a sequential fashion. Okay, for example, the mitotic germline stem cells are displayed at this end, then the ones transitioning into meiosis follows, then a long packetine region is here. I marked it as meiotic region. Then each of these stages can be readily recognized by simply staining with the DNA binding dye DAPI. So this is the morphology of stem cells, which is quite different from the transition zone, different from the packetine, different from the oocytes. So very easily, even a beginning undergraduate student can very readily recognize the different temporal stages of germ cell development in this organism. So that is how C. elegans is a great model to study germ cells. And these meiosis fates are tightly controlled. I'm not getting into the details of it. Very shortly, a long story shown in this one sentence here. Puff family proteins are key regulators of these processes, that is mitotic proliferation, as well as the fate switch from mitotic to meiotic are tightly controlled by puff family proteins in a diverse group of organisms. Essentially, puff proteins are key for germ cell developmental decisions. So our laboratory studies puff proteins by focusing on puff8. So C. elegans has a number of puff family proteins, of which puff8 is the one that is closest in its similarity to the puff proteins of other organisms, including the human puff proteins called PUM1 and PUM2. So our work earlier showed that puff8 is expressed in the germline all through the larval stages. This is the newly hatched larvae. I'm just showing one section of the larva where the primordial germ cells shown here in green visualized by puff8 GFP. So starting from the primordial germ cells all the way through the adult, puff8 is expressed all through. And an interesting aspect of the null allele of puff8 is what attracted our attention. So this is the wild type male. This is the germline stem cells, and here are the gametes. Here it is the sperm. This is primary spermatocytes having finished the first meiosis, and this is sperm having completed both the meiosis. So this is the puff8 null allele, like completely lacking the open reading frame. It is fairly all right, although the regions may be shorter, etc., but it is functional. It produces functional sperm, which can mate a normal hermaphrodite and produce viable progeny. When grown at 20 degrees, this is the temperature at which we grow worms in the lab. However, when you switch to 25, a temperature at which wild type does perfectly all right, the perfect mutant germ cells will exit at the beginning of spermatogenesis, and they de-differentiate, returning back to the mitotic fate and make a germline tumor where normally you should see sperm. And exploiting this feature, that the temperature sensitive nature of this phenotype, a former graduate student, Muhammad Aris, he did a genetic screen looking for new mutant alleles in other genes that would enhance perfect phenotype. Meaning now the double mutant worm will be sterile at 20 degrees. So this is the wild type worm. This is undissected. The gonad is seen within the worm. So this is the germline stem cell end, and this is the meiotic region that I showed you earlier. And in one class of mutants that he found, there were no germ cells. So he called this as class 1. And another class of mutants where the mitotic proliferation continues and the worms, the germ cells never switch to the meiotic fate. So you have only mitotic proliferation, filling the worm essentially with germ cells only. So these two opposite phenotypes attracted our attention, and that is how finding out what is the role of perfate in controlling the mitotic-meiotic fate decisions became an obsession in our lab. So we are essentially focusing all the different mutants that Aris isolated and trying to find why they genetically interact, what is the biochemical basis for it. And so that is the kind of work we have been doing for the last 15 years or so. So while trying to map one of the class 1 type of mutants, Linita, another graduate student, she found that a mutation in PLP1. So this mutation was not isolated in Ari's screen. It is while mapping one of such mutants, she chanced upon PLP1 as an enhancer, a mutation in PLP1 enhancing perfect phenotype. And since that is the kind of mutations that we were looking for, she decided to focus on PLP1 phenotype. And this is the wild type that I already showed you. This is perfect single mutant, perfectly all right, except that it has less number of cells in each region. And this is PLP1, complete deletion of the open reading frame. I'll quickly tell you what PLP1 in the next slide. She found that the null allele is perfectly all right. It is almost as good as wild type. But however, the double mutant is extremely shortened and does not produce any oocytes. So to figure out what PLP1 does, Linita decided to look at where it is expressed, and so she did both, an antibody. She produced antibody and stained the gonads with it, and as well as she generated a transgene where PLP1 is expressed as a GFP fusion, and these are shown here. As you can see, it is expressed throughout the germline. And you see these circle-like structures. These are perinuclear granule-like things that I will show you close up in the next slide. And she also noticed similar perinuclear dots in somatic cells. This is near the head region where the nerve ring is located, as well as in tail region and in some of the somatic cells in the embryo. So essentially, it seemed to be arranged in a perinuclear fashion in germline throughout the germline development, as well as in somatic cells. So we were aware of this perinuclear granular arrangement of other proteins that are involved in RNA binding. Therefore, we decided to look closely upon it. So before I go further, let me introduce these perinuclear RNA granules in the germline. So these are actually granule-like structures, as you see here, particularly the lower panel, which is the 2-cell embryo, where you can see these dot-like structures. So these are RNA protein complexes. These are liquid droplet condensate kind of nature. They readily fall apart if you have a short change in temperature or if you lyse the cells. And we believe, like we meaning number of labs fascinated by these granular structures, believe that there are some evidences also. These granules are involved in translation regulation, RNA stability, RNA quality control, and so on. And anyone who works on germ cells usually get attracted by P granules. And we wanted to know whether PLP1 is on P granules. And as you see here, the first one, we wanted to see whether these granules are outside the nucleus or inside the nucleus. So we use this nuclear envelope marker, and you find that these are actually outside. And this PEGL one is a known component of P granules, and we find that it co-localizes. And in the embryo as well, we find co-localization, but not all the granules. Here you can see some of the green ones are PLP1 granules, but they are not co-localized with the PEGL1 granules, which is taken as the standard for P granule marker. So next, now let me tell you what PLP1 is. So PLP1 is an ortholog of this human protein called PAR-alpha. It is called PAR because it binds to purine-rich single-stranded DNA and as well as single-stranded RNA. And it has the PAR repeats. The alignment of the human PAR-alpha and PLP1 is shown here. Basically, you need to see that there are 3 regions where you see conserved region, and each one is a repeat. So 1, 2, and 3 repeats. And what is known about this protein is that it co-immunoprecipitates with other RNA granule components in cell culture studies, mammalian cells, as well as in Drosophila. So it is consistent, what we have seen, that its association with P granules is consistent with these observations. And cell culture studies have shown that the PAR proteins are involved in diverse processes ranging from transcription to RNA transport and translation. And there is only one genetic study, that is knockout in mouse, is lethal, primarily due to problems with the brain development. So this is what was known about PAR proteins. So in relation to PUF8, so I told you PUF8 is an RNA binding protein. This binds to 3' UTR and regulates translation. And since PLP1 enhanced PUF8 phenotype, we were wondering whether PLP1 also could bind to some of the targets of PUF8 and regulate their translation. So therefore, Vishnupriya, another graduate student, she decided to look at PLP1's effect on the translational control activity of 3' UTRs of some of the PUF8 targets. So the kind of assay we do for this is shown in this cartoon. We use this promoter and its downstream to express reporter like GFP-H2B in the germ line. And when you insert 3' UTR of your mRNA of interest, then you see the translational activity of this by seeing where the GFP is expressed. H2B is primarily to concentrate the signal in the nucleus and facilitate the visualization of the signal. And this we published a number of years ago, and it is a standard technique in the field. So we have several lines for potential targets of PUF8. Vishnupriya started to screen whether any of their expression is altered when she removes PLP1. And when she did this, she found one of them, which is PAL1. This we have shown earlier is a bona fide target of PUF8. This transgene in the wild type is not expressed anywhere when you grow the worm at 20 degrees. So indicating that this is not expressed at all, translated at all in the germline. And when she removed PLP1, now it appears in the oocyte. So these are the oocyte nuclei seen here. So suggesting or right away, you may be tempted to conclude that PLP1 controls PAL1 UTR mediated translation in the oocytes. But there is a twist to this story, and the twist is that this in wild type condition itself, if you grow it at 25, you see this expression pattern. And we know that transgenes normally are silenced in the germline, and we know through practice with other genes that if you grow such silenced transgenes at 25 degrees, you see expression. So this wild type PAL1 3' UTR itself, when grown at 25, you see the same pattern. So now the question is PLP1 regulating PAL1 mediated translation, PAL1 3' UTR mediated translation, or PLP1 is involved in transgene silencing in the germline? So this is what we wanted to figure out. And for that, she used other integrated, meaning chromosomally inserted other transgenes that are known to be silenced, and she wanted to test it. So before I go into that, a small introduction. Given the time constraint, I can only give you a brief introduction to this large field called gene silencing in the germline. So as I showed you in the first slide, germline is the one that continues from one generation to another generation. And the entire set of, the genetic blueprint you derive through the germ cells, and therefore its genome needs to be guarded and protected against foreign genetic elements that would like to get integrated into the germline. For example, viral genomes or transposable elements and so on. And there is a machinery that works to recognize and silence foreign genes, and the machinery works very similar to the way miRNA regulates gene expression or double-stranded RNA triggers RNA response. All of them, very briefly, use short RNA fragments derived from the original foreign material, and using that an RNA binding proteins, a set of RNA binding proteins called organelles, they find the targets and silence the target in one way or the other, starting from blocking promoter activity to degrading mRNA or blocking translation. So, so that is what is about germline silencing. So we wanted to test whether PLP1 is involved in it. And for that, she introduced other transgenes. For example, GOLD1, it is silenced in the wild type, whereas in PLP1 it is active. It's an integrated single copy transgene. And then this is another one, PEGIL1. Remember the P granule component I talked about? That again is activated in PLP1, indicating that integrated transgenes silencing requires PLP1. This is extra chromosomal array. These are repetitive, array formation of plasmids that we inject. That's definitely always silenced in the germline. That gets activated in PLP1. And these piRNAs are endogenously encoded miRNA-like molecules. These are tens of thousands of them. They are responsible for silencing primarily transposable elements in many organisms, and in C. elegans, many endogenous genes in the germline. And we use a sensor for assaying it, and that is again desilenced. So then another feature of mutations that affect germline silencing is temperature sensitive sterility. So I told you PLP1 perfect is sterile at 20, but PLP1 when we put at 25, that itself is sterile. So this is again consistent with PLP1 role in the germline silencing, because many mutants involved in germline silencing show this temperature sensitive sterility. Then another experiment that we did is several endogenous genes are upregulated when germline silencing components are removed. For example, PRG1 and MUT16 are well-established components. In their mutants, many endogenous genes are upregulated. We wanted to see whether the same set of genes are upregulated in PLP1, and you find a large fraction are upregulated among all three, consistent with our expectation. And when we take only the germline enriched subset of all the three upregulated sets, then we find a 76% overlap there. And very, intriguingly, 96% of them are associated with sperm related. So we still do not know. This is, some other time I'll talk about it if we understand better why sperm-related genes are particularly controlled by the germline silencing pathway. So the next experiment is at what stage is it expressed? PLP1 functions. Is it at the initiation of silencing or maintenance of silencing? For that, we introduce already silenced transgene, and we find it remains silenced after several generations. So this is a gene known to be involved in maintenance, and in its mutant it comes on the same silenced transgene. This is just a control. So this indicating that it is not required for maintenance, it's only required for initiation. And this is another example. I'll skip in the interest of time. And for example, PRGO, a well-established molecule involved in silencing pathway, in its mutation, small RNA production is actually affected. So we wanted to find out whether PLP1 is also involved in biogenesis of small RNA molecules, and sequencing small RNA molecules in C. elegans is little tricky because of a 5' modification. And Andy's lab is an expert lab, and they do it all the time. So I talked to Andy, and then we collaborated with them. And his postdoc Lamia, what she found is that this is PRG1 is the control, which is required for the biogenesis of piRNAs, and in the control you don't see the piRNAs being produced, whereas in PLPO and piRNAs are produced as much as in wild type, and similarly other classes. These variations are not statistically significant, showing that PLP1 is not involved in biogenesis of small RNAs. And to conclude, what I have shown you is that PLP1 is involved in gene silencing in the germ cells, and it seemed to function at the initiation step somewhere downstream of the small RNA biogenesis. So what we think in terms of how it functions is it probably binds to small RNA molecules and helps in interacting or taking the small RNA to the proteins like organoids that help in actually identifying the target and silencing. It probably couples the RNA and other proteins through its PER domain. So this is what we are thinking, but of course, a lot of experiments need to be done. So that's the first question. And the other one is, what is the sperm connection? So that is one intriguing question we would like to address. The third one, in the interest of time, I'll skip because I'm sure I have overrun the time. Finally, I would like to acknowledge the funding agencies. None of my lab work would have been possible but for the initial Wellcome Trust International Senior Research Fellowship that set us in motion. Then subsequently, ICAR and DBT and DST funded us. And finally, thank you for your attention.
Thanks, Subbu. We have some time for a couple of questions. Can't see the hand sign. Preferably, I would take questions from students. Any questions for Subbu? Well, Subbu, there seems to be any questions for you. Looks like your presentation was crystal clear. And we thank you for telling us your very interesting work, and good luck with your future directions.
Thank you. Thank you very much. Thanks for the opportunity.
Well, with that, yeah, Subbu, you have something to say?
No, I'm trying to figure out how to stop screen. I'm going to stop.
I can log. Okay, meanwhile, I have a great pleasure in welcoming our next speaker, Sendurai Mani. Mani is ever looking young. Mani graduated from our department in year 2000 with working with Professor G. Padmanabhan, and he moved to MIT to work with Professor Robert Weinberg in the field of cancer biology. And year 2007, Mani moved southwards to Texas to start his own lab at the MD Anderson Cancer Center in Houston. And in very recently, he was elevated to the rank of full professor in the Department of Translational Molecular Pathology. Mani's research program is mainly focused on understanding the signaling pathways during epithelial mesenchymal transition in a set of cells contexts in cultured human cells, patient-derived xenografts, and transgenic mouse models. And his lab is also looking for some cancer therapeutics. So that said, a great pleasure in welcoming Mani to request him to make a presentation of his work and give us some insights into cellular plasticity and its significance in cancer metastasis. Mani,
thank you.
It's all yours.
Thank you. Can you hear me?
Yeah.
Wonderful. Thank you. Thank you, Dr. Maniappa. And, it gives me great pleasure and honor to give a lecture in this Sentinel Symposium. And so, in 1993, when I walked into the biochemistry department, it was like a dream come true. So I came for an interview on the previous day night. So I didn't see the department. And the next day when I came for an interview where Dr. Maniappa, Dr. D.N. Rao, Dr. Naraja, and almost everyone was there in the interview, it was like, this was scary. But, it was wonderful at the same time. They made you comfortable, and it was really more of a discussion rather than, do you remember or do you understand? That was the thing which they were trying to understand. They were trying to find out. If you understand, they're fine. If you remember, that's not what they were looking for. Very nice. That really gave me a different perspective towards science, how we should think science. And then I got I was fortunate to do my PhD with Professor Padmanabhan. He gave a lot of freedom. So at the same time, so we got Dr. Rangarajan also joining the department as a faculty. So he was there in-house teaching us how to culture bacteria, do a column purification, whatnot. All the techniques I was fortunate to learn from Dr. Rangarajan. So this is a picture. I just have some picture I thought I would show. This is a picture. For whatever reason, my colleagues at that time, they were trying to push me into Professor GP's chair. I have no idea why. We were all having that fun at that time. And we used to work very late. And here is a lab, the old building. And here is a picture from the group. It was a great time. I thank and I really treasure every moment I spent on that campus. And here is a place where I came from Madurai. So in Bangalore, it was very cold on those days. So I used to wait for sun to come out. So you can just go out from the lab to stand out. And then, here is a place where all of us were just spending time. So in 1999, I moved to Bob Weinberg's lab. And that's where, it's Bob Weinberg. I started, I developed interest in understanding the biology of cancer metastasis. I will explain to you why and how, and then the significance and importance of this. So if you look at a cancer metastasis, it's quite a complex process where you have a primary tumor, and then the tumor become highly vascularized or become angiogenic, and then once the tumor cells at some point start leaving the primary site, getting into the vasculature through this intravasation. Now, the cells leaving the primary site travel to a distant site, they extravasate at a distant site, and they remain dormant for years, many years. At some point, they choose to proliferate and then create a new tumor or metastasis. Now, the metastasis could reseed and create more secondary metastasis, so it goes on. So why do we care? The reason is, in this slide, it shows first come, first saved. For example, if somebody has a localized disease, independent of the disease subtype, cancer subtype, they tend to do better. For example, in prostate cancer, you can see the survival rate is extremely high if they have been diagnosed early when the tumor is localized. But there is a metastasis, then the survival rate is extremely low. So that's true for breast cancer, that's true for many cancers. So in India, you can notice that 7 in 10 patients, this is from India Today, patients, they walk to the doctor only at the terminal stage, which means they would have developed metastasis. So it's very important for us to understand the biology so that we can treat metastasis. At this point, we don't have many option to treat metastasis, but we can treat primary tumor very well. So the reason why, again, I got interested in this area is because when I joined Bob Weinberg lab in 1999, these two guys, they were taking Bill Hahn and Brian Ellenboss, they isolated normal human mammary epithelial cells. To that, they introduced SV40 early region telomerase and V12HRAS. This is inactivates SV40 inactivates large T and inactivates RB and P53, and the telomerase gives the continuous proliferative potential, and the HRAS kind of transforms them. When they did that, one of the very important observation they kind of noted was they were able to transform them by, when you inject them into the mouse mammary fat pad or mouse breast, they were able to form a tumor, but they were never metastatic. So that's when I got interested in saying, how can I make these tumors metastatic by introducing known set of genetic elements? So, so to cut the long story short, so if you look at a normal tissue, any breast tissue or any normal tissue, they are all beautifully organized, and they continue to express these various cell adhesion molecules such as tight junction proteins, gap junction, desmosome, etc., etc. This is what holds them together in one place. When a cell like this get transformed into a cancerous, they continue to express all these cell adhesion molecules. As a result, they remain local. So we hypothesized that a transformation will lead into generation of non-metastatic tumor, but in order for these cells to migrate from point A to point B, they need to break cell adhesion, and also they need to break cell matrix interaction. And we tested and proved in number of papers we published showing that this is what is happening during metastatic process. So I'm not going to go into that for the interest of time. So in addition, one of the another puzzle is that a pathologist, when they look at a tumor, let's say in a lung, and they could predict whether that particular tumor is coming from breast, prostate, or lung, or liver, or spleen, just by looking at a histopathology. And what we found previously is that if you take an epithelial cell and they undergo this EMT, become mesenchymal, and that's what lead to development of metastasis. So therefore, I hypothesized that cancer cells not only become mesenchymal following EMT, rather they become stem cells so that they could go and recreate a tumor histopathologically similar to the tumor where they come from. And in fact, that turned out to be true, and we published this story in this paper, which is one of the highly cited paper, which kind of shows that this paper made a huge impact beyond my lab. So this brings us to the next important problem, which is today, if you look at a tumor is made of both differentiated cancer cells and cancer stem cells, and the treatment what we have, or the conventional treatment, which predominantly targets differentiated cancer cells. As a result, you can reduce the tumor bulk, but then the cancer stem cells are not eliminated, and then they eventually proliferate and differentiate and create either tumor relapse or metastasis. And if you have a cancer stem cell specific targeted therapy, then you would be eliminating cancer stem cells. If you, in my, what we found, that even a differentiated cancer cells can de-differentiate through this EMT program, and then they can proliferate eventually differentiate and create a tumor relapse. In other words, targeting cancer stem cells alone is not sufficient. So you need to combine both. You need to target both cancer stem cells and differentiated cancer cells. So our finding suggested that one could target EMT to eliminate cancer stem cells, and these are the things we are doing in the lab now, combining EMT specific inhibitors and the differentiated cancer cell inhibitors, and then see whether we can eliminate tumor. So I'm not going to discuss that part of the story. Rather, I'm just going to give you a paper which we just got accepted. This is showing that CD8 T cells inhibit metastasis, and then CXCL4 regulates its function. This story was led by Rubia Joseph and collaborated with various folks shown in this picture. Sorry, my slide is it's a big file. So similar to the story I was mentioning earlier, not only tumor cell can change its behavior from non-metastatic to metastatic, even the immune microenvironment can change from tumor killing immune microenvironment to immune suppressive and tumor promoting microenvironment. So you have various immune cells shown here. But the way we systematically approach this problem is by taking a two tumor line, one is called a 67NR, and one is called 41. They both are syngeneic tumor line. In other words, you can inject these tumor cells back into the mouse with an intact immune system as long as you are injecting them into the same background. These are BALB/C derived cell lines, so you have to inject them into the BALB/C mice. When you do that into the mouse breast, both develop tumor with the equal rate, which is shown in this panel B. But if you look for metastasis, these are luciferase labeled, so you can look for a metastasis to the lung. But again, we are looking for a metastasis from the mammary fat pad to the lung. When you do that, you see only 41 goes to the lung, but not 67NR. And you can also look for circulating tumor cells. You see only 41, the highly metastatic one, has metastasis or CTCs in the blood. So now, if you take the primary tumor of the two tumors 67NR 41 and look for CD31, which marks angiogenesis, there is no difference. If you look for a proliferation, there is no difference. But then when Rubia put these cells in suspension culture, asking can they survive in suspension, it's kind of mimicking what happens in the blood. They were able to survive equally, both of them. This suggested that maybe if we introduce these cells directly to the vasculature into the blood, they may be able to survive and develop metastasis. In fact, that's what she did. She injected, again in a wild type mice, she introduced 67NR and 41 through the tail vein. Now she was able to see metastasis to the lung, suggesting that here is a entire lung loaded with the tumors, both in 67NR and 41, suggesting that what they were missing, particularly the 67NR, was ability to get into the blood. If they manage to get into the blood, then they can complete the rest of the step. So what is the thing which is controlling the entry of 67NR going from primary site to the blood? So we thought instead of looking at a tumor cell point of view, we will look at it from an immune microenvironment point of view. So for that, she took not-skid mice, which you all know don't have a T cells, B cells, and NK cells. So introduced both 67NR and 41 into the mouse breast. And to our surprise, in this mouse, they found a tumor with a similar rate, the primary tumor. But if you look for metastasis, even 67NR was able to develop metastasis. And you can see there is no difference in number of CTCs. This suggested to us that what was missing or what was stopping from 67NR developing metastasis is the immune cells, but it could be T cells, B cells, or NK cells. So then we have a BALB/C nude mice. So we bought a BALB/C nude mice, injected both the cells into the mouse mammary fat pad, and you could see again they formed a tumor with equal rate. And if you look for a metastasis to the lung, we were able to detect metastasis even in this case by 67NR. The nude mice, they don't have a T cells. So now we have narrowed down to T cells and eliminated the other two cell types, B cells and NK cells. Now within T cells, as you all know, you have many subtypes. So we collaborated with Sean Zhang and he backcrossed CD8 knockout mice into the biopsy background, and then in that Rubia injected either into the wild type or into the CD8 knockout mice. For some reason, the tumor grew pretty big in the absence of CD8 T cells. And when we look for metastasis, in this case, she's putting only 67NR. In the absence of CD8, the 67NR cells, otherwise non-metastatic, started developing metastasis or migrating from mammary fat pad to the lung. So this kind of, we also tested CD4. It is not CD4, and maybe I don't have that. So it's not CD4, and we reconfirmed that this finding using function blocking antibody as well. So now we want to know how CD8 is regulating metastasis. So when she took cells 67NR41 either grown in a petri dish or grown in a wild type mice or in a nude mice, and she performed a growth factor or cytokine array, what she found is that the expected kind of cytokine was something which should not be expressed when these cells are cultured in a petri dish, and it should not be expressed in a cell 67NR when it is injected to the wild type, but it should be expressed in 67NR when it's put in a nude mice. It should also be expressed in 41 in a wild type as well as in nude mice. So we scanned number of cytokines. The only one fit our hypothesis, our expectation, is CXCL4. I will explain to you what is CXCL4. CXCL4 is it's a cytokine predominantly secreted by platelet. It's also called a PF4, platelet-derived factor 4. What Rubia did, she bought a CXCL4 from a company. She administered mice having a 67NR tumor with CXCL4. And what she found, even just by administering CXCL4 in a wild type mice, she was able to make a non-metastatic 67NR tumor to become metastatic. And you could see more CTC, suggesting that CXCL4 is one of the rate limiting factor for these cells to develop metastasis. So another thing we wanted to know is how CXCL4 interacts with the CD8 T cells, right? That's something. So the CXCL4 is known to act through CXCR3. So she went ahead and blocked CXCR3 in a highly metastatic tumor, which is the 41, and she was able to reduce the metastasis of those cells as well in a 41 in a wild type mice. So now the question is, who is producing CXCL4? We know it is previously it is known that it is platelets, and we reconfirmed it. We grow the cells 67NR and 41 in a petri dish. They don't produce any CXCL4. You isolate, these cells are labeled with RFP, so we put them in a biopsy wild type mice and then isolate RFP positive cells of 67NR41. They don't produce any CXCL4. We also isolated CD45 positive fraction, which is an immune cells, and they also don't express any CXCL4. The only platelets, if we isolated from these mouse tumor, they produce CXCL4, and you can see we went ahead and checked their presence in 67NR41, and you can see the CD8 is highly abundant in 67NR, but in 41 they are less. But if you look for a CD61, which marks platelets, they are low in 67NR and slightly high in 41, and that's quantified here. In fact, we did something called IMC CyTOF, where you can label nearly 30 antibodies with the different metal, and then you can label cells in these tumors and look for presence of these various cell types. And what you see here, particularly CD8 cells, 67NR has more CD8 T cells compared to 41. So we went ahead and also tested the same idea in a human tumor tissue microarray here in collaboration with Dr. Fei Yang. So you can see patients with CD8 positive cells tend to have a low CD61, and the patients who had a low CD8, they had more CD61, and which is quantified. Most importantly, the patients who had high CD8 and low platelet, they did better compared to the patients who had a low CD8 and high platelets. So overall, this kind of told us that the tumor cells they are, they trying to develop metastasis, but CD8 T cells puts a brake on them, and platelets by somehow promote the metastasis, which is kind of known in the literature, the right part, not the left part. So then we were, Ruby, I was interested in understanding how platelet is able to manipulate CD8 T cells, or how the CD8 T cells manipulate platelets. So what she found is that when you take CD8 T cells, incubate with platelets, for some reason platelets turn on annexin 5, suggesting that they may be dying. But the issue is they don't have nucleus, so we didn't pursue that further. Rather, we asked, can platelet manipulate or modulate the function of CD8 T cells? So she isolated platelets or the CXCL4 and then incubated them with the monocytes. The reason is we do know that MDSCs are another cell type. So just one step back, when you take a platelets or CXCL4, incubate them with the CD8 T cells, she observed nothing. The platelets or CXCL4 on their own not able to impact the function of CD8 T cells. So therefore, we hypothesized that it must be acting indirectly through a cell type such as MDSC. So she took platelets and CXCL4, incubated with the monocytes, and asked, can they make more MDSC? In fact, we were able to make more MDSC. And those MDSCs were able to go and inhibit the function of CD8 T cells. I'm not showing you all the data for the interest of time, but we could demonstrate that platelets as well as CXCL4 produce more MDSC from monocytes, and those MDSCs inhibit CD8 T cells, and that's how it works. So here is another data where we inhibit MDSC function, which is myeloid-derived suppressor cells. You could see in a 41, the highly metastatic tumor, the metastasis is inhibited. The most important finding is there's another postdoc, his name is Suhas Vasekar. He used to be in my lab. He took entire TCGA data set and he analyzed the correlation of MDSC with platelets, and you could see that whenever you have a platelets, they tend to have an MDSC across various cancer types, not just in breast cancer. If you look at a CD8 and MDSC, which is known in the literature that they are negatively correlated. But this is another thing which we found, that if you take a CD8 and platelets, they are also negatively correlated across various cancer types, not just in breast cancer. And they also predict, this various combination predict poor clinical outcome. For example, here is a survival probability where you have a CD8 low, platelet high, MDSC high is bad for cancer patients compared to CD8 high, platelet low, MDSC low. So in summary, this part is that the tumor cells in general, the CD8 T cells inhibit tumor cell migration, invasion, and metastasis, and CD8 T cells also inhibit platelets. So as a result, you have a high CD8 and low platelets. Under that condition, you tend to have no metastasis. But at some point, we don't know how and what that triggers, that platelets find a way to survive. Once they overtake it, now they are able to promote the migration, invasion of tumor cells. Simultaneously, they make more MDSC, which inhibits CD8 T cells. Collectively, this leads to metastasis. So that's a summary of the paper. Finally, in the last 2 minutes or 3 minutes, I just want to present you with a new initiative which we started. Probably you all know precision medicine, particularly the undergrads and the grad students, where if you think about patients with the treatment, like most of the treatment works for only one or few patients. For the rest of them, it does nothing, particularly the cancer treatment. So here is the data, like the conventional treatment is you take cancer patients, you give a standard treatment. So some patients benefit from that, but not everyone, right? For some patients, there's no effect at all, as is shown in this graph. But for many patients, they also have a side effect, so which is a common problem today. So the precision treatment, where you cancer patients, you profile them based on various genomic profiling, then you separate them into group A, B, and C, and give their respective treatment, right? If they have a mutation in a particular gene, and you give their treatment. If they have a mutation in particular condition B, and you give their treatment, and now you achieve effective treatment. So this is a classic example where a melanoma patient with the BRAF mutation was given with the vemurafenib, and you could see a patient with so many tumors all over. The patient recovered significantly in panel B, you could see, but the tumor relapsed, right? That's a problem because the tumor is constantly evolving unless otherwise you eliminate every single tumor cell, which is difficult. But at least this patient had another 23 weeks of extra survival, and hopefully they find another mutation and you could go and target. So today, it's kind of where we are is that integrating various omics data and then putting them all together and treating them. So that's kind of a future. So the way I see is that you have a various omics, feed them into supercomputer that can go into the phone, and that will create cocktail of less toxic medicine. The patients recover. So unfortunately, this is far away from today. We will get there, but not today. So what we did, we created a small consultation company where these guys plus many others, they all joined us as advisors, and this is what they do for their living. They look at these profiles and then they predict medicine. So fortunately, they all agreed to work with us and through the phone, and we can use their expertise to design these less toxic medicine. So we just formed a company called Ilon Precision Oncology. It's kind of very moderate fee, just more like a service. This Dr. Seventhi Limaye and we started this company called Ilon, which is for a precision medicine. She's the one who treated Sanjay Dutt, for example, whom now considered cancer-free almost because she applied precision oncology. So this is another thing which we are almost there. Probably in a month we will be out there, but I just want to put this idea out that particularly in an Indian context, this would be very beneficial for patients with advanced cancer. So in summary, I've shown that EMT program play a central role in regulating metastasis. EMT program induces stem cell properties on a differentiated epithelial cells. The CD8 T cells regulate metastasis in a platelet-dependent manner. So these are the people whom I collaborated. I didn't get a chance to speak a lot of work by these folks. So this is my group. So this is a funding support, and I thank you all for your attention. Thank you.
Thanks, Mani. And any questions or comments for Mani?
Dr. Mani, this is Suraj Bhatt from UCLA. How are you doing?
Good. How are you?
That was a terrific talk, and so was Dr. Subramaniam's talk before that. That was a terrific talk. I was just wondering that since 80 to 90% of all cancers come from epithelial cells, how critical is the EMT? Does EMT happen before metastasis or after metastasis? Is it critical?
It's extremely critical. So originally, when we hypothesized that EMT is critical, we were trying to understand that whether cell need to undergo complete mesenchymal transition, or can they become mesenchymal, just acquire some mesenchymal properties. So what we found today, or what we know today, is that the cancer cell need not become mesenchymal. All they have to do is they have to inherit some mesenchymal properties. So that kind of, when they inherit some mesenchymal property, they also tend to get some stem cell properties. Together, that helps them to leave the primary site, manage to survive in the blood. The key is ability to survive, and then establish a tumor at a distant site. So it's EMT is critical, not like the way we originally thought in 2000. You need to become mesenchymal, but you don't have to lose epithelial.
So if I may, can I ask a follow-up on that?
Of course you can, yeah.
CD8 knockout cells, what is the proportion of stem cells, tumor stem cells in those mice?
It's a good point. So that's something we are looking at right now. In other words, so originally we looked at it from a tumor cell point of view, right? How the tumor cell will manifest the immune microenvironment, not immune microenvironment, the metastasis. So then now we looked at how the immune cells manifest or manipulates the tumor cells. One of my postdoc, he took 15 different mouse lines, syngeneic line. We try to use mostly syngeneic line simply because that allows us to model immune cells as well. And then he's asking, what is the contribution of EMT in manifesting the immune cells? So he has a nice finding showing that number of immune cells, particularly the immune suppressive cells, they get turned on following activation of this EMT program. So we're trying to understand that. So
that's a terrific interface between stem cells and immune system. Thank you.
Yeah.
So can I ask a quick question, Dr. Muniappa?
Of course you can, Sukumar. Good to see you.
Yes. So nice to see you again. I mean, a lot of you talk tomorrow. Sundar, a great talk as always. I'm always a big fan of your work, as you know. So nice to see you as well, and good morning. I had a very quick question. The CXCL4 data you showed, do you have any data or any information on how it regulates stemness or EMT per se, or does it have any effect on the stemness directly, or the effects you are seeing are totally CD8 mediated cytotoxicity, that effect? Is there any data available on CXCR4 expression on the stem-like cell? Does it change during EMT, anything like that? Yes, you are still mute. Yeah.
Somebody muted me. I didn't mute it. So, it's a good point. That's another thing we are kind of puzzled. Like, what triggers tumor cells to, or the platelet? When the platelet manage to survive in the tumor microenvironment, then they start producing the CXCL4. But how the CXCL4 impacts the tumor cells, we don't know that. We don't know. We didn't explore that. All we know is if you add a CXCL4 to a tumor cells, they tend to express CXCR3, the receptor. They express more, they become more highly migratory and invasive and all that. But we don't know its impact on stemness. It's a good point. Thank you.
Kumar? Yeah, Kumar, yes.
Mani, this is Kumar from MCB department. Please let me correct me if I'm wrong. What I understood from your talk, you mentioned 67 NR breast cancer cell line, which is not metastatic to begin with, became metastatic in a nude mice. Thereby, you identified CD8 cells are the one responsible for inhibiting their ability to metastasize. Okay. Now, earlier you have shown the ability of cancer cells to metastasize depends on their ability to undergo EMT. Okay. And which is also kind of overlapping with the stem cell phenotype, which you had discussed when you came to give a talk some time ago, a couple of years ago. Now, can you please reconcile this the ability to undergo EMT, stemness property, that makes them metastatic versus in today's finding that absence of CD8 T cells only made 67 NR metastatic. That does mean 67 NR is not EMT cell line. Is that the reason originally it is not metastatic? And can you please combine these things and give your opinion on that? Thanks.
Absolutely. Thank you. That's what we are right now examining right now as you speak, as you just said. So again, in a tumor, you have epithelial, you have a mesenchymal. There are something in the middle. They kind of acquired mesenchymal, but not completely mesenchymal. So the 67 NR is more of a mesenchymal. So when a tumor cell in the primary site itself, they undergo this extreme differentiation, they somehow lose the ability to develop metastasis. But if they are extremely epithelial, then also they don't. Probably the immune cells tend to kind of inhibit them once they undergo the complete differentiation into mesenchymal program. So they need to have this dual personality where they could be epithelial or mesenchymal. So that's one of the area we are right now examining. So we have taken again several models, and if you take again syngeneic tumor lines with extreme mesenchymal, they are differentiated into mesenchymal. They lack the ability to differentiate back to epithelial. Under that scenario, they cannot develop metastasis even if they've undergone an EMT. Okay, so 67NR is one such example where the cells have undergone a complete mesenchymal transition. They're differentiated. There's another tumor model we use, something called T11, which is developed by Jeff Rosen's lab here in Baylor. They've become a mesenchymal transition. They also don't develop metastasis from the mammary fat pad. But if you put them in the blood, inject them directly to the tail vein, they do develop metastasis. So you need to be in the middle, midway, not right or left.
Mani, we have a question in the chat box. This is from Iris Sivatson. I'll read out the question. What are the CSC numbers with respect to metastatic versus non-metastatic cells? And they get reduced with chemotherapy, but then they need to come back for normal cell functions. So your response.
Thank you. I think so. Initially we thought that it could be just a simply stem cell number, but then later we realized that it's not just a stem cell number; it's a stem cell quality. So there's a guy with the name Max Vicha who's at the University of Michigan. He found that the stem cells in a tumor they tend to be less plastic. In other words, he isolated stem cells with a CD44 and something called ALDH, and he found that the stem cells which are more of a tumor stem cells are tumor initiating stem cell. If you remember, any tumor in order to grow, they need a stem cell, but those are tumor initiating stem cells. And there are stem cells which are capable of developing metastasis. They acquire something more than just a tumor initiating stem cells. So the tumor stem cell number may be different between different tumors, but the quality more than quantity of these stem cells are different between metastatic and non-metastatic tumors. And the chemotherapy definitely works, depending on the chemotherapy, works on both the they don't work on stem cells. So that's a problem. On top of that, chemotherapy can induce inflammation, and inflammation can cause EMT and make more stem cells. So most of the time when patients are treated with the chemotherapy, they tend to left with stem cells rather than differentiated cells. Hope that answers the question.
Mani, finally, I have another question from my colleague. This is Purusharath Rajaguru. Is it possible for tumor cells to release some factors to trigger platelets to produce CXCL4?
Good point. I think it's a very good point. We don't know what those factors are. So one of the things we've done is we just got a new mission instrument called a Rhapsody, BD Rhapsody by BD. So what it does is you can sequence single cell, both the immune cell as well as tumor cell. So we are sequencing as we speak all these 15 syngenic line plus patient tumor tissue at a single cell level, hoping that would yield us that information what you just asked. And we are also taking all these tumors, performing something called a Luminex assay. What it does is that it looks for cytokines specifically produced by these tumors at a greater extent. So hopefully these two will help us identify those factors what you just mentioned, Dr. Munyappa. Thank you. Really.
Well, Mani, I think I should wrap up in the interest of time. So thanks again for telling us your very, very interesting work. I hope we'll have opportunity to meet you and talk to you more in detail sometime in the future. And more importantly, thanks for joining us with all your the weather conditions and pandemic and all that in Houston. All the best.
Thank you so much, and thanks everyone. It's a pleasure. Thank you. I really appreciate the opportunity.
Well, now I have great pleasure indeed in introducing my very good friend, Professor Dhanasekaran. In the way of introduction, Professor Dhanasekaran obtained his PhD in 1984, working with Professor N.R. Maudgal. And after very successful postdoctorals at Madison and Denver, he established his independent lab at Temple University in Philadelphia, and very rapidly was promoted to the full rank of professor. Well, about 10 years ago, or over a dozen years ago, Professor Dhanasekaran took up a leadership role as the director of the Center for Cancer Research and concomitantly as Samuel Roberts Nobel Foundation Endowment Chair in Cancer Research, both at the University of Oklahoma. I'm going to embarrass him with a little bit now. Well, that Danny has a very high reputation of thinking outside the box and is very fearless in questioning established dogmas. So much so that has resulted in him running a very vibrant and robust research programs focused on understanding self-regulatory mechanisms in somatic as well as in endocrine cells. In particular, his pioneering work has substantially expanded our knowledge on the roles of G alpha 12 and G alpha 13, often referred to with the acronym GEF oncogenes, in signaling pathways in normal as well as in cancer cells. Now, I would like to invite Danny. I believe he is going to talk to us about, in continuing this theme, the bidirectional signaling in the tumor microenvironment. Danny, it's all yours.
Thanks, Muniappa. Let me, can you guys see me? Hello? I can't, okay. Thanks, Muniappa. I think, I had to, it's really a real pleasure and honor to give a talk here. And I think, one of the things I recalled when everyone was talking about their experience in IAAC, one thing I recalled was, I remember the book by Clinton, Hillary Clinton, that, it takes a village to bring up children. So like that, I always felt a successful career not only depends upon the lab you're in and also your peer group. And I had a good, I was fortunate to have good friends, and most of them are in the meeting today. And I think I can recall one incident because at some point, I was thinking about different careers when I was in IAAC. I think one of my good friends advised me, and rather, I had a counseling with him, and he suggested that, I should continue on biomedical research. And without him, probably, I wouldn't be here talking about science at this point. And that is, Dr. Muniappa. So I'm really, I should be grateful to him for being here as well. So today I'm going to talk about, I'm going to share the screen now. I'm going to talk about some of our recent work, as well as I'm going to tell, where we are going with that, with some of the recent findings we have got. And okay, so hold on a minute. So the talk I'm going to give is bidirectional metabolic signaling loop in tumor microenvironment. Dr. Mani, in fact, previously he talked a little bit about some of the interaction between tumor cells and immune cells. And our lab is primarily focused on looking at one of the signaling pathways, like Dr. Muniappa mentioned. We're very much interested in looking at the G protein signaling pathway because that is our vantage point. And one of the things what we realized was some of the pathways we were working on almost for 2 to 3 decades that cross-talked with the pathway that was going on in ovarian cancer cells. So then recently we started looking at some of the tumor and the other cell interactions. So if you look at the tumor or if you look at the cancer tissue, it's not a single type. That notion is long gone because you have multiple cells which acts in synergistically as well as kind of symbiotically to form invasive tumor tissue. So if you look at that, they always have multiple communication pathway. In fact, it is a multiplex pathway involving cancer cell and other resident cells in the tumor environment. So in fact, if you can look at it, most of them are carried out by some of the cytokines and chemokines. And we were primarily interested in the cancer fibroblast or normal fibroblast that is converted into cancer fibroblast to assist the cancer cells to move around as well as to sustain themselves. So if you look at the hallmarks of cancer, you can see there are at least 10 different hallmarks have been identified. And if you look at the cancer-associated fibroblasts, I think they almost regulate all the different aspects of the hallmarks of cancer. So we're very much intrigued to find out how these normal fibroblasts are converted into cancer fibroblasts and how do they function and how do they the phenotype of the cancer-associated fibroblast is maintained all through the cancer development and growth. So if you look at the cancer-associated
fibroblast, I guess you are showing a slide. But we are not seeing any of your slide.
Oh, really? I'm sorry. I'm sorry. Okay. Can you see it now?
Yes.
Okay. Okay, so I think, so what I was trying to tell was, if you look at the association or, the symbiotic interaction between the cancer cells and the fibroblast cells, which are present in the cancer microenvironment, they are almost involved in regulating different hallmarks of cancer. Almost all the 10 different hallmarks of cancer is being regulated by or assisted by the cancer associated fibroblast to maintain the cancer phenotype. And our focus was to identify how the cancer-associated fibroblast is differentiated by the cancer cell. Because if you look at the cancer-associated fibroblast, they can originate from different cell types. Primarily, we were interested in how the normal fibroblast that is present in the tumor microenvironment, how do they get converted into cancer-associated fibroblast, and if so, how does their phenotype being maintained in the cancer tissue? So if you look at that, there are at least, there are several, studies that have been done in different cancers involving TGF beta as well as PDGF alpha and beta. That is involved, the tumor cell derived, these growth factors, in fact, convert the normal fibroblast into cancer fibroblast, and the cancer fibroblast in turn communicate to the normal fibroblast. So what we were interested was to look at how is the metabolic crosstalk goes on between these cells. If you look at the cancer-associated fibroblast, they do undergo a functional shift, that is from oxidative phosphorylation, they undergo the glycolytic shift, so that there goes that aerobic glycolysis goes on. So you have the lactate is produced, that lactate is being uptaken by the cancer cells. So the cancer cells can go through, again, they also undergo aerobic glycolysis, so they can start building their, the building blocks for cancer cell growth. So we were kind of interested in this aspect, how they are involved in this kind of interaction. Our focus was on ovarian cancer because I think one of the major problems of the ovarian cancer is there's only less than 60% of the patients survive more than 5 years, primarily because the early diagnosis was very difficult. And also there is no anatomical barrier to prevent the intraperitoneal metastasis. In addition, there is no targeted therapy unlike in other cancers. So kind of, we were interested in exploring the ovarian cancer, the context, to see how we can identify some of the vulnerable therapeutic target, especially the ones involving the cancer cell and the fibroblast cell interaction. So one thing which we observed was that we can grow the cancer cells and we can take the conditioned medium and then incubate the normal fibroblast. We use MRC5 lung fibroblast and look at what happened to them. And in fact, in at least by fifth day onwards, the normal fibroblasts are converted to cancer fibroblasts as indicated by this cancer-associated fibroblast marker. In addition, that also induces the functional transformation of the normal fibroblast in terms of stimulating the glycolysis in these cells where the oxidative phosphorylation goes down. Once we had this data, we started wondering how or what kind of the components in the cancer, the conditioned medium stimulate such the differentiation. So if you can look at the ovarian cancer, one of the major growth factor that is produced in the ovarian cancer is lysophosphatic acid. This lysophosphatic acid, even though initially it was identified as a platelet-derived factor, this has been now known that almost many of the cancer cells do synthesize lysophosphatic acid, and they do activate a cell surface membrane protein or membrane receptor that are coupled to G proteins. And that's where our interest comes in because we have been working on G protein coupled receptors for a long time. And the LPA has been shown to be involved both in the proliferation as well as migration of cancer cells, including ovarian cancer. And also one thing we found was in ovarian cancer, you can see that the ovarian cancer patient develop ascites and this whole peritoneal cavity is filled with acidic fluid. In fact, if you look at the lysophosphatic acid levels, the lysophosphoric acid level goes very high in this acidic fluid. In fact, we could measure them, and I can show you one of the slides. So what we did was, since there is a lot of LPA in the tumor microenvironment, in the ascites, that is bathing this ovarian cancer tissue, we looked at whether LPA induces any of the growth factors that could stimulate the differentiation of cancer-associated normal fibroblast into cancer-associated fibroblast. When we looked at it, we can see so many different cytokines, including IL-10, IL-6, that are being upregulated by LPA that has been known as candidates for inducing the differentiation of normal fibroblast into cancer-associated fibroblast. Then, once we knew that some of these ones in red, they're all known to be involved in the differentiation of normal fibroblast to cancer-associated fibroblast. Then we started asking the question whether you really need these growth factors to initiate the differentiation, or whether the LPA, which is present in such a high amount in the acidic fluid of the patients, this is one of the data we have that show the LPA concentration in different patient-derived acidic fluid. So we started looking at the role of LPA in inducing differentiation in different, ovarian cancer tissue. In fact, we can also show that how acidic fluid also stimulates all these cytokines, which we have shown in the LPA-induced the ovarian cancer cell condition medium. So then we looked at the metabolic shift as a functional parameter to identify the functional differentiation of normal fibroblast into cancer-associated fibroblast using the CHARS system. So when we looked at it, what we could show was when we stimulated the normal MRC5 lung fibroblast, we can see that, again, just with the LPA alone, we can show that they are getting differentiated into the cancer-associated fibroblast as indicated by the marker. In addition, we can also show that the LPA stimulates the glycolytic shift in this MRC5 fibroblast. In fact, we can use the inhibitors for LPA to show that this effect is specific to LPA stimulated system. And also another thing I should point out here, that while the differentiation takes at least 3 days, this glycolytic shift, that is functional differentiation, that happens within 2 to 3 hours. That means we thought the functional differentiation precedes the morphological differentiation of the fibroblast into cancer-associated fibroblast. Once we had this data, we really looked at, instead of looking at the normal fibroblast which is derived from lung cancer, lung tissue, we used the transition of ovarian normal fibroblast, which we obtained from one of our collaborators in MD Anderson. Here also we can show that when we stimulated with the LPA, we can show that the markers for cancer-associated fibroblast goes up, showing that LPA could differentiate the normal fibroblast in the cancer-associated fibroblast. And also we can show that the same functional transformation in terms of glycolytic shift, that is being inhibited by the inhibitors of the LPA, showing that this effect is specific to LPA. Once we had this data, the question is, okay, the LPA stimulates the normal fibroblast into ovarian cancer, in the cancer-associated fibroblast in ovarian cancer. And they also undergo a functional differentiation, that is in terms of, going for glycolytic shift. Now the question is, what happens to the fibroblasts that have been already converted into cancer-associated fibroblasts? Whether they still require LPA to maintain the cancer-associated functional phenotype. In order to do that, we again looked at the cancer-associated fibroblast from patients and looked at whether the LPA can stimulate glycolysis and whether it could be inhibited by the LPA receptor antagonist to demonstrate the specificity of the LPA effect. Again, you can see that when we stimulated the ovarian cancer-associated fibroblast, this is different from the last experiment which we used in a normal fibroblast from ovarian tissue, and this is a cancer-associated fibroblast. Again, we can show that when you are inhibiting the LPA, your glycolysis, the glycolysis of this cancer-associated fibroblast goes down. So what it means is that in addition to inducing the differentiation of normal fibroblast into cancer-associated fibroblast, LPA, which is derived from the ovarian cancer cells, which is accumulating in the acidic fluid, in fact, is required for the maintenance of the functional phenotype of the cancer-associated fibroblast. Once we had the data, we started looking at the mechanism involved in the pathway. So in order to do before doing that, we also wanted to check, as I was telling you, we have seen that, the acidic fluid contains a lot of LPA. Now we want to know whether the acidic fluid, the LPA which is present in the acidic fluid, is really involved in converting the normal fibroblast into cancer-associated fibroblast. In order to do that, what we did was we took the normal fibroblast both from the lung as well as from ovaries and stimulated with the LPA, stimulated with the acidic fluid, this time not with the LPA, just with the acidic fluid, but we used, the LPA antagonist, LPA receptor antagonist. Then again, we can show that, when we treated the acidic fluid with the LPA receptor antagonist, the glycolytic shift is downregulated, suggesting again that the acidic fluid itself, that the LPA which is present in the acidic fluid is the causative factor that induces the differentiation of normal fibroblast into cancer fibroblast, cancer-associated fibroblast in ovarian cancer. So then, again, we want to demonstrate that is also required for the maintenance of cancer-associated fibroblast functional phenotype. Again, we did the same, set of studies, but using cancer-associated fibroblast cells. So again, we can incubate the cells with acidic fluid, but treat them with the antagonist for LP receptor. We can show that there is an increase in the acidic fluid, the glycolytic rate increases. When we treated the acidic fluid with the LPA inhibitors, it goes down, suggesting that you do have the LPA, which is present in the acidic fluid, is involved in the activation of the functional phenotype, even in the cancer-associated fibroblast. So what it means is that the LPA, which is present in the acidic fluid, it is not only required for the differentiation of the normal fibroblast into the cancer-associated fibroblast, it is also required for the maintenance of the phenotype of or at least the functional phenotype of the cancer-associated fibroblast. So meanwhile, we also had a study which we already published showing that LPA could induce glycolytic shift in ovarian cancer cells through a pathway involving different G protein, which is known as GIP2 oncogene, which is G-alpha-I2, and that activates the RAC, and the RAC activates the NOX2 pathway, and that involves the generation of reactive oxygen species and the activation as well as the increased expression of HIF-1 alpha. That stimulates the hexokinase pathway. In that way, it induces glycolytic shift. So then, we wondered whether the same paradigm involving HIF-1 alpha is also involved in the activation of the CAF differentiation in the fibroblast to CAF differentiation in ovarian cancer. So what we wanted to do, show us whether indeed the LPA can stimulate HIF-1 alpha, like in the cancer of ovarian cancer cells. So when we can show that, when we are incubating the cells, whether it is a normal fibroblast or cancer-associated fibroblast, stimulating with the LPA indeed increases the levels of HIF-1 alpha, suggesting that, this pathway may be functional in the fibroblast as well. Then when we looked at the HIF-1 alpha, what we did was that we took the normal fibroblast and treated them with the silencer expression using siRNA to HIF-1 alpha, then looked at whether the LPA-induced glycolysis goes on. So as you can see here, when we use the siRNA to HIF-1 alpha, that inhibited the glycolysis, suggesting that the differentiation of this pathway involves the HIF-1 alpha, which is induced by the LPA. Likewise, the same thing happens in CAF140 CAF cells also, suggesting that you require both for the normal fibroblast conversion into CAF phenotype as well as CAF phenotype to maintain its functional differentiation. Once we have this data, then the paradigm we have developed now is that you have ovarian cancer cells and the LPA, which is generated from the LPA receptors, in fact, stimulate HIF-1 alpha, and that involves, the functional conversion, the glycolytic phenotype or CAF differentiation, and this transition occurs very early in the development. And finally, you have the morphological differentiation of the CAF with the CAF phenotype. Once we have this data, I think, so now what we are targeting is whether we can use the targets in this pathway to have a therapeutic approach for ovarian cancer that can target, both the compartment, ovarian cancer cell as well as LPA. However, one of the questions which remained in our mind was, how is this things are being maintained, or how is the CAF phenotype is maintained. So one of the things which has been recently published was that this lot of epigenetic regulation goes on in maintaining the CAF phenotype. So we have been working on some of the link RNAs that is involved in epigenetic regulation. So we started looking at whether there is a role for link RNAs in this pathway. So you have LPA, LPA stimulates this HIF1 alpha, so there is a functional differentiation as well as morphological differentiation. Then in the maintenance, at least in the functional maintenance, we know that the LPA is involved. However, we want to know how is this long-term maintenance of the CAF phenotype, presumably through epigenetic regulation occurs. So we looked at some of the link RNAs. I don't want to go into the detail, but we were looking at the intragenic non-coding RNAs and long intergenic non-coding RNAs, not looking at microRNAs. When we looked at the link RNAs, what we know is that they can do multiple functions. They can either quench microRNAs to elicit some functions, or they can bind to some of this polycomb repressive complex protein to silence some of the tumor suppressors as well as the genes that could be critical for the differentiation of normal fibroblast or maintenance of normal fibroblast in the CAF phenotype. So we did array analysis. First, we found out what is the link RNAs that acts as a key intermediate, or at least what is increased in the CAF compared to the normal fibroblast. One thing we observed was, I can go into all the details of all these things, but I'm going to focus on the XAST link RNA. We showed something like 2000-fold in the array analysis. In fact, we verified it in other cell types. The interesting things about XAST link RNA is this is known as X inactive factor. That is, when you have in human genome, one of the X always gets inactivated. That X inactivation actually occurs by the synthesis of XAST link RNA by the X chromosome that is involved in inactivation. So this XAST link RNA, in fact, binds to some of the PRC protein, coats the complete one of the chromosomes so that everything is suppressed in this X chromosome. So only one X chromosome is active. This is not known to have any role in somatic cell type, but what we found are any other role in cancer. But recently it has been observed that the XAST is overexpressed in some of the cancers, including ovarian cancer. So what we found was that this is being upregulated in the cancer-associated fibroblasts. In fact, the repression by this XAST involves several PRC1 and PRC2 complex protein. One thing I want to stress here is the ECH2 protein, which recruits a specific, methylases that, methylates and suppresses the gene expression. I want to kind of keep you, kind of aware of that, because next one, what I'm going to say is, when we looked at the XAST function, one of the things that has been observed, at least in cancer cell, is that the XAST can actually decrease the functional microRNA 101, primarily by binding to them and acting as a sponge. And this microRNA 101 targets the ECH2, which is a part of the PRC2 complex. So what happens is now this suppresses the microRNA, XAST, ECH2 level goes up, so the ECH2 can take part in suppressing some of the tumor suppressor genes. So we looked at this paradigm, which is known in cancer cells. Then we looked at whether this kind of levels are increased, this kind of things are going on in the fibroblast. In order to do that, what we did was we used the XAST overexpression system in the CAFs and looked at the microRNA that goes up in these cells. In fact, what we can show was when we, when we downregulate the XAST, you can see the upregulation of the MIR or microRNA 101. So what we are thinking is this 101, in fact, leads to the increase in EZH2, and that actually involved in epigenetic and aberrant silencing of specific genes that leads to CAF phenotype maintenance. Some of this data is, we are still, we are working at some of the finer points. So I'm not going to have much more on this at this point. So, but in summary, what we have is that we have a metabolic interaction between the ovarian cancer cells and the fibroblast that leads to the transition state and finally the maintenance of the functional differentiation of CAF. And then what we also have data is the XAST mediated CAF phenotype is involved in terms of the maintenance of the phenotype by this XAST link RNA. So in summary, what I can say is that we have found the critical role for the LPA in terms of inducing differentiation of normal fibroblast into cancer-associated fibroblast in ovarian cancer. We have also demonstrated the role of LPA in functional differentiation involving CAFs, and also we think that this is a new paradigm involving XAST link RNA in phenotype maintenance through the epigenetic regulation of some of the genes which we are trying to characterize at this point. One of the interesting things which we are developing in the lab at this point, which I don't have much time to talk, is that since this pathway involves the HIF1 alpha, what we can do is we can target some of these pathways. So in fact, there are several FDA-approved repurposed drugs that can actually target the LPA receptor to HIF to the glycolysis. So we are targeting several of these to see the efficacy in treatment of ovarian cancer. The good thing is that it can target both the cancer-associated fibroblast as well as ovarian cancer cells. With that, I'll stop and I can answer some questions. But before that, I want to acknowledge and these are all the people in my lab who are involved in the work, and I do have some clinical collaborators both in OEHSC as well as MD Anderson Cancer Center. And in addition, I also serve as a visiting professor in Seoul National University and Novara University in Italy. So I think some of these collaborations, we are looking at the different population, how the ovarian cancer etiology differs in different geographical geography in all these countries. With that, I'll stop and I can answer some questions at this point.
Thanks, Danny. That is a very fascinating set of findings. I'm sure you'll have some questions. Any questions or comments for Danny?
I just have one quick question.
Sure.
So excellent talk, Danny. Excellent. So the question I have is, as you know, LPA can modulate both Th2 as well as CD8 positive T cells. So have you looked at the relation between the two, either using an experimental setting or in a patient setting?
When you were talking, actually, I was thinking about it because we have a large set of data because we characterize all the cytokines that can be induced by LPA. So I thought, once I heard your talk, and there was a question on the role of platelet, how the platelet derived factors. I thought I should talk to you later and find out because we have a whole you are doing RNA-seq and we have RNA-seq data as well as the bioplex data and some of the cytokines and chemokines induced by LPA. So I think we can look at it carefully. I'll definitely I'll get in touch with you afterwards. Thanks, Maria.
Thanks.
Yeah, Danny, there is a question from Kumar Somasundaram in the chat box. I'll read out. Ovarian cancer has different stages based on tumor size, lymph node spread, and metastasis. Different stages have different survival outcomes. Could you please explain the role of CAFs between different stages?
I think so. What we are thinking at this point, because our vantage point is from LPA. So what happens is the early stage, this pathway is not fully functional because it involves a different set of G proteins. And in fact, we have a paper coming out showing some of the gene signatures of the early stage that primes the cell for some of this, the later stage. Primarily what I am talking now about this HIF1 alpha pathway, that comes at the late stage 3 and stage 4, because that is the time when these the cooperation between the ovarian cancer cells and the calves occur the most, at least in the ovarian cancer. In fact, the initial glycolysis happens much before that, but I think this pathway becomes more pronounced at the later stages.
Well, Danny, I think we have to move on. Thanks again for your very interesting talk, as always. Thanks again. All the best.
Thanks, Munipa. Thank you.
Well, we have to move on, and the last speaker for this session is Professor Suraj Bhatt. I'm indeed very happy to invite him and also introduce to you about Professor Bhatt. Professor Bhatt obtained his PhD in 1975 working with late Professor J.D. Padiati, and he subsequently moved to NIH to do his postdoctoral studies. At NIH, Professor Bhatt has the distinction of introducing the then nascent field of recombinant DNA technology to vision research through cloning of human delta crystalline. In early 1980s, Professor Bhatt moved westward to California to begin his independent research at UCLA School of Medicine, Los Angeles, where he has stayed since then. He is currently the Oppenheimer Brothers Endowed Chair Professor in Ophthalmology. Throughout his postdoc and as an independent PI, Professor Bhatt has made a number of conceptually important contributions to the field of crystalline research, and most notably demonstrating the catalytic non-structural functions of crystalline both inside and outside the lens, and most recently connecting or establishing the relationship between crystallines and Alzheimer's disease. Without further ado, please join me in welcoming Professor Bhatt to make his presentation. Professor Bhatt, all yours. We can't hear you. I think unmute your. Professor Bhatt, we can't hear you. Kindly unmute. Professor.
Okay.
Now you're okay.
Now
Yeah.
I can unmute myself. Yes.
Yeah.
Hi, George. Dr. Muthu Krishnan. A lot of people I recognize on the panel here, and it's just a remarkable excitement to be here this morning. We are going away from cancer into a totally different arena, and I would also like to remind you that I'm the last speaker, and I can keep you for the rest of the night here. So it's a pleasure. It's a real joy. I'm very excited to be talking with you. I just want to right away thank Subaru here in San Francisco, who told me about this terrific symposium and asked me would I speak there, and I want to thank Dr. Rangarajan and Dr. Rao for making it possible for me to be part of this symposium. Let me share my screen with you, and so that we can start our So can you see the slide? Do you see the slides?
Yes, sir, we can see.
Can you see the slides?
Yes, sir, we can see the slides.
Oh, terrific. And you can hear me?
Yes, sir, we can hear you.
That's terrific. Oh, that's fantastic. Okay. You know, I connected with Sandeep yesterday and made sure that I could communicate with you with the slides and all that. But obviously, it worked, and now I'm trying to find out where yes, it is here. Everything is here. Okay. Let me take you the very first slide here. This is the first molecular biology laboratory in the Department of Biochemistry at the Indian Institute of Science, which was headed by Dr. Joseph Podiati. You see Dr. Joseph Podiati right here, and 5 of his students who received PhD under him. This is Srivatsan, Suraj Bhatt, Shivakumar, Ramakrishna, and Sushil. Sushil and me, I submitted my thesis first, and then I think a few months later, Sushil submitted his thesis. We basically graduated actually at the same time. Being in this lab was extraordinarily meaningful because, at least in my case, our life has been shaped by what we did in that lab. And my mentor and my guide, Dr. Padyati, had everything to do with it. It's a big emotional time, but let's go to the next slide. The title of the talk here is Spatial Determinism in the Lens Transcriptome. Introduction to the single cell analysis in the ocular lens. Now, before I really go into it, I want to recognize Rajendra K. Gangulam, Dongjae Kim, and Raj K. Kashyap, the 2 phenomenal undergraduate students. Rajendra Gangulam has moved on to a scientist position in Gilead by now. Professor David Alashoff and Shinkai Zhao helped us with the statistical analysis. Sergey Mungul is a computational biologist and helps us with understanding data and its manipulation. And Dennis Mark is a phenomenal mathematician who talks about quantum computation, electron entanglement, and human consciousness. And when he gets some time out of that, he draws us to the interface between biology and mathematics. And the most important discussions that we always have and have not resolved is about noise in biology and noise in mathematics. He thinks they are same. I think they are different. I think there is no noise in biology. There's a causal element in biology. Something does change and then something happens. But you will see that in what I'm going to talk about in next 15, 20 minutes. The work that I'm going to present is supported by NIH grants in my lab and funds that the Department of Ophthalmology receives from Research to Prevent Blindness. I have a feeling that a lot of you must have seen this landmark paper from Cole Trapnell and Jay Shandure's lab from Washington University. This was published, I think it was published in 2019. What you see here, what these authors did is they took a mouse embryo at 9 to 13 days of gestation. They homogenized the whole embryo. They put all the cells on the fax machine and isolated cells and sequenced 2 million cells. Once they sequence 2 million cells, they get the RNA single cell data from the RNA sequence analysis. And then they do a T-distributed stochastic neighbor embedding computational program, which simply takes cells which express similar genes with similar numbers. That's all. So you have a cell type which expresses gene X, and you have a cell type which expresses gene Y in a similar fashion quantitatively. And then we get this abstract presentation because we know what those gene markers tell us. We get connective tissue progenitors, we get jaw and tooth progenitors, we get hepatocytes, we get cardiac muscle. what we also get is lens. That's what attracted my attention here, because they found that there are cells, there are clusters. Cluster number 37 makes crystallins. Now, crystallins are proteins which generate transparency in the ocular lens. So you wonder, what are crystallins doing in a dark developing embryo? You will immediately come to understand that they don't act as crystallins possibly. They possibly are doing some other catalytic phenomenon. But then it gives me an introduction to what I am going into the next slide. But if you look at this abstract phenomenal piece of work, what you find is there is something extraordinarily missing in this whole thing. And that is the constraints of time and space are amiss in this whole phenomenon. Development, tissue phenotypes are critically dictated by the constraints of space and time. And it's in that persuasion that we talk about the lens as to how the location, the spatial presence, as well as the time as when the gene is expressed and where that gene is expressed is very critical. And therefore, we go to the next slide. I have simply downloaded this picture from the web, and I wanted to orient you to tell you where the lens is in your eye, because most of us don't think about the eye or the visual system until we are around 80 years of age. This is extraordinarily made tissue. Here is the lens in the anterior of the eye, and I want to point to lens epithelium, which is right one single layer of epithelial cells which faces the world. The phenotype of the lens is transmission of light into the eye, activate the retina, and that initiates vision. But what kind of light do you want the lens to transmit into your eyes? It's not any kind of light. This light has to be focused, and if this is the retina, it has to have a single pinpoint focus. It has to have absence of scatter, and it has to have absence of spherical aberration. You cannot focus here or anywhere. That happens in some certain diseases. But most importantly, in this fish lens, this biological contraption with millions of years of evolution, what you see here is quite remarkable. And that is, if you look at this laser beam, it goes through the center of the lens and hits this focus. If you look at this laser beam, it has to bend in order to reach that focus. If you look at each one of these laser beams from here and from here, you find that the laser beams bend only so much so that the light reaches a single focal point. That is made possible primarily because of a gradient of refractive index from the surface of the lens to the center of the lens. If you cut this section, this lens into two here, this is what you see. You see a capsule, this red thing here, and you see this epithelium here, the anterior epithelium, which covers the lens, which I talked about in the last slide. It is from the differentiation of this epithelium. Here you get nascent fibers. This is also known as the bow region. Nascent fibers, the cortical region, this is equatorial region, cortical region, and the nuclear region. You can see all these fiber cells are put together in such a fashion. Here, if you cut a transverse section, you find an hexagonal array. There is no space between the fibers because that is conducive to allowing light to transmit without any scatter. I was talking about the bow region here, and that's here. The first thing that you start thinking about the lens is there are only two cell types, lens epithelium right here, and lens fiber cells, and this goes from anterior to the posterior side of the epithelium. What happens with this epithelium is morphological transformation from simple cuboidal cells to long slender fiber cells, which go from anterior to the posterior side. And this process of differentiation puts these nuclei elongated into the anterior of the lens. These nuclei eventually degenerate. That's why this picture, it gives it a picture of the bow, and we call it a bow region. Now keep yourself on this bow region, and you will see that this transition from the epithelium to the fiber cells is not only morphological, it is accompanied by tremendous molecular transformation. It makes a huge number of proteins, crystallines. But keep yourself right here and go to the next slide. This is the bow region here. And if you go from bow region to the inside of the lens, you start seeing there is a gradient of refractive index. For me, the fact that there is a gradient of refractive index is associated with protein density indicates to me there is a genetic control of this refractive index. If you look at this lens in a whole, you have at the edges of the lens low refractive index, the visual axis or the center of the lens, very high refractive index, and low refractive index at the other edge. Now, what are these proteins that are made in this transformation? This is an old slide. There are ubiquitous crystallines and taxon-specific crystallines. We will not get into that. Yours truly was involved in growing some of these things. But the ubiquitous crystallines are alpha crystallines, beta crystallines, and gamma crystallines. Alpha crystallines are heat shock proteins, and they basically make about 30% of a human lens soluble protein. Gamma crystallines is what we are going to focus on because these are the proteins that have been reported to be present in the center of the lens. It is this gamma crystalline in the center of the lens and beta crystallines in the cortex of the lens that's got us thinking. Until we found about a decade and a half back, the whole perception that all proteins have the same refractive index was negated, was proven wrong. Yes, most of the proteins fall in this Gaussian distribution, but alpha crystallines, beta crystallines, and gamma crystallines fall on the tails of this Gaussian distribution. What we call the refractive index increment, dn by dc. The n, the refractive index, is simply how much does the light bend. N is here c divided by v. C is the speed of light in the vacuum, and v is the speed of light in the medium that you are looking at. Gamma crystallines, alpha crystallines, beta crystallines are on this side of this curve, which suggests they have high refractive index. This got us going because high refractive index and its association with specific proteins must indicate that every fiber cell of this ocular lens, every fiber cell of your lens is an optical unit which dictates how much the light has to bend. And if you want to see clearly, it has to hit that one single focal point. So there must be a genetic determination to how do you regulate protein expression, both quantitatively as well as qualitatively. This got us excited almost 2 decades back. And we wanted to find out where is the control. So we randomly picked up cells from here, equatorial, cortical, and nuclear regions, and did single cell analysis. We isolated single cells, one at a time. These are two cells from the equatorial region. These are two cells from the cortical region. These are two cells from the nuclear region. This is a simple PCA analysis. It looked very good, but when we started looking at the genes, I simply want to run through and tell you that none of those pairs of these fiber cells have same kinds of genes. At least 30 to 50% of the genes are very different in each cell. So what would happen at that point? For those of us who thought the fibers are homogeneous, they make proteins, same proteins everywhere, we suddenly started doubting the techniques that we are using, and particularly making libraries. Those of you who make cDNA libraries, you know it's more of an art than science. And we could introduce a lot of abnormalities and artifacts in this whole thing. So we decided that we can't stay with single cell sequencing. We need to do something more quantitative. We therefore developed a method, which I am not going to go into, wherein we could isolate fiber cells from the equatorial region, from the cortical region, and from the terminally differentiated fiber cells. You see, differentiation goes this way. We don't know what are the neighbors of fiber cells, but we know where the fiber cell is coming from. This is published now, so I'm not going to go into the methodology of having done that. So we took these fiber cells. Please don't give up on me. This is a very simple slide. We took 500 fiber cells, and those fiber cells are numbered here, which you cannot see at all. And we interrogated 500 fiber cells only with 100 genes, not 20,000 genes as you would do by RNA sequencing. We picked up all the crystallins, these little purple dots you see here, and which is 17 crystallins and about 80 genes. So we interrogate every fiber cell with those 100 genes in a multiplexing system, which is the biomark fluidigm system. Every pixel here shows you the number of the fiber and shows you the gene that is expressed in there. What you see here is the red, which is high expression of crystallins, and you see them here. And you also see very high expression of non-crystalline proteins, but it looks like that they don't make crystallins. It's as if in the absence of crystallins, these fiber cells compensate by increasing the refractory index, by increasing the concentration of these proteins. Now, how do we know where we are in the lens here? We have already tagged these fiber cells. We call these cells red when they come from the equator. We call these cells cortical when they are green, and we call these cells blue. We tag them. And here is the blue. This is predominantly a blue cell association here. This is predominantly nuclear cells. This is predominantly equatorial cells. So you can basically look at the number of the fiber you are talking about and get the whole gene expression story with respect to 100 genes. What I want to indicate here to you is, for example, HSF4, it's expressed only here. You don't see it anywhere else. If you look at HSPB2, it is expressed in these cells. If you look at HSPB6, these cells also express HSPB6. If you look at HSPB1, it's expressed in cells which don't express HSPB6. If you look at ITGB1, it's expressed in cells which don't make HSPB6. If you look at IGF, insulin growth factor, it's made in these cells. It's not made in these cells. So you can keep on looking into it, and you will find that there is tremendous heterogeneity in every fiber. So therefore, our earlier assessment that there is tremendous heterogeneity in these fiber cells was correct. It was not an issue with making cDNA libraries, although I still feel making cDNA libraries from millions of cells, it's hard to keep them on one specific plane. So there have to be artifacts introduced in there. But then we looked at this whole data together and simply did what's known as PCA analysis, principal component analysis, again, a computational technique which makes clusters of cells These are green cells, these are blue cells, and these are all mixed cells, predominantly green. And we can go back to that matrix and tell you this is cluster 1, which is basically green, and this is cluster 2, which is basically nuclear. And we can look at genes, and here are a couple of examples. We can look at transcription factors, and these are on the x-axis. These are numbers of fibers. You can compare cluster 1 with cluster 2 with cluster 4, cluster 3, and you can keep on looking, and you don't need statistical analysis. You can look at PAC6 and you say that PAC6 from 1 to 3 cluster has reduced in expression by half almost. Similarly, in HSF4, it's predominantly expressed in cluster 1, not in cluster 3. You can then go from these genes and go to the proteins. CRY-AA is the alpha-crystalline I was talking about. Alpha-crystalline A and alpha-crystalline B, the world knows that they are only part of one single protein. But we indicated a long time back that they are not one single protein. They are 2 different proteins. And here, there are cells which don't make any alpha-A crystalline, but there are cells which make alpha-B crystalline. You know why people said this, because the cluster 2 makes both alpha-crystalline A and alpha-crystalline B. You can go back to the transcription factors which regulate these proteins and look at them in the cluster 1 and look at them in cluster 3, and you see these differences. You can get tremendous amount of information. But more importantly, I want to go to this matrix again and take all of this data and tell you we simply did a Pearson correlation analysis with it. And by doing Pearson correlation analysis, which is simply global analysis, which associates one number with one more number, it is not causal in any way. It is simply developing a relationship between one is highly correlated and -1 is poor correlation. This is cells from the equatorial. We did the whole matrix, all cells together, but I'm not going to show you that because that's what we see there also. And we go to the cortex, you see a group of proteins. It's not important which are these proteins. I'll come back to that. You go to the nucleus, here is this group of proteins. Here it's not correlated at all. And this group of proteins, when you look at by way of crystallins, equator, cortex, and nucleus, all the crystallins go up and come down, go up and come down, except this set of genes which keeps on going up. Now, is it statistically significant? Yes, because every other gene goes up and comes down, goes up and comes down, except these genes. How do we know where these genes are? We again want to reemphasize, they are this whole commitment to make those proteins is made right here in the equator. That commitment is retained in the cortical fiber cells. Then it is enhanced in the center or the visual axis of the lens. We go from here because it is enhanced in the visual axis of the lens, because these gamma crystallins and alpha crystallins, they have higher refractive index. Now you don't have to make huge amount of protein because they already provide you high refractive index. You can avoid aggregation, and that is the motivation the evolution possibly has to keep your visual axis clean. And if we simply take this data of the expression of these proteins and put them superimpose on the ocular lens, you find this Pearson correlation here in the differentiating fiber cells. Cortical cells, same crystallins are maintained, their expression is maintained. Nuclear cells, their expression is enhanced. We can simply come to one specific conclusion, that the expression of specific crystalline genes in the visual axis is because of spatial determinism. That spatial deterministic expression was started right here and maintained because somebody knows somewhere that we need these crystallines right in the middle of the lens because we need to be able to see. I'm going to stop here and entertain any questions that you may have. I want to thank you for your presence here and for listening to me, and I will stop here. Thank you.
So questions or comments for Dr. Bhatt?
Hello, may I speak?
Yeah,
Sure.
please.
Sir, very nice talk. So actually, I have 2-3 questions from your first slide. You showed that there is a crystalline gradient forms in the lens. So that is already well known. So I want, and here also this gene expression changes also quite comparable to that gradient. So my question is that what controls actually this gradient? And second question is that you have told about spatial determinism. So how the cells know that, okay, it is now in cortical region or now it has to move to the nuclear region? So what is the molecular mechanism of that?
I don't know your name, but
My name is Shobik.
Shobik, that is a billion-dollar question. I wish I could tell you what initiates spatial determinism. You see, at the single cell level, it is very difficult to associate phenotype with the transcriptional component in the cell, except in the lens, because there is a refractive index, which depends on the proteins, and not only simply proteins, but proteins of special kind. So when I move from here all the way down here, there has to be a specific feedback loop that tells the cells that these proteins have to be increased in expression. I did not tell you the whole story because that will take, I think, 12 more hours. But this area is highly heterogeneous. The cortical gene expression is the most heterogeneous gene expression in the lens between the equator and between the nucleus. Cortical gene expression is very variable. We think it is in the cortex of the lens that a manipulation is introduced which allows these cells to progress towards terminal differentiation. We have no idea what's the shape and form of that input. It will be terrific to know, but I think this is the beginning of an example in biology of spatial determinism in a eukaryotic tissue. And it is dictated by the interaction between a physical component, that's light, and that's refractive index, and the gene activity. How is that manipulated? You know, you will be hearing, I think, Dr. Bullendel, Tom Bullendel, I don't know whether he is listening to this talk at all. He was the first person who did the X-ray crystallographic structure of the gamma crystallines. And I was in Columbia University when I cloned the first gamma crystalline, and we found out from X-ray crystallography, it should have only 165 amino acids. And when I got my clone with 174 amino acids, I had to reclone it because I didn't know whether that was the right sequence. You will hear from Dr. Blundell possibly about gamma a little bit, but we think refractive index is not simply an isolated amino acid related phenomenon. It is related to the structure of the protein also. And therefore, that feedback from the protein and from the structure of the lens dictates how the progression of differentiation takes place. I can keep on talking about it without giving you anything substantial.
Actually, I want one more doubt on this part. So about the manipulation. In one of the slides, you showed that the cells, when they go to the nuclear region, they get devoid of all the cellular components. So that I feel that manipulation should be done before their, and one of the first organelles, if I read properly, one of the first organelles that gets removed is the nucleus. So is it true that actually the manipulation is happening very early and actually cell knows from the Bow region itself that it has to go to do something in the cortical region, or it has to make a particular crystalline?
We will be able to address all of these questions if once we find the neighborhood of every fiber cell, and we are planning on doing that. But more critically, very importantly, if you remove the nucleus, that takes a scattering principle out of the cells, you see?
Yeah,
Light has
correct,
to go.
correct.
So you take the nucleus out, so you don't have scatter. You take the organelles out, you take out the scatter. But not only that, you must adjust the membrane refractory index with the cytoplasmic refractory index. So there is tremendous transformation of the membrane once the cells move on to the nucleus. We don't know the whole scheme of things, but we know there is determinism at the molecular level, which must conform to the morphological level. We have no idea how does it happens. What I think we'll have to find out neighbors of these fiber cells,
And I
that
have
will
one last question is that
No, I think, so Sobik, I
okay,
think
okay.
there is a, I will
Okay,
come
sir.
back
Okay,
to
sir.
you, okay? Here is a question, Dr. Bhatt, in the chat box from Puduru Jagadishwaran. Do you think a similar mechanism is operating in the lens of the compound eyes? Your response.
Jagdishwaran, hello.
Yeah.
It's good to hear from you. I think in eukaryotic biology, there is determinism is extraordinarily important. Why? Because a tissue has to perform a function. And determinism is, we see it, but we can't prove it. It's very difficult to prove it. For example, in heart, in kidney, in muscle, in brain right now, I heard a talk last week from Allen Institute. They have exactly started taking these gene products and putting them on single cells in particular areas in the brain, in the hope of correlating transcription to the phenotype of a single cell. Yes, I think in the compound eyes also there is determinism. There has to be determinism because we are dependent on the final phenotype, which is transparency, and we need to see. You must keep in mind that in primate evolution, the evolution of the vision is extraordinarily critical, which develops very early on. If you don't let focused and formed light hit your retina, the visual cortex does not develop in your brain. So there is already a selection that has gone, what we all have gone as human beings through, which has removed most of the obstacles and most of the mutations. That's why eye seems to be so perfect. So the short question, the short answer to your question is yes, there is determinism there, but it will be dictated by the data that once you go into single cells there, because again, the refractory index is very critical. And the refractory index possibly could be generated by huge number of proteins.
Well, that's very engaging, Dr. Bhatt. Thank you very much. I would like to wrap up this session and thank all the speakers for joining our celebrations of the department and also telling us about their very, very interesting work. You must all know that we're very proud of you, all the speakers in this session.
Thank you so much.
Now I'll turn over to the organizers if there is any business part they would like to take care of.
Thanks a lot.
Thank you so much, sir. We from our end would like to thank all the speakers of this session for sharing their work and memories of IISc. I would also like to thank Professor K. Muniappa for chairing this session. Finally, I would like to hand over to Kshitija for today's concluding remark.
They have taken over. I don't know.
Thank you, Mani. On behalf of the entire biochemistry department, I thank all the speakers for sharing their work with all of us. All of the talks have been absolutely intriguing and stimulating. We also thank session chairs for coordinating the meeting so smoothly. We hope to host you in large numbers tomorrow too at the same time. Do follow us on Facebook and Twitter for regular updates on recent publications, events, and recognitions in the department. With that, we'll call it a day. Good night to all the participants from India and a good day to all our attendees abroad. Thank you.